Good morning to all dignitaries, guests, and delegates with great joy and immense pleasure. We feel privileged to extend our warm welcome to all presented here for the International Conference on Consortium of Universal Research Erudition, iCURE, organized by Madurai Kamaraj University. Tamil Thai வாழ்த்து. Good morning to all dignitaries, guests, and delegates.
நீராறும் கடலுடுத்த நிலமடந்த கெளிலொழுகும் சீராறும் வதனமென திகழ் பரத கண்டமிதழ் தெக்கணமும் மது சிறந்த திராவிடனல் திருநாடும் தக்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இத பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Welcome address. I invite Dr. N. Shankar, coordinator of iCURE, to welcome the gathering.
So very pleasant good morning to one and all present here. So first of all, I would like to welcome our Honorable Vice Chancellor, so who is instrumental for this iCURE conference. Actually is one of the brainchild of our Vice Chancellor. So initially this iCURE actually was initiated to have a interdisciplinary research programs in Madurai Kamaraj University itself. So just to make the scientists from biological sciences, biotechnology, chemistry and physics to collaborate with each other for an interdisciplinary studies. And when we had a discussion with this, so he was the one who introduced and just the international. And again, we want to have this we have to disseminate this knowledge to all over, and also to bring people from various fields to Madurai Kamaraj University, and also to interact and to initiate the interdisciplinary collaboration. So first of all, I would like to welcome our Honorable Vice Chancellor, Professor J. Kumar. Welcome you, sir. Then our respected Registrar, I welcome you, sir. Then I welcome our Chief Guest, Sri M.V. Muthuramalingam, sir, Chairman, Vellamal Educational Institutions. I welcome you, sir. So then, I welcome Professor Ulrich Pfeffer and Scott Kramer for this wonderful event today morning. And I welcome all the distinguished professors, eminent speakers who is going to present for this the next 4 days, and especially the participants, especially the student participants. I welcome you all for this wonderful conference, and I hope you will have a very good knowledge share in various disciplines of biological sciences, physics and chemistry. And again, I welcome the media and all my friends for this wonderful occasion. Thank you very much.
Light fights away the darkness. Light symbolizes knowledge and wisdom. I request the galaxy of intellectuals, honorable guests, to lightning the Kuthuvilakku. I request our Honorable Vice Chancellor, Professor J. Kumar, to honor our chief guest. I request our Vice Chancellor to honor Sri M.V. Muthuramalingam Sir, Chairman of Velamal Educational Trust. I request our Vice Chancellor Sir to honor Professor Ulrich Puffer from Italy. I request our Vice Chancellor Sir to honor Professor Scott D. Kramer from USA. Thank you, sir. More than 30 years of teaching and research experience, published over 160 research papers, and guided 25 research scholars, well-known scientist, and the great administrator, Vice Chancellor of Madurai Kamaraj University, Professor J. Kumar. Sir, we invite you to deliver the presidential address.
Vanakkam, my dear and most respected Professor Maradamuthu, the former Vice Chancellor of this great university, Professor Dharmalingam, Professor Velathambi, Dr. Parsardi, Professor Velraj, the former Vice Chancellor of Bharatiyar University, Professor Balasubramanian, my colleagues, syndicate members, and dear participants. First of all, it is our honor and privilege to welcome the chief guest, the chairman of Velamal Educational Trust. He had been the most inspiring person to many of us who are around this region. He had been most fondly supported by Dr. Kalam. Kalam admired him very much, as many of you would have heard. On many occasions, Dr. Kalam was associated with Thiru Muthuramalingam. And this very hall stands in the name of our eminent former president. Kalam is a testimony to the inaugural function. I also would like to join in welcoming all of you once again on behalf of this entire team of faculty members, scholars, students, the other office bearers of this university. Ever since I took over, I had a very series of meetings with all the faculty members, and I realized that there has been a legacy in this university. Though you do some homework before you take up a position, I did do some homework, but at the end of the day, when you are in a situation, you have to only evaluate the reality. So, in order to understand what can be done to move forward, I discussed with almost all the faculty members in the initial couple of months, and I realized there has been a follow-up of the legacy of the erstwhile Bhatnagar Award winners of this campus. As many of you know, there is no other university to our knowledge which has produced so many Bhatnagar Award winners, and that being one of the highest achievements for any scientific person of this country. It was a great beginning to keep this team intact, and that was the conceptual reason for initiating this program. Many of you may ponder upon as to what is this title, because any conference of any nature has a title which is mostly either scientific or artistic, or let's say arts, discipline, commerce, etc. This title was very popularly coined by our colleague in this campus, Dr. Shivakumar, the immediate registrar who had to be away for his own health reasons, but he coined this term, and I'm very proud to say this term is very, very seriously applicable for all the conceptual thinking we have had in order to promote excellence in this campus. The campus, as all of you know, had many disciplines of active research and teaching. So to say, there was a lot of Tamil work at the beginning years, and subsequently there was a lot of scientific activities. And many of the scientific activities in this campus, as like in any other university, was always very unique in its approach, and they had been trying to accomplish. And on one such occasion, I had the opportunity to, initial days, as I say, I had the opportunity to inaugurate the immunology activities, the society, Indian Immunology Society activities. At that time, I realized that there was a student from other state. He was from West Bengal. He volunteered to speak on that occasion, and we just accepted it. And then he went on to narrate how this university, in particular the department associated with biological sciences and biotechnology, has evolved. He said, he narrated it in such a dramatic way with all the historical facts. I don't know if even Professor Dharmalingam would remember or can recollect all those facts that the boy narrated, but he was so nicely narrating every chronological event which evolved and how people interacted from across the country. People came from all distances to interact with the faculty members and the researchers of this university. Once again, I have a great sense of pride in standing before you, because this university has a long way achieved a lot of things, and it will continue to march forward. There are very many unique things which are happening in the recent days. Just as I recollect a few of them, I would like to say, there has been some patents, and among the patents, one of the patents which will take forward in a very nice way is the oxygen breathing system, just a small little mask which has been triggered by a circuit so that the outside air is kind of filtered, and then it allows only an adequate amount of oxygen to go inside the human system. And this in turn is the filtering mechanism as also a nanoparticle which will enrich the oxygen, and therefore the oxygen only be let inside. To believe it, I had a visitor from North India, a young guy, and then I just asked him, can you please use this mask and say if there is anything different for you? And then to my surprise, he said, sir, I am a marathon runner. He came to collect his certificate for the distance education course he had. And then he said, give me a 5 minutes time, I'll run around the campus and come back and tell you. So he ran around with the mask, and then he came back and said, sir, I am very happy that you have given me this great opportunity. He said, with the mask, he feels that he is able to run very normally instead of having any breathing issues. So that was the one demonstration of this particular patented product. We would like it to be to be handheld by some of the industries and then taken forward. Similarly, there was another program which I was very fond of, which Professor Chandrasekhar, the chairperson of the School of Biological Sciences, has evolved over the years. It is a portable kit funded by an ICMR project, and in this kit, he is able to identify the dengue-causing mosquitoes, and he has the project in order to access the real mosquitoes along the banks of the Vaigai, and therefore this kit is essentially enabled by artificial intelligence by another person who had no idea about artificial intelligence, but he learnt it on his own, and then subsequently he has evolved this portable sensor cum identification tool in order to identify the mosquitoes. The beauty of this is that the mosquito frequency is very clearly evaluated, and thereby there are other methodologies which are enabled to identify whether the mosquito is belonging to the category of dengue causing or otherwise. There are several such success stories. I would like to continue on, but for want of time and for the sake of the program, I just had mentioned a couple of them which are very fond to me. But however saying, an university cannot promote only products, and as like Professor Kaliraj and others have been very solidly saying, we need to promote an activity of fundamental research and then subsequently translational research. And in this concept, I felt that there are some areas which are very unique to this society. For example, this city of Madurai is more popularly known as Kadambur based on the number of Kadamba trees which were here. And one of our faculty members, who was also the syndicate member, Professor Nagaratnam, took me to a place where there was one Kadamba tree. Many of you may get surprised. If somebody is so fond of something, there is something unique about it. He took me to a place and said, sir, we'll have to keep it very secret. We should not disclose it to everybody. Maybe they will just cut this tree and take it away. This has been happening in the recent times. That's the reason he was very carefully mentioning about it. In this Kadamba tree, there are three categories. One such category is very rare or becoming extinct. And so there has been another research scholar from Coimbatore, Bharatiyar University, who came all the way to classify these kind of trees which are in and around. And then subsequently, one of our faculty has taken it on herself to have this tissue engineering enabled growth of this Kadamba tree, though this researcher from Coimbatore has brought some seeds. But we would like to try this on a methodology of scientific nature. And so this is one fundamental research which we want to translate as to be a development for the society, not for the sake of bringing out a product. In similar nature, there are a couple of things. One is, once again, a recent approach to genetic understanding of various species, in particular those excavated samples. Under the RUSA scheme, Professor Kumaresan has published a Nature article in identifying some of the genetic characters of various excavated samples. And we have built a BSL-3 lab in order to very carefully analyze not only genetic related issues, but also other species which are to be very carefully analyzed. We have been approached by the government of Tamil Nadu to establish a research park in line with what has been done for Coimbatore under the leadership of Professor Kaliraj at Bharatiyar University. We have been requested to identify a land, and we have been asked iCURE is not just a conference, so to say. We have a plan in the anvil. Very specifically, we have a program of a larger magnitude funding, and we seek the support of all the presentees, in particular the invited speakers and the learned audience. We would like you to guide us in this initiative. It is for the sake of not only the Madurai Kamaraj University, we want to strengthen the activities of research and subsequently innovation and evolutionary ideas. Yesterday I had the opportunity to talk to my longtime friend Dr. Arun Balakrishnan, who is running a company, and he said in his early days after his postdoc, to get into academics was one of his early dreams. And subsequently, he left academics and then joined an industry by name Prinamal. And then later on, he has built another company. And he said on the walk of his life, he has realized that students and scholars have to be motivated, have to be guided in order to innovate and then incubate a product, rather than just making them as another researcher, another scientist who feels happy towards publishing. But on the contrary, every idea has its own views, and Professor Scott had a different thought when we were discussing, and he said academics is also equally important, and so therefore fundamental research has to be very carefully supported. So with all this thinking in mind, it's not about a couple of people, many of them are informing us at various occasions how to evolve. So this is a great opportunity for the university and in particular for the young scholars who will take forward the society. I would request all of you to be very carefully following this entire 4-day event and come up with your own plans or methodologies to promote the university and also get networked with the society. I will be failing in my duty if I don't thank all my colleagues, particularly those who had been running around for more than something like 6 months to make this event a successful. I am very cautious not to mention the names because if I leave out any particular name, that would be doing the biggest injustice. However, I am very much indebted to all of them because I had run many programs. I know what is the running around for any such event. So I would like to thank each and every one of them. And once again, I take great pleasure in welcoming the chief guest along with my colleague registrar Dr. Sadasivam and Dr. Shakila for this inauguration program. Thank you all and I welcome you all.
iCURE has always been your dream. Today, that dream has come true. Thank you for your presidential address, sir. Now, I cordially invite Dr. K. Sadasivam, Registrar of Madurai Kamaraj University, to release the abstract of iCURE. I request the dignitaries on the stage to receive the copies of abstract. Born in Ladanengal, small village at Madurai, his service mind and keen interest to provide education to the society made him to dispose his own family property to promote the Velamal Matriculation School. His contribution towards educational sector includes establishment of many number of kindergarten, matriculation schools, international and residential schools, CBSE schools at various places providing high quality of education. It has not stopped at the school level education. His service has been extended to the health sector, establishing world-class medical college and hospital and servicing the needy. Sir, mere words would not be enough to describe your service. I cordially invite Shri M.V. Muthuramalingam Sir, Chairman of Velamal Educational Trust, to deliver the inaugural address.
Honorable Vice Chancellor of Madurai University, my dear friend Dr. Kumar, former Vice Chancellors of various universities, learned professors, my dear student friends, well-wishers, I convey my sincere thanks to all of you for having invited me today this very big program today. First of all, I congratulate the Vice Chancellor for having organized this program on time. Many of the institutions where I visited, I will be irritated by not starting on time. Today, the famous Vice Chancellor in the university It is well organized, the band, and started on time. So I should congratulate. Second thing, it talked about the relationship with Abdul Kalam sir. Everyone says that Abdul Kalam sir asked to have a dream. I differ from it. He asked all of us to have a dream which is not be achievable. Not able to achieve it. I am one among it. Even though I was in a small village without any family background, I had a dream when I was in 8th standard and wrote in my notebook to buy a Plymouth car which was famous at the time. Now I have bought many cars, costly cars, made it true. So far, I am not good in studies. I was not good in studies. I failed in SSLC by means of not able to produce 2 marks. At the time, 24 marks is the minimum mark per pass. In any one of the subject, I produced only 22 marks. I failed it. Even then, I had a dream to go higher and higher. I started a school in a small way near Thatcher set in 1986 with 183 children and 13 staff members. Now I am very proud to say that I have more than 1.6 lakhs of children as well as 15,000 students in various institutions, including engineering colleges and medical colleges, so on, without any family background of my own. The self-made. What I want to emphasize here, the research. Even though I was not able to operate, I have 3 cell phones, latest cell phones, but I'm not able to operate actually, only on, off. I can't send a message. I can't even send a mail. I don't want to spend much time for all these things. My friends are helping me to do it. But my focus is to take our institutions to the height further and further. In this aspect, I request our Honorable Vice Chancellor to associate with us because we have started a research institute and Sahila and other professors have come forward to help me. We established a lab there, brought some of the research scholars from Aravind Eye Hospital. I started doing a small way, but after having come, I think that I should involve more and more. And I promise that Vice Chancellor need not run pillar to post. Even my trust will be able to sponsor some amount of for doing research. It should not be for Namakevas. Many of the programs are conducted to satisfy the officials or the ministers. So after some time, miserably, it failed. This should not be the case. Because I conducted so many conferences in Chennai because of my engineering college there. But after the conference, I lost my friends. You have presented the papers. How you to put it in action? That is most important thing. Conducting a conference and this type of program, it should be useful to the society, converted as a project. That project should be useful to the needy people. So even I told Sahila to take a case in our medical college. The Elanarain சொல்லுவாங்க இல்லையா இப்போ சின்ன பிள்ளைங்களுக்கே வருது இளநரை வருது நந்த பின்னால் பண்ணுறோம்ல இதை ஏன் நம்ம பண்ணக்கூடாது செனட்டிக் இப்போ மீத்து அதை ஒரு பண்ணுங்க ஓஜியிலேருந்தே அதை அனலைஸ் பண்ணிவிட்டு குழந்தையாக இருக்கும் பொழுதே அது ஹெரிட்டரியாக வருதா என்னன்னு சொல்லிவிட்டு அதை அவாய்ட் பண்ணுறதுக்கு இது பண்ணணும் அப்படின்னு நாங்கள் நினச்சிட்ருக்குறோம். ஏன்னா இது நிறைய ப்ரோக்ராம் செய்யணும், நிறைய பண்ணணும் அப்படின்னு ஆசைப்படுறோம். அதனால தான் என்னமோ வைஸ் சான்சலர் என்ன கூப்பிட்டாங்களோ என்னமோ தெரியாது. பட் அது மாதிரி இன்னொன்று என்னென்னு கேட்டால் ரிசர்ச். I invited the former owner of Saravana Bhavan, Mr. Rajagopal. He was not a learned person, but he did a research of making this paniyaram into various items and idlis, so-called idli variety and all, by means of his research. Saravana பார்த்தீங்கன்னா க்யூவில் நிற்போம். இப்போ இல்லை அந்த கடை. அப்போ அவர் எங்கள் காலேஜ் கூப்பிட்டுருந்தேன். எப்படி அவர் ரிசர்ச் பண்ணிவிட்டு, ஏன்னா பணியாரம்னா கேவலமாக பேசுவாங்க நம்ம ஊரில், என்ன பணியாரத்தை போய் வச்சுருக்காங்கல்ல, அதே பணியாரத்தை க்யூவில் நின்று வாங்கி சாப்பிட்றது இருக்குது. வெரைட்டி ஆஃப் பணியாரம்ஸ், வெரைட்டி ஆஃப் இட்லிஸ். எதுக்காக சொல்லணும்னா ஒரு சாதாரண பலகாரத்துக்கே ரிசர்ச் அவ்வளோ வேண்டி இருக்கும் பொழுது நம்ம மாதிரி லேர்னர் பர்சன் இருக்கும்போது நிறைய நம்ம ரிசர்ச் பண்ணணும். அந்த ரிசர்ச் பண்ணுறதோட பேப்பர் ப்ரெசென்ட் பண்ணதோடு இல்லாமல் புக்கு போகிறது இல்லாமல் இது மாதிரி கான்ஃபரன்ஸ் பண்ணுறதோடு இல்லாமல் அது சொசைட்டிக்கு எப்படி யூஸ் ஆகும் அப்படிங்கிறத நம்ம பண்ணணும்னா இது மாதிரி நிறைய கான்ஃபரன்ஸ் பண்ணிவிட்டு நிறைய ஸ்காலர்ஸ் இப்போ தே ஹவ் கம் ஃப்ரம் இட்டாலி as well as USA and also many eminent professors have come, vice chancellors have come. All of us should join together and take this program, not only the conducting of these 4 days, and put it in practice, involving my student friends. And I request all my student friends, research is a must. The chairman has come to this level now by means of commitment, involvement. அடுத்து என்ன பண்ணலாம்? I am not a doctor. I am having more than 450 doctors and many JRs in our hospital. So, you need not be a qualified person, but if you have a commitment, if you involve yourselves fully, then you can actually do something to society in this aspect. I should thank all actually the Vice Chancellors as well as the faculty members and heads of departments of our Madurai Kamaraj University, my student friends, for having invited me for this program. I convey this should be a successful one. Do the best. Thank you very much.
Thank you, sir. Your speech and of course, sir, yourself have inspired us. Gratitude is the fairest blossom which brings from the soul. I now invite Dr. H. Shakila, Coordinator iCURE, to propose the vote of thanks.
Good morning, everybody. Now we are in the very important session, because after this the program is going to start, and all the stalwarts who are seated here are going to deliver this respective speeches also. So may I take this opportunity first of all to thank our Vice Chancellor, who was the brain behind this iCURE, which is nothing but the International Consortium of Universal Research Erudition. And this is the platform which is going to bring all lots of research activities at MKU. So I thank you, sir. I also thank our chief guest, Dr. Muthuramalingam, sir, who is the management trustee and also an educationist, an academician, industrialist. So he was again a very inspiring person, because right now we have heard his inspiring and thought-provoking speech also about his story, how he came out and how he developed. Thank you so much, sir. And we also have to thank Velamal Management Trustee, because they have given funding also for us to bring about this particular conference. So even for that, sir, we are thanking you so much. And I take this opportunity to thank our registrar in charge for bringing about all the administrative activities, and also the syndicate members who are here to come for this function. And I take it is my honor to thank our Professor Marudamuthu, our former Vice Chancellor, and also Professor Kaliraj, Vice Chancellor of Bardiyar University. They were all our, I should say, you were all our mentors, sir. So we are so, we are so, we are so happy, and there are no words to thank you, to make yourself free and come and grace this occasion, sir. Thank you so much, Professor Marudamuthu, sir, and Professor Kaliraj, sir. And also I take this opportunity to thank our stalwarts of MKU, Professor Dharmalingam, sir, Professor Veluthambi, sir, and Dr. Arun Balakrishnan, and then Dr. Parthasarathi, all the stalwarts who had been our alumni and also representing Madurai Kamaraj University always, as and when we ask them and we call them. And so now they have come here to grace the occasion. So thank you very much, professors. And I also thank all the funding agencies, starting from Velammal and DRDO, Indian Immunology Society, and SERB. So they were the major funding agencies who had come forward, and they have helped us to bring about this occasion. And I particularly, I should take this time to congratulate and also thank all the participants. Till now we have 230 registrants. So may I now take this opportunity to thank all the participants and the registrants. And I also take this opportunity, a special opportunity, and also special thanks to the guests who are seated here, Professor Kalmer and Professor Ulrich. So thank you so much for gracing this occasion. Now I just wanted to take some time because about this conference, I would like to say a few words because we are having 6 plenary lectures and more than 6 countries. People are coming here, so we have invited speakers from more than 6 countries, and there is going to be 36 invited lectures, and also apart from that, we have our own MKU faculty members. They are also going to give lectures, so we have 25 lectures of that. And so all together, put together, it comes to, I think, you'll be having fully with all the sessions of scientific sessions covering biological sciences, biotechnology, physics, chemistry, computer science, everything, all sorts of what our Vice Chancellor was mentioning. So this is going to be a platform which is going to involve many interdisciplinary sciences, and hence all the sciences, I mean schools of sciences, we have come forward to take this conference into a bigger activity. So may I now take this, and I'm so opportunate enough to thank all the members and all the faculty, all the participants and students and volunteers who have come forward to bring this conference into a great deal. So with this, I thank the organizers for giving me an opportunity to propose the vote of thanks, and I also thank everybody. Thank you so much.
சார் வாங்க. நான் வர சொல்றேன். ஆமா நீங்க ஸ்டார்ட் பண்ணுங்க.
Thank you so much, dignitaries, for inaugurating this international conference. And now we are initiating the technical sessions. I request the participants to stay here. Within 5 minutes, we will start the technical session 1.
Sometimes air conditioning Check, check. Hello, hello.
I request the participants to stay back. We are going to start the technical session 1.
A warm greetings to all. We are about to start the sessions. To start with, the first session of the day is on molecular therapeutics. As known, it is the promising technology which is potent enough to lead into a new era of individualized molecular medicine, where we can treat patients with prescriptions based on their genetic makeup. To get enlightened more into the topic, I cordially welcome the chair of the session, Professor Dr. G. Kumaresan, School of Biological Sciences, Madurai Kamaraj University. Along with him, the co-chair, Dr. S.B. Anand, School of Biotechnology, Madurai Kamaraj University, to carry over the session.
Okay, thank you. Yes. Thank you. Good morning to everybody, and I would like to thank the organizers and especially the Vice Chancellor, Professor Kumar, and Professor Selvamani and Professor Nyanya Kumar for organizing this and for inviting me. It's a great honor for me to be here. It's my second time in India and I like it really a lot. Today, given the context of multidisciplinarity, I try to give an introduction to prevention, diagnosis and therapy of cancer, the state of the art and the next challenges, and the contribution that cancer genomics has made to that field. Next slide, please. So technology undoubtedly is driving the progress in medicine, especially in the last 30 years. But can our understanding of disease processes keep the path with the technologies that is evolving. So with other words, does ever more data really mean ever more insight? And I won't give an answer to that question, but we should keep in mind this question when we do cancer genomics. It's not only technology. At the end, we want to come to insight into disease processes. Next slide, please. So what I will do today is I will give a short history of cancer genomics and speak about the power and the limits of genomic technologies, the application of genomics in the clinics, and upcoming omics. And then I will go to advances in cancer therapy, and I will talk on targeted therapy and immunotherapy, the two instances that have determined a real change, paradigm change in cancer therapy. And at the end, I will talk a little bit about tumor prevention, a field that in my eyes on targeted prevention, and I will make a, let's say, a real daring hypothesis on immunoprevention, a new concept. Next slide, please. So the molecular classification of cancers essentially will identify molecular classes. In the case of the low risk, we have the follow-up in no therapy or general therapies. In the higher risk class, we need response prediction because we have different drugs available, and we must decide which drug, which therapeutical approach is the best for the patient, depending on the molecular class. And at the end, molecular classification, molecular characterization of tumors can also identify new targets and allow to develop new targeted drugs. Next, please. So it all started in, oh, he told me I shouldn't. Okay, I try. It all started in my eyes more than 20 years ago with that paper from Thérèse Sorli. And the classes belong to estrogen receptor negative and estrogen receptor positive tumors. That was already known from the pathology. But there were also subclasses, subtypes, that were not known by the pathology, by the classical pathology. And these subclasses have a prognostic incidence. So they are clinically relevant. That was the proof of concept that molecular classification of tumors can actually be clinically relevant and can go further than classical pathology. Another approach shortly after showed essentially the same thing. You can see here, when you see this line, that are the metastases, and you see that the two classes identified have a different risk of metastasis despite lymph node positive disease. So in lymph node positive disease, you can identify a class that has a relatively low incidence of metastasis. And these signatures were supposed to be used in the clinics to decide whether or not to give chemotherapy to women with breast cancer of low risk of metastasis. So avoid chemotherapy. But actually, if you look what these signatures do, so we have here on the right side the intrinsic subtypes from Thérèse Sorli, and here we have the subtypes of the 70 gene signature. And if you look at proliferation, you have cases with higher or lower proliferation in both cases. And you can see that the low-risk cancers, which are depicted here in blue, essentially are in the different class, so are in the estrogen receptor positive class, those that are of low proliferative potential. Whereas higher proliferative potential, even in the estrogen receptor positive class, determines a higher risk of recurrence. And the same you can see for the the intrinsic subtype. So that essentially means that what we have done with gene expression profiling can be done by the pathologist by just adding a proliferation marker. So essentially there is not so much more information in genomic classification. And as a matter of fact, these approaches are used relatively relatively rarely in the clinical decision making. But what I would also like to show this application where you simply try to define a genomic grade. We have cancer grades 1, 2, and 3. And when you do expression profiling, so in the clinics, grade 1 is low risk and grade 3 is high risk of metastasis. And the oncologist is at unease with the grade 2 tumors because he doesn't know that whether it will develop metastasis or not. And so what you can see by expression profiling, that you can divide grade 2 essentially in a high risk and in a low risk population, with a high risk population being almost very similar to grade 3 tumors and the low risk to grade 1 tumors. And you can observe that essentially also the metastases that have actually occurred, the red dots here, behave in that way. So you could use expression profiling to define a grade, a genomic grade, and you would just have grade 1 and grade 3, no grade 2 anymore. So essentially, these technologies have not been adopted really by clinical decision making. That's mainly due, I think, that in the Western world we have an established pathology, and it will be very hard to introduce a completely different system. But if I were to establish clinical decision making in a situation where there is no type of classical pathology at the moment, I would opt for genomics because it can do the same, and it might do so with a much lower cost and with a much higher potential for centralization. But that's up to the authorities who decide on medicine. Eventually, in the years after, this was even more complicated when we introduced, together with expression profiling, also the copy number analysis. And you see that in breast cancers you have many, many copy number alterations, which are shown here for the 23 chromosomes. And if you look at the color of the bars, you see the Sorley intrinsic subtypes, and specific copy number alterations are associated with the specific subtypes of breast cancer. And if you combine the two information, you come up with a list of 10 integrated classes that rely on expression profiling and copy number alterations. So that's an even finer definition of molecular classes in breast cancer. But if you look at the Kaplan-Meier curves of survival, then you see that these classifications have a biological meaning, but they have no clinical application, because how could an oncologist decide how to treat with these 10 classes. It's clear that that's low risk and that's high risk, but all the rest is somewhat intermediate, and it's still difficult to decide whether you should treat by chemotherapy or not. So again, that's a more complicated and biologically meaningful classification, but it has no clinical application. We tried to investigate a little bit into the problem of misclassification, because the ideal signature, molecular signature, would divide the cancers in low risk, so that nobody gets metastasis, and high risk, where most of the women get metastasis. And that's not possible. If you look at what we tried, is simply to develop functional signatures based on cell-cell signaling, cell death, cell growth, cell proliferation, and so on. And what we observe is that we can always obtain two classes of differential gene expression. And if you look at the pink bars here, you see the metastasis in this group. And you see that the metastasis always tend to cluster in one of the two gene expression clusters, and much less in the other one. But you always have some cases that are in the low risk cluster. So some cases of misclassification. And if you take all these functional signatures together, then you get a very nice distinction between low and high risk, but still you have misclassification. And when we analyze these cases, they are have really low risk profile, and by all the signatures, the same cancers are misclassified. So that means that are low risk cancers, but they have developed metastasis. And that's the problem of misclassification derives essentially from the probabilistic nature of metastasis. It's a very complicated process, and it's probabilistic in nature. So any cancer, any invasive cancer, may form metastasis, and it will do so with a highly varying probability. So you can decide high risk and low risk cancers, but it will never be perfect. And that essentially depends on the fact that metastasis is determined by environmental facts, so the stroma and inflammation in the tissue, pre-existing facts like the cellular background and the genetic background of the patient, and stochastic elements that are the mutations, the transforming mutations and additional mutations. And essentially what influences most heavily the expression profile is the cellular background, mainly the cellular background. You get two clear clusters of estrogen receptor positive and estrogen receptor negative cancers because they derive from different cells, from luminal and basal cells. And the transforming mutations. But that's much less important because mainly in a given tumor, the many mutations that you can find and the many different driving mutations that you can find are in the same pathway. So at the end, it doesn't show up so much on the expression profile. So at the end, we can say even the best ideal signature will misclassify some tumors because it's a probabilistic process. In 2012, I edited this book. Don't buy it because there will be a new one in a couple of years. But that made a little bit the point of 10, 12 years of molecular classification of tumors, and there's a chapter for each tumor that has been analyzed in this way. What is important, especially you have seen that when I showed before the integration of copy number variation data and gene expression data, there is data science. We have so many data, and it's growingly important to analyze data in innovative manners. So I take here the example of uveal melanoma. It's a rare cancer of the eye. It's a melanoma, but it's very different from the cutaneous melanoma. And I take this example because it's a molecularly very simple tumor, and we know it very well by the time, even if it's a rare cancer. And it's my pet cancer. I'm much working on uveal melanoma. And we have, thanks to the Cancer Genome Atlas Consortium, we have much data on all the tumors and also on uveal melanoma. That are 80 cases that have been analyzed by DNA methylation, RNA transcription, and copy number alteration, and DNA mutations. As you can see, it's a simple tumor because it makes not so much mutations, and the mutations are highly prognostic, especially the BOP1 mutation. It's a tumor suppressor gene that drives metastasis in this cancer. And we started with this simple cancer because it's simple. You can integrate all these data, and then you get something like that, where you have the classification based on gene expression data. Gene expression data allowed the distinction of 4 classes, if you wish, or you can say 2 classes and 2 subclasses. And these subclasses and classes are related to outcome. And you see also that there are, here we have specific gene mutations, so it's related to gene mutations. It's related to copy number alterations and many other aspects. For example, also the infiltration of leukocytes, which is very heavy in one subclass that is of bad prognosis. Here you see the prognosis. The black ones are death from metastasis. That's data integration. It's all based on, the classification is based on a single domain, just on gene expression. So our question was, what if we would do classification taking into account from the very beginning all the genomic information that we have? So there are different aspects of data management. We have multi-platform, so you have, for example, microarrays and RNA sequencing, so there are different platforms to analyze gene expression. And we have multi-domain data. Multi-domain means we analyze DNA methylation, gene expression, and copy number variation. And data merging is simply where you merge different experiments, maybe with different methods, but on the same domain. So different data sets from gene expression, and you create one data set for one domain. So you still continue to work on a single domain, most often on transcription. Data integration, you have different experiments, different methods, and different domains. So you have gene expression, DNA methylation, copy number variation, and you create multiple data sets with multiple domains, not just one data set, because you keep the domains separated. And data fusion, on the contrary, where you have different experiments, methods, and domains, you create one data set that contains all the multiple domains, and you can do classification using this one data set. And that might be important because genomic aspects are not completely independent. So most often copy number alterations, so amplification of genes determines overexpression of genes. Most often, but not always. And for example, DNA methylation usually, but not always, reduces RNA expression. And obviously mutations can affect expression and methylation. So the question was, can we add power to classification by doing data fusion? And that's again uveal melanoma. And we have the Robertson subtypes, so that's only based on gene expression that I showed you before. And you see all the information on the tumors, BOP1 mutation and metastasis, and you see a nice distinction of cases with metastasis and cases without metastasis. So primary tumors that eventually develop metastasis or not. And you must always consider that if there is high risk, but it didn't develop a metastasis, that can mean it will do so. It hasn't developed metastasis in the period of follow-up. It might also mean it has responded to therapy. That's not the case in uveal melanoma because nobody responds to therapy in uveal melanoma. That's the big issue with uveal melanoma. So, and what you can see is that the data fusion classes, we decided to build 4 classes that you see here. They are related to the transcription-based classes, but they are not completely identical. But if you look at the prognostic power, it's not much different from the transcription-only classes. No. Oh, how do I go back? This one, okay. Okay. So we did, so essentially in uveal melanoma we had the information that data fusion, multi-domain analysis is not really superior to working just on transcriptomic data. But as I told you, it's a very simple tumor, and molecularly speaking, and it might be special for this type. So we went to, and we did that very recently, to cutaneous melanoma, which is much more complicated because if uveal melanoma is the tumor with the lowest tumor mutation burden, melanoma is the tumor with the highest, cutaneous melanoma is the tumor with the highest mutation burden. And we can obtain by data fusion approaches a classification of 3 classes. And what I'm showing you is the analysis of the classes that we obtain by data fusion on the level of transcription and methylation. So it's not classified by methylation or by transcription, but obviously the 3 classes that we define by data fusion are related to expression and to methylation. Especially we can identify the cluster 2 that is very different from the other 2, especially in transcription and methylation. And if you look at what that cluster contains, the genes that are differentially expressed in the class 2 tumors are mainly in immune-related, and essentially this type of cutaneous melanoma has a better prognosis. And it's important to note that here we are speaking about cutaneous melanoma before the introduction of immunotherapy, in most of the cases even before targeted therapy. So when prognosis was much worse than it is today. And this is an indication that still data fusion classification is very similar to single domain classification, transcriptional or demethylation classification, but it works and it might work better. So we are working on better algorithms for data fusion, and we will apply it to other tumors. Because the one lecture that we have learned from cancer genomics is that whatever you do, it will work for one cancer type, but not for all cancer types. And 2 words on what I call dernier cri omics. Dernier cri, that's a word we use in fashion. And in a way, genomics has become also a fashion. So apply the last technology is very useful to get your article published at a higher level. And not always we are so sure that we have really new information. But it's interesting. What we can do today is single-cell RNA-seq, so RNA sequencing to understand gene expression on the base of the single cell. So you have a tissue, a tumor, and you isolate and sequence single cells. So you barcode the RNA that is contained and the cDNA that is produced at the level of the single cell, and then you just sequence, and you get the number of sequences you obtain for gene 2, for example, in cell 1 or in cell 2, and so on. And you get the classical gene expression profile, but this time for single cells. And you can do further analysis to essentially principal component analysis to understand the different types of cells that you have in your sample. And then you can look at the level of the single cell whether you find cells that have a differential expression of specific genes that might make the difference. And we are applying this to cutaneous melanoma, where we try to increase the response to immunotherapy by adding epigenetic drugs, in this case guadocytabine. And this is essentially in press. So if you look at the methylation level, in the principal component analysis, you see that the tumors that are experimental tumors in the mouse, no? Because in the patient we cannot treat only with guadocytabine, because that would withhold the patients from more effective treatments. But in the mouse we can. So whenever there is guadocytabine or not, you see a clear difference in the principal component analysis, and as expected, the demethylating drug, it's an inhibitor of DNA's methyltransferase 1, you get undermethylation in the cases that have been treated with the drug. You get differential methylation. You can look at the genes that you obtain in differential expression. And again, what we see, it's mainly immune system genes, and that's encouraging because the real aim of the study is to show that you can revert T-cell exhaustion. So the T-cells that normally would recognize the tumor cell, but they become exhausted, they don't work anymore. Or you can also call it reinvigoration of T-cells. And that's why we go to the single cell level. And so far I can tell you that we see some differences at the level of T-cells quantitatively, and we are looking into the expression profiles to identify, because the real thing is that if you can touch the right gene in just a single T cell, that might lead to a proliferative burst in that cell, and that can make the difference, even if it occurs at the single cell level. So because treatment with epigenetic drugs affects each cell in another way, but it could occasionally, and some T cells, have an effect that the T cell starts to proliferate, an anti-tumoral T cell starts to proliferate and makes the difference, and that's what we are looking for. The next step is spatial genomics, where you don't get only the information on the single cell, but you also get the information on the position in the tissue, or even within the cell. So for example, if you are doing RNA sequencing, you can do that from a single cell, but you can record the location of this single cell within the tissue. You barcode and you sequence, you can identify the gene expressed in this cell. For example, here, if you get CD33 expression in this tissue, then you can say there is a myeloid-derived suppressor cell in this position. And this is done essentially by creating an overlay over the tissue section with a record of where which bead is positioned. It can be a microarray, or it can beads or polonies, which is kind of gel that is put on top of the slide, or microfluidics. Essentially, you need the special information that must be encoded in the beads that you are using, and you do again barcoding, then you do sequencing, and you get gene expression data, and you can relate these gene expression data to the position within the tissue section. And here's a comparison for mouse embryos. You can get, from different technologies, you can get a good representation of gene expression. You don't get, at the moment, you don't get whole genome data. You don't get 20,000 genes or 10,000 genes that might be expressed in the cell. You get something like 1,000 to 2,000 genes that you can observe. So that will help you, for example, if at the level of the tissue, you can build a tissue atlas, histopathology, you can decide what do the different cells with the different morphology actually express at the gene level. You can also do subcellular localization. You can say these two, if you do, for example, combined DNA and RNA sequencing, and you can have an interactome where you can say different molecules are close to each other. When it comes to the context, you can work on clonal heterogeneity that you find in almost any tumor, because you can see in a tissue section where the different clones of the tumor are positioned and how they develop. You can obviously get epigenome, so the special information on epigenetic modifications, the chromosome organization, and so on. You can do the same at the protein level. That's like as always, it's lagging a little bit behind the protein analysis, but it's possible. And you can do a kind of special screening if you do genetic perturbation, for example, by gene editing with CRISPR-Cas or something like that. You have many, and at the end you can study the temporal sequence and tumor evolution, the dynamics of the tumor. So there are many things to come, and it's certainly a technology that is extremely important for our biological understanding, but I think it will be of difficult application in the clinics as it is. So again, maybe there will be some tumor where special genomics or single cell transcriptomics can make the difference, and at the end it will be applied in the clinics, but it's not so likely that it will be applied to all tumors. That might also depend on the development of technology. So if it becomes more simple, and especially if it becomes less expensive, then it might find a wider application first in research and then in the clinics. And as said, as always, some specific situation will benefit from the technological development. Any revolution at the end turns out what is initially called a revolution, and say that with much emphasis, because in 2000 something, I started with microarrays in 1997, when they were still like that. And after the article of Thérèse Sorli, I was really convinced with that technology we will finally understand everything of tumor biology. And essentially we didn't. And most importantly, we didn't change much in the clinics. So any revolution at the end turns out to be a step forward for one or the other disease. But it's an important step forward. So let's come to the real important steps forward, the advances in cancer therapy. And I'm speaking on melanoma. I know that in India melanoma has not such a high incidence. It's not so important, but melanoma happens to be
Excuse me, Dr. Fulrich. So can you please complete in next 3-4 minutes, please?
One? Yeah. I have how many minutes?
So maybe another 5 minutes maximum.
5 minutes, okay. I should go a little bit further. That all the targeted therapy that is based on the knowledge of the molecular targets, and you develop drugs that go blocking the signaling pathway, and the experience in melanoma, vemurafenib, that blocks BRAF mutated cancers. There is a response, there's an important response, but is eventually the tumor progresses because it becomes resistant. Resistance depends on the molecular features of of the tumor, and there are many, many different targeted drugs being used in the clinics. And again, molecular classification is important for the response prediction because today the oncologist has to decide among many drugs for the same tumor, and he has to decide so on the basis of the molecular characteristics of the tumor. The real advancement is from immunotherapy, where we essentially today use antibodies that block negative signals on the tumor cell, that on the dendritic cell that blocks T cell from recognizing the antigen, and on the tumor cell that blocks the T cell, the prime T cell, to kill the tumor cell. And that was a Nobel Prize from 2018, and it makes a big difference, and that's not against no therapy, it's against a single immunotherapy, and that is double immunotherapy, where you see that it has really changed the situation in melanoma and in other cancers. And if you go into the adjuvant setting, you even get higher response rates, no? And that's a real paradigm shift in therapy for those tumors that respond. The problem is there are many side effects, and it is very difficult to predict response to immunotherapy because there are so many different aspects of the tumor cell and of the host that can determine failure of immunotherapy. So I would make the prediction that it will never come really to a clear prediction. And yeah, I said so immunotherapy leads to a long-term advantage, whereas targeted therapy doesn't. Immunotherapy must further be developed in order to increase the response rate and the duration of response. That might be new immune checkpoint blockers, so I showed 2 of them. There are others already in the clinic. T-cell therapy, CAR Ts, so combined antigen receptor T cells. Drug combinations aimed at reinvigorating immune cells, what we are trying to do with the guadocytabine. And maybe I skipped the part on prevention because I'm going out of time. So let me just show you my city where I'm working. That's Genoa in Italy, and that's my team. And that are my funders. Thank you.
So thank you, Dr. Ulrich Weber, for your wonderful talk on cancer genomics, like possible classification with RNA, single cell RNA seq and everything. And this is a wonderful talk. I'm sure that students might have inspired from this talk. In view of time, we'll just have the discussion after the during tea break or lunch break. So thank you very much.
Professor Selvamani, Anna University of Trichy, for introducing Professor Ulrich to our society. So in the midst, I would like to call upon Professor Kumaresan to deliver a small memento to the plenary lecture speaker, Professor Ulrich. So now we have a tea break, and we will be back after 10 minutes. So after 10 minutes, we will assemble here. We will have the plenary lecture 2. Then afterwards, we will have a parallel session at the back side of this CIC hall. So the chemistry participants are requested to engage yourself in the parallel session at CIC hall. Please be back after 10 minutes. Thank you.
signaling. And it's activated by IL-1, TGF-beta, toll-like receptors, etc. And it activates JNK, P38, NF-kappa B, and MAP3K7. AMP kinase, sorry. And it's involved also in trail-induced autophagy. And so keep in mind that these pathways now are going to be lost when you lose MAP3K7 expression. CHD1 is chromodomain helicase DNA binding protein 1. It binds to H3K4 trimethyl marks, so those are associated with active transcription. It regulates global chromatin structure. It helps to keep the DNA in an open transcriptionally activated state, and it's thought to help move the nucleosome down the DNA. And, oops, sorry. Oops, going the wrong way here. How do I go back? Okay. Let me see. There we go. Hit the wrong button, sorry. And it's also been shown to regulate embryonic stem cell differentiation, and it also, loss of it may contribute to genomic instability. So we have a kinase that's lost and a chromatin binding protein that's lost, that when the two are lost, it's associated with aggressive prostate cancer. So let me just tell you a little bit about the prostate gland. It's male specific. It's at the base of the bladder. It is a secretory epithelium that makes contributions to the seminal fluid. It's composed of a basal layer and a secretory luminal layer that makes the secretory products. It has in the basal layer an occasional stem cell, neuroendocrine cell, and then it's surrounded by a complex stroma. I won't go into those markers. During cancer, what happens is, here's another way of depicting that normal prostate where you have a basal layer and a luminal secretory epithelium. And during cancer progression, first there's dysplasia, or we call it also prostatic intraepithelial neoplasia, or PIN. And when you have low-grade PIN, what's happening is you start to get nuclear abnormalities. The nuclei are no longer basally oriented. They have prominent nucleoli, and the cells, as it progresses to more aggressive dysplasia, the nucleoli get even more prominent, and nuclei are heterotypic, and also there's it looks like this cell is just kind of floating in here, but if you actually look pathologically, there's a little thin appendage that still sticks down onto the basal layer. And as this progresses to cancer, what happens is you lose that basal layer, and now these cells are invading into that surrounding stroma. That, by definition, is cancer. And so we use that as a pathological indication in animal models, whether you have a tumor or not, if you lose that basal layer. So my lab, many years ago now, developed a mouse prostate epithelial stem cell culture system, and basically what we can do is grow epithelial stem cells and then in culture, and then we mix those with fetal urogenital mesenchyme. And so the mesenchyme has, during development, has the inductive signals that drive that stem cell to differentiate and undergo and promote ductal morphogenesis. And so what we can do is we can take this fetal urogenital mesenchyme, mix it with our stem cells in a little collagen button, and then graft it under the renal capsule in a mouse, and then wait a period of time. Anywhere from 6 to 12 weeks is our typical experiment, and then you get a graft under the renal capsule. And then you can look pathologically at what that structure is. And so, and if they're normal cells, what we'll get is that normal glandular phenotype of a luminal cell and a basal cell and secretory products. And our hypothesis was, at this time, that if we had a tumor suppressor gene, if we manipulated that tumor suppressor gene, it would alter the pathology of that tumor in a way that might be aggressive. And so what we did was we knocked down with shRNA TAC1 or CHD1 or the combination of those in our stem cells and carried out a tissue recombination. We call this tissue recombination because we're recombining the mesenchyme with the epithelium, and then let that go and asked what did the phenotype of those glands look like. And I wouldn't be telling you about this if it didn't work, but so it did. So if you have the control cells, you get a normal glandular structure. You have a, it's hard to see here, but you have a luminal cell that's making this secretory product here, and you also have basal cells. And we validated this with specific stains for specific markers for these. If you knock down CHD1, you get mostly a benign looking structure with an occasional low-grade pin. If you knock down MAP3K7 alone, you also mostly get dysplasia and high-grade pin in there. So there's bunching of the cells. It's hard to see in these images, I know. But if you knock down the two of them, you get high-grade pin and invasive carcinoma. So here's an example of a cribriform pattern, high-grade pin. So you have these sort of spongy-like looking structures. It's still benign because there's still a luminal layer, but the cells now are, so here's these are the same, these were taken at the same magnification. I've kind of manipulated them with Photoshop, but so you can see that this doesn't have all of those honeycomb shape as in here, and actually in here we have bona fide invasive carcinoma. So the loss of just these two genes promotes invasive carcinoma in this mouse model. I don't know what happened to this figure, but it's not coming up. Yeah. Well, so we, I'll just tell you that these. We also did a human prostate cancer cell line and manipulated MAP3K7 and CHD1, and what we found was that made the cells grow faster. It was only when the two of them were lost, and in a mouse model, we found that the tumors were much larger, and the animals survived at a less time. So, but I don't know why this is doing this. I hope this isn't the way my whole presentation is. So just to summarize, co-deletion of MAP3K7 and CHD1 is associated with relapse. Low protein expression predicts relapse. Co-loss drives lineage plasticity in the stem cell model. I didn't show you that, but by using specific markers, we showed that we had altered lineage differentiation of those cells in the tissue recombination. It also promotes EMT and increased invasion and motility, and again, I didn't show you those in the interest of time. And it also loss of MAP3K7 specifically is associated with increased brain metastases. And in a clinical model, I also didn't show you those data. And co-loss of those drives tumor pathology and robust in vivo growth. And I didn't show you those because the slide didn't work. So I want to switch gears a little bit and talk about prostate cancer and what drives prostate cancer that we know about. And one of the main things is androgen signaling. So the prostate is a hormone-sensitive organ, and it's driven by androgens, and androgens stimulate the differentiation and prostatic secretory functions of the normal prostate, and it also in prostate cancer, the androgens stimulate growth and tumor promotion. And the way this works is that in the cytoplasm there's the androgen receptor. It's a nuclear hormone receptor. It's a ligand-activated transcription factor. Androgens are as a steroid hormone. Testosterone or dihydrotestosterone binds to the androgen receptor. It's translocated into the nucleus and then stimulates gene expression. So this is one of our major driving pathways that we know about, and androgen signaling is targeted in prostate cancer as a therapeutic, either by blocking the productions of androgens, originally by Huggins and Huggins in the 1940s. They showed that castration would delay cancer progression, but now we have therapeutic methods of blocking androgen production in the testes and in the adrenal glands, and also specific anti-androgen receptor targeted therapeutics. So androgen receptor inhibitors. So what we found was that co-loss of CHD1 and MAP3K7 stimulated made the cells hypersensitive to androgen signaling. So we use R1881, which is a synthetic androgen. It's more stable than testosterone or dihydrotestosterone, so it works well in tissue culture. And so in the control cells, you can see that androgens stimulated the growth a little bit, which is consistent with what other people have shown. If we lose MAP3K7, androgens stimulate growth a little bit more. If you lose CHD1, there's really no difference between the control cells and the CHD1 knockdown cells. The double cells, however, even in the absence of androgens, grow much more robustly, and they are super stimulated by androgens to grow. And so that's just a single dose of 10 picomolar of R1881. If we look at a dose response curve, so I should tell you that in tissue culture, at least, we know that androgens have a biphasic response in this cell line, LINCAP cells, in the sense that at low doses it stimulates growth, and you get to a point where at a high dose it starts to inhibit the growth of the cells. And so that's seen here if you look at, for instance, the green line here, which is the knockdown of MAP3K7. You have a growth that's stimulated, and then you get to a point, and at this point it's 10 picomolar, that's the peak, and then you start to inhibit the growth of the cells. And the control cells are here in the blue. Here's the control lane. Here's the CHD1, nothing really. MAP3K7 loss shifts that dose response so that the peak is right at about the same spot, but it's a more maximal stimulation of growth. If you lose both, actually you shift that peak over, so now the inhibitory peak is much later, or tenfold later with this dose response curve than the control cells. So not only does it make them super stimulatory, but it shifts that point at which they start to be inhibitory. So what this data is telling us is that they're one of the potential mechanisms of how loss of MAP3K7 and CHD1 works is by altering the responsiveness to androgens. So one of the ways that we block androgen signaling is with this compound enzalutamide. It's an androgen receptor inhibitor. It binds to the androgen receptor, and it blocks androgen receptor from dissociating from heat shock proteins, and so it can't translocate into the nucleus and bind to the androgen receptor, and so it can't translocate into the nucleus, and so you block the signaling pathway. And this is used commonly in the clinic. And so what we found is that when these cells are treated with enzalutamide, the double knockdown cells now have a shift so that they're not as inhibited. So here's the inhibition curve for the control cells, and at every dose you can see that the enzalutamide has less of an effect on the knockdown cells. So it promotes androgen resistance, or what we say in prostate cancer, castrate resistance. So we wanted to know what happens to gene expression. So we conducted a study looking at how these cells respond to androgens. So basically we treated cells for 4 hours with 1 nanomolar of R1881 or ethanol, which is the vehicle, and we treated control cells, MAP3K7, CHD1, or double knockdown cells in triplicate, and we did RNA-seq, and we did AR ChIP-seq and a number of other ChIP-seq experiments. I'm not going to have time to talk about the ChIP-seq experiments today, but I'll allude to them a little bit, and it's published. We treated both plus and minus R1881, so it was a complex experiment with a lot of data. And there we go. What we found in LINCAP cells is that, as we expected, and so this is a curated set of androgen receptor target genes. And if you look at the RNA-seq data, you can see that in the control cells, in the absence of androgens, there's very little expression of those genes. And when you stimulate with androgens, you get expression. That's what you would expect. And in the MAP3K7 and CHD1 cells for this gene set, there's really not much difference, a little bit of difference between those. But when you treat the double cells, you get superstimulation of those genes. So this is in one cell line, so we wanted to repeat that. So we use 2 other cell lines, and we get essentially the same effect. So here's 22RV1 cells, which is a castrate-resistant line, and you can see that in the absence of androgens, there's not very much expression in the control cells. Endogenously, without androgens, you get more expression in the double knockdown cells, and then when you add androgens, you get superstimulation, and it also, the same kind of effect occurred in LAPC4 cells as well, another androgen-responsive human prostate cancer cell line. So our AR signaling study suggests that co-loss of MAP3K7 and CHD1 leads to enhanced growth in castrate and androgen replete conditions. Loss of MAP3K7 alone stimulates growth, while loss of CHD1 is neutral or slightly inhibitory, actually. Co-loss leads to resistance to anti-androgens, enzalutamide in the case that I showed you, and co-loss leads to increased AR transcriptional activity, both in the presence and absence of androgens. And I didn't show you, but co-loss leads to redistribution of the androgen receptor across the genome and leads to new AR binding sites in the genome. And it also leads to enhanced AR binding at existing AR target genes. And CHD1 loss promotes, MAP3K7 loss promotes increased transcriptional activity. So MAP3K7 loss doesn't affect where the androgen receptor binds, but it enhances the signaling when the androgen receptor is bound there. So we think that all of these data have pretty significant impact on a man who potentially might be treated with anti-androgen therapy, and that they may not respond well if they have loss of these 2 genes. And again, remember that there's a significant proportion of men that have loss of these 2 genes, so this has significant clinical implications. So men who are treated with prostate cancer, if they're treated with anti-androgen therapy, the typical progression is that they'll respond to that anti-androgen therapy by a suppression of the growth of the tumor, and the tumor will shrink. But eventually, they bypass that anti-androgen therapy and become castrate resistant and then start to grow again. And usually that is anywhere from 2 to 4 years, typically. And once they relapse, then we have very little therapeutic targets. And some of the mechanisms that people have identified how the cells bypass the anti-androgen therapy is by increasing the expression of the androgen receptor. Or they start making their own androgens within the cell, so they upregulate the enzymes that convert the precursors to androgens. Or they get mutations in the androgen receptor that allow promiscuous ligand activity. So now the androgen receptor binds to glucocorticoids, for instance. And so now, or actually the anti-androgen itself can now be stimulatory rather than inhibitory. And the other way is by making a new kind of androgen receptor, a truncated androgen receptor that no longer has a ligand binding domain and is constitutively active. And that's what I'm going to talk to you about, because that's what we found. We've looked at all of these other mechanisms and haven't seen any effect on how MAP3K7 and CHD1 affect, but we do find alterations in splice variants. So I'll walk you through that a little bit. So how does this work? So here's a schematic of the androgen receptor. You have N-terminal domain. You have the DNA binding domain is encoded in this region right here. And then there's a hinge region. And then this C-terminal portion is where the androgen actually binds to the receptor. And normally in 3 dimensions, that hinge region allows that ligand binding domain to fold over and block the DNA binding domain, so the DNA binding domain cannot bind to the androgen receptor. And when the ligand binds to it, it opens up and allows that DNA binding domain to bind to DNA, and then the N-terminal domain recruits transcription activators or repressors depending on the target gene and its surrounding elements. One of the most common androgen receptor variants is ARV7, and ARV7 has a cryptic, is the result of expression of a cryptic exon 3. And so instead of exon 4 being spliced in, this cryptic exon, which is in the intron between 3 and 4, is spliced in, and now you get a premature stop codon, and you get a truncated receptor that no longer has a ligand binding domain. So now this DNA binding domain is always there, and it's transcriptionally active. And the way, and I'm going to talk to you a little bit about the splicing factors, and the one that I'm going to talk to you about is the U2AF2. So in splicing, this is just a generic schematic of splicing. You have a 5' splice acceptor and a 3' splice acceptor. And U2AF2 has been shown to be specifically driving this cryptic exon splicing. So it's been shown to be very important in making these AR variants. So what we found was that if you look using PCR that's specific for the different variants, so for full-length expression, if you have, so this is, the blue is the control, and so the solid bars are in ethanol, and the hatched bars are plus R1881, and you can see that the full-length androgen receptor in the control cells, you don't really change much of anything at all. You have more AR full-length in the double knockdown cells, but it doesn't change in response to androgens. And for ARV7, in the control cells, there's expression, but it's not very much, and it's not changed. But there's much greater expression when you have the double cells. And so this is suggesting that when you lose MAP3K7 and CHD1, you're getting increased splicing of the variant, and this could be driving this castrate resistance and enzalutamide resistance that I showed you. And I'll talk about that in a minute. So what we did was I don't know what's going on with the slides, but we knocked down either full length or V7. Some of my things are missing here, and with specific siRNAs that target either the full length at the C terminus or the V7, or we knock down total targeting the N terminus. And what you can see is that when you have control cells, you get a specific amount of growth. If you knock down full length, it doesn't really affect the growth of the cells at all. If you knock down ARV7, however, that promotes less growth. And if you knock down the total, those cells are dependent on androgen receptors, so that also knocks down growth. Although in this experiment it wasn't statistically significant. And that's associated with increased apoptosis. And here I'm showing you cleaved PARP, but we've also used cleaved caspase 3. You can see that if you knock down full length, you get some cleaved PARP. V7 alone, you get some cleaved PARP, and in total, you get a lot of cleaved PARP in the control cells. Sorry? I don't know what that was. And then, but in the double cells, it's quite a different story. So if you knock down full length, there's no effect on cleaved PARP. But if you knock down ARV7 specifically, you get a lot of cleaved PARP, so you get a lot of apoptosis, and you also get a lot in the total as well, as would be expected. So I was talking to you about, whoops, sorry, go back here. So again, I just want to remind you that U2AF2 has been shown to be involved in this splicing. And so we looked at the expression of U2AF2, and in a clinical cohort using GSCA, we found that if you have loss of MAP3K7 specifically, you have, or low expression of MAP3K7, you have an enrichment of changes in splice factors. And one of them specifically was U2AF2, so we pursued that a little bit more. And so if you look in clinical cohorts again and look at the expression of U2AF2 in either MAP3K7 and CHD1 high cells or MAP3K7 CHD1 low cells, you can see that there's an increase in that U2AF2 expression. And that's also recapitulated in tissue culture. So the double knockdown cells have higher expression of U2AF2. And then we developed SI against U2AF2, so we can specifically knock down U2AF2 either in the control cells or in the double knockdown cells, and that actually changes U2AF2 expression. So there's part of my slide that's missing, actually, unfortunately. ARV7 is if you knock down U2AF2 in the control cells, you get a loss of ARV7 expression, and in the double cells. And in contrast, if you knock down U2AF2, the full-length AR actually goes up. So there's a blockage of that splicing now, and so you get more expression of the full length. And what this bottom panel is supposed to show you is that when you specifically knock down U2AF2, it also induces apoptosis and changes in growth as well.
Excuse me, Professor Scott.
Yeah.
For the time consumption, can you try to complete within 5 minutes?
Yeah, no problem. I'm almost done, actually. We also looked at how this affects the growth of the cells. So in the control cells are the diamond-shaped ones, and the squares are the knockdown cells. And you can see that so just looking at the control, either the double knockdown versus in black versus the blue, which is the control cells, you can see that the double knockdown cells grow faster, as I showed you before, than the control cells. And then if you knock down U2F2, that blocks that growth almost to the level that it blocks growth in the control cells. So U2F2 is driving this increased growth, and our data would suggest that it's through blocking this AR splicing, but it also now affects how these cells respond to enzalutamide. So here's data on enzalutamide expression or responsiveness. So the control cells, if you stimulate with it, and if you treat with enzalutamide, you get increased apoptosis. And if you and so these are the control cells, and if you knock down the full length, then they don't respond to enzalutamide because there's no androgen receptor there. And if you knock down ARV7, now you get responsiveness to ARV7, because so if you knock down the full length, you no longer respond to the enzalutamide because there's no ARV there. If you knock down the V7, they still respond to enzalutamide because they have the full length with the ligand binding domain where enzalutamide binds to respond to. If you knock down total, they of course have a lot of apoptosis, but they don't respond to enzalutamide because there's no with additional apoptosis because there's no AR there. In the double cells, it's even more pronounced. So you have in the double cells, you're resistant to inherently to the enzalutamide inhibition, unlike here. And when you knock down full length, you no longer can. You also can't respond to the enzalutamide. If you knock down ARV7 now, you don't respond to the enzalutamide to the same extent as the control cells, but you still have a response to them. And if you knock down the total, you also have a response. And there was supposed to be another slide there that shows that is associated with apoptosis as well. But that's missing from these slides for some reason. So loss of MAP3K7 leads to increased AR splicing. ARV7 drives castrate growth. U2AF2 is increased by MAP3K7 loss and drives AR splicing. And U2AF2 and AR splicing infers resistance to enzalutamide. So overall, what I've shown you is that MAP3K7 and CHD1 co-loss drive aggressive prostate cancer. Co-loss leads to increased growth, invasion, motility, and in vitro, and co-loss leads to tumor pathology and growth in vivo. Co-loss leads to enhanced AR signaling, castrate growth, and resistance to anti-androgens, and AR splicing drives this aggressive disease. And so now what we're doing is looking at what are alternate signaling pathways that we can target rather than anti-androgen therapy because these cells are resistant. And we published some papers on that. I didn't have time to talk about that today, but you can look those up. So who did the work today? Anu Rao was actually my first graduate student in the lab, and her and another graduate student, Wendy Barkley, they both helped develop the stem cell model that I talked about today. Lindsay Olkus and Leah Ryder were the, Lindsay was a graduate student and Leah was a postdoc in my lab, and they two worked together to define the MAP3K7 CHD1 subtype. And Lauren Gilson most recently graduated as a PhD student in my lab, and she did the AR signaling work. Currently, Gabriel Yet and Claire Gillette are two postdocs in my lab now, and Gabriel's working on a project that I didn't talk about today, which is that deletion of chromosome 8 and 16 and trying to identify the genes in there. And Claire now is working on the splicing work that I talked about today. And then Lena Romero is my technician for 18 years now, and she holds the lab together. And with that, I'd be happy to take any questions if there's time.
Thank you, Professor Scott, for a wonderful talk. And the question, can we have discussion with the students during the lunch hours?
Sure, that's great.
Thank you very much for this.
I'm very happy to introduce the next speaker, Professor Ratnamudi, Ratna Vatlamudi, and he's Professor, Vice Chair, and Program Director of MD PhD from University of Texas, San Antonio. And he has a very long association with School of Biological Sciences, Madurai Kamaraj University, and indeed he has visited us 2-3 times. And he has extensively worked on estrogen receptors and breast cancer biology, and he has more than nearly 200 publications in estrogen receptor signaling alone in breast cancer. And he has taken the research from laboratory to the bedside, and he has a lot of molecules and drug molecules and for estrogen receptor, like the hormonal breast cancer. And today we are happy to have him here. And it's indeed he's in San Antonio right now, and the time is around 1 o'clock in the early morning. So sorry, it's really sorry to keep him waiting. So actually he's joining online. Yes, Dr. Ratna, sorry to keep you waiting. So we just gave the introduction about you. So please start your talk, please. Yeah, please unmute the mic. It's an our screen. Yeah, we are. Yeah, they are controlling from there actually.
Can you hear me?
Yes.
Dr. Well, can you hear me? Can you hear me? No.
Professor, we can hear you. Yeah, we can hear you. Please start the session.
Okay. Okay. Good afternoon.
Good afternoon.
Thanks
For inviting us to give this talk. Special thanks to iCURE committee and also Professor Kumar and the chair of this session.
The
The topic I'm going to present you is a novel targeted therapy for treating breast cancer. These studies are funded by NIH grants. Other than this, Evastra is the company which is helping us to design these drugs.
As you know,
breast cancer is one of the leading cause of mortality for women in the world.
In US, see about 200,000 cases, and of them 40,000 people.
Will die of this
In
cancer.
India, also it's a
In
major
India, also it's a major problem. It's 178,000 people diagnosed in 2022, and about 90,000 people
I
died.
was reading a
I was reading a statistics that saying in India it is a leading cause now. 14% of the cancers in the women represents the breast
And
cancer.
if you're a woman,
And if you're a woman, you have one in 8 women has a lifetime risk of developing a breast cancer.
and more
More notoriously, the stats shows every 4 minutes an Indian woman is diagnosed with breast cancer.
there are three
There are three different subtypes of breast cancer. we call luminal
estrogen receptor breast cancer, which is 70 to 80% of them belongs to this one. These cancers are driven by hormone estrogen, and we have very good therapies to treat this subtype of breast cancer. Then the second subtype is HER2, or human epidermal growth factor receptor breast cancer, which represents 15 to 20% of the cases. For this group also, we have good therapies for the HER2 positive breast cancer. The remaining subtype is triple negative breast cancer, which do not express estrogen, progesterone, HER2 receptors. The more mortality is associated with this subtype because of lack of targeted therapies. Some of the data I'm going to present mainly focused on this subtype, triple negative breast cancer.
The target we are looking
in the study is leukemia inhibitory. This is an IL-6 family of cytokine, which is initially described in 1987.
widely studied.
Initially, it was shown this cytokine is involved in inhibiting the growth, but now we know it has multiple functions. One of them is promoting the proliferation of the cancer cells. Its normal function initially in the embryonic stage, in the embryo implantation, we need to suppress
system. So the lymph is being secreted during implantation to protect the embryo and it grow in the womb. The cancer cells are abused in the same system using this hormone or cytokine to suppress our immune system so they can be protected. How does this cytokine signals in the cells? This cytokine LIF binds a receptor called leukemia inhibitory factor receptor, or LIFR, and once the ligand binds to the LIFR, it dimerizes with another receptor called GP130, which is a common receptor for many cytokines. And when they both form dimers, they start signaling through the cytosolic kinases such as JAK, STAT, AKT, and mTOR kinases. These kinases send signals to the nucleus, activate a number of genes,
which contribute to the proliferation, reduction of the apoptosis, increase of the stem mass, which all contribute to therapy resistance and proliferation.
The
LIFR also has other ligands. In addition to the LIF, there is OSM, CTF, CNF. They can also bind the LIFR, and various cancer cells and body cells secrete these 4 ligands in different ratios.
So as I alluded,
the normal function for the LIF is in the embryo implantation to suppress the mother immunity. So in this case, these cancer cells also secrete a lot of LIFs to suppress the immune system, telling the immune attack not needed in the place of where the tumor is growing.
So there's significant literature.
For the last 15 years on this pathway. There is so much indication showing this pathway is involved in promoting chemotherapy resistance. It also involved in promoting invasion of the cancer cells. And it serves as a growth factor in promoting the growth and migration of cells. And also through mTOR pathway, it can influence the DNA repair and also contribute to therapy resistance such as radio resistance. The tumor microenvironment, LIFR pathway contributes to the suppression of the immunity, immune tolerance. All these factors facilitate the tumor growth significantly.
My interest
came into this field when these papers came in Nature and Nature Communications 3 years ago.
The first paper shows
using a pancreatic cancer model. They showed pancreatic cancer express a lot of this LIF mRNA and protein. When LIF is high, the survival of these patients are very low. And also they took the tumors at different stages of the tumors, and then they show in a mouse model where the pancreatic tumor progresses, they showed the expression of the lift significantly increases. At the same time, another paper published in Nature Communications, this one also showing KRAS-driven pancreatic cancer. The key findings in this paper is the lift is an oncogenic driver in this. The lift is contributing to drug therapy resistance, and they used an antibody which binds to the lift and blocks lift signaling, and showed with a standard of care drug gemcitabine, and they show when you combine these two therapies, gemcitabine along with the lift antibody, the tumor was suppressed, suggesting the lift is a valid target for cancer, at least in the pancreatic cancer model. And it's not limited to pancreatic cancer. In many cancers, if you see the TCGA databases, higher expression of the lift correlates with poor survival in lung, head, and neck. And we also see this in the breast cancer. And they show in vitro it is very high affinity. It blocks the downstream signaling such as STAT3. And by analyzing the tumor microenvironment, they show it reduces blocks the M2 macrophages, which are bad, and Tregs, which are also bad for the tumor cells, and increases the recruitment of CD4, CD8 cytotoxic cells, which promotes the tumor suppression. So all is good. This drug went into phase 1 clinical trials and showed some efficacy, but it was stopped due to little bit toxicity at the end of the study. So the antibody therapy is good, it works, but it's very expensive to give to the patients. Cost $10,000 to $50,000 for treatment. It's a lot of money. So what we hypothesized was, if this pathway is significantly contributing to cancer progression, and it's a valid target for cancer therapy, can we develop a small molecule which can be taken as a drug, oral tablet, which will be much cheaper to give it to the patients. So our hypothesis is, even if this antibody is blocking the lift, if the cancer cells can secrete the other growth factors or cytokines such as OSM, CTF1, so they can bypass the therapy which we are doing with the lift antibody. So if we can model a molecule which can bind to the LIFR and blocks all 4 molecules binding to the LIFR, that could be a valid therapeutic. This is the rationale we use, and the main purpose is to develop a orally available small molecule inhibitors. How do we achieve this feat of developing a small molecule? So we started using a homology modeling. We were lucky that these structures are already available in the literature. So we use the 3D structure which is already present for the human LIFR. As you can see in the LIFR protein, in this cartoon shows the structure of this protein. It has 2 cytokine binding modules, CBM1 and CBM2, and they're linked by the IgG-like domain. And if you see in the context of the interaction between the human LIF, which is this 4 helical bundle, interacts with the D4 and D3 region, and the computer predicted there is a protein-protein interaction, PPI, which is shown in the red circle. So we reason that if we can make a small molecule that can bind to this region, which may interfere binding of the LIF to the LIFR, and that could be first as an inhibitor of this pathway. Initial screen identified a number of molecules. The 4 molecules are shown here. These are our lead molecules. The computer predicted these molecules can bind to this interface, and further modeling and docking studies showed the 11B substitution is the key, and if we have the bulkier substitution, it is interfering better. Accordingly, when we see the binding energy calculations, this high affinity is shown for 359 and low affinity was 330. In the initial cancer cell assays also, 359 showed a bigger activity. So when we do the further modeling, this one shows when the EC359 binding to the LIFR interferes LIF binding to the D4 domain of the LIFR. And several binding forces were also shown. Many of these forces interferes the LIF binding to the LIF receptor. So to gain further evidence, we biotinylated this compound,
And we use Erudition to pull down the biotin drug and see if it binds to the LIFR. So using the purified protein as well as the cell lysates, we were able to pull down LIFR using the biotin-related compound. The another assay we use is microscale thermophoresis assay. In this assay, you take a pure LIFR protein and add the drug. When the drug binds, it's going to change the shape of this protein, which can be measured. These studies shows the drug binds to the compound binds to the LIFR in a dose-dependent manner with a KD of 10 nanomolar. Very high affinity compound binding to the LIFR receptor. So the next assay is to see does this compound has activity on breast cancer cells. We use cell viability assay. So you took a 94-well plate and you plate the cells and throw the drug in different concentrations and measure them after 6 or 7 days. If the cell viability is reduced, you will have a less number of cells. To our surprise, the ER positive cells showed no activity. And when we went and do the Western blot, we saw they express very little of this LIFR and LIF proteins. On the contrary, triple negative cells have high levels of the LIFR, and they're very susceptible for this drug, suggesting you need to have a LIFR target for this drug to work. We also validated this concept using a CRISPR-mediated knockdown of the LIFR. And when we knock down this gene, this is inducible knockdown, significantly knock down the protein to 80 to 90% of the protein. And when we knock down the protein, the drug efficacy is significantly reduced. Control cells, the drug in a dose-dependent manner reduce the cell viability, but the cells which are knocked down showed resistance for this, the drug activity. Then we went into tumor tissue arrays. So these arrays contain different subtypes of patient tumors plated on the slide. So this has 89 ER positive slides and HER2 slides and triple negative breast cancer 61 slides. IHC analysis shows of all these subtypes of this cancer and normal cells, triple negative has higher expression of the LIFR. We then tested the ability of the EC359 to block downstream signal by the LIFR. When the ligands such as LIF, OSM, CTNF, or CTF, when they bind to the receptor, the first downstream is an activation of this JAK-STAT3 pathway. So we use the triple negative breast cancer cells which stably express STAT3 reporter. And in those cells, when we treat with EC359, the reporter activity is significantly decreased. When you stimulate these cells with hormones such as LIF, the activity is significantly increased, which can be further blocked by addition of the drug EC359. We validated the same assay using 3 other ligands. In all the other ligands activated the STAT3, but which efficiently attenuated by the addition of EC359. We then did some Western analysis showing the hormone such as cytokine LIF stimulates the phosphorylation of STAT3, but which can be blocked by the addition of EC359. Using 2 different models, we showed this one. And also we validated the downstream signals such as S6, MAP kinase pathway, and AKT pathway are also decreased when you incubated the cells with the drug, suggesting the drug is blocking LIFR-mediated activation in these cells. We further validated using the functionality of the drug using a qRT-PCR of several known LIFR target genes. As expected, addition of the EC359 into this medium, the gene expression of the LIFR target significantly decreased. We also did some long-term colony formation assay. In these assays, we treat the cells 5 days, then you remove the drug and culture them for 2 more weeks. In these assays, control cells formed a nice colonies which are visible. And EC359 treated completely eliminated the colony formation. At least 90% of the colony formation is decreased. The quantitation is shown in the bottom panel. We also examined whether it promotes the apoptosis because STAT pathway is known for the survival. Blocking of the STAT pathway contributes to apoptosis. In both models of the triple negative breast cancer cells, addition of the EC359 significantly increased the apoptosis of these cells. The lift pathway is known to play a key role in stem cell proliferation and differentiation. So, and first we initially test whether the lift correlates with the stem cell gene set in the TCGA databases. High lift expression correlated with the memory stem cell genes and also similarly lift also highly correlated with the stem cell genes, which is expected considering these two proteins play a significant role in the stem cells. And then we purified the stem cells using ALD5 FACS sorting, and we incubated them without the drug. This significantly reduced the stem cell proliferation in these assays. We also showed by Western blot, you can see the significant decrease in the lift downstream signals in this pathway. The next step is to see, can we translate this drug into the clinic? For that one, you need to have a good pharmacodynamic properties and pharmacokinetic properties. The PK analysis shows this drug is available orally and with good systemic exposure. And kinetics showed it has a half-life of 4.5 hours. We also did some toxicity assays for a 1 week feeding of the mice. With from 0 to 100 milligrams per kg, we didn't see any notable toxicity during this period. So we started using a dose of 5 milligram per kg in our assays. So this first one is a xenograft study using 231 cells. Control cells, untreated, vehicle-treated ones grow significantly over a period of 25 days. But the mice treated with EC359, the growth is substantially reduced. The body weight of this mice is not affected, showing this drug is not toxic. And once the treatment is done, we took the tumors out, and the tumors were stained with the proliferation marker Ki67. As you can see, the proliferation marker is significantly reduced, suggesting it is slowing the tumors down. We also did, LIPFIS plays an important role in invasion and metastasis. Some of the deaths in triple-negative breast cancer is due to metastasis from the original tumor formation to spreading all over the body. So in vitro assays, EC59 able to reduce the migration of these cells. This is a Biden chamber assay. And then we also did in the mice where we injected a lung-tropic metastatic breast cancer cells through tail vein injection. The control cells have a significant tumor in the lungs, but TC59 significantly reduced the tumor formation in the lungs. The last assay we did is, does it applicable to the patient? So in this assay, this is called a patient-derived explant assay, where we take the fresh tumor tissue from the surgery of the breast cancer patients and incubate them without drug in the medium. So when the drug is not given, these tumors grow very nicely and proliferation marker. When the drug treated ones, they have very little proliferation. We also put these pieces into mice. We culture them as a patient-derived xenografts. Here also, control tumor grows significantly, and when the drug-treated mice showed the tumor growth is decreased, no changes in the body weight. To summarize the study, we identified LIF as a potential target for treating triple-negative breast cancer. We developed a first-in-class inhibitor EC359, which binds to the LIFR and reduces the survival and induces the apoptosis of triple-negative breast cancer cells. EC359 can be used to reduce the progression in mice models. We believe EC359 represents a novel mechanism to target LIFR oncogenic functions. We saw also similar effects in endometrial cancer and ovarian cancer, which we are writing the manuscript on those findings. I will stop here. These are my collaborators who helped me with the generation of this data. I'll stop here and take questions. Thank you.
Thank you so much, Professor, for your wonderful presentation. So now the session is open for questions.
Yes, yes, yes. Yeah.
Thank you for that nice talk. I have a question. You say there are no toxic effects, but can you tell us where normally LIF receptor is expressed and whether whether you have monitored any, even if non-toxic effect on other tissues or normal tissues that express the receptor?
So the LIF is expressed in many tissues, but predominantly the LIF is expressed widely during implantation and for the stem cells, and also in the brain. So our drug does not go to the brain, so we can't comment on brain toxicity. But other organs, the dose which we are using is well tolerated because it's a nanomolar inhibitor, 10 nanomolar to 20 nanomolars. So we always use the low dose. In the clinical trials also, when they do the LIF antibody, they didn't see big toxic effects. But the effects they see are different when they go really high dose. So we expect to see a low toxicity when you use the low concentration of these things. But as you can see, like chemotherapy, anything when you go with higher dose, there will be side effects for any of these pathway drugs because they're naturally present in our body.
And you said at the beginning that LIF belongs to the class of interleukin-6 molecules. The drug, does it interfere with the interleukin-6 signaling?
You can repeat the question. Yep. Please repeat. Okay.
I am asking for, you said LIF belongs to the interleukin-6 family.
Yes.
Does
different than LIFR receptor. They share one subunit common, GP130, but usually it should not interfere with IL-6 signaling.
Okay. Which eventually would not be bad.
Much for your talk. Really sorry for keeping you waiting. Thank you very much.
No worries, no worries. It's a pleasure talking to you.
Thank you.
Heartily thanks to all the speakers of the session for the valuable lecture, and I would extend our gratitude to the chair and co-chair of the session. Thank you, sir. So we are about to start our next session. It's about ocular diseases. Vision impairment and eye diseases are the major health problem, wherein 3/4 of the world population is suffering from one of such ocular diseases. We all know that this is the major talk of this town in the current scenario. To dive in further, I would like to welcome the chair, Professor Dr. K. Balakrishnan, Secretary in Indian Immunology Society, along with the co-chair, Professor Dr. P. Varalakshmi, School of Biotechnology, Madurai Kamaraj University, to lead this session. We welcome you, ma'am, and sir.
Thank you for the nice introduction. I'm highly privileged and feel proud to introduce the first speaker of this session, Professor K. Dharmalingam, Director, Aravind Medical Research Foundation. Professor Dharmalingam was the eminent professor and scientist of Madurai Kamaraj University. Started his career, both UG and PG he completed from Tyagarajar College, Madurai. Did PhD subsequently in Madurai, School of Biological Sciences, and had his PDF from Tufts University School of Medicine. Professor Dharmalingam has a lot of credentials to receive close to 10 unique awards, prestigious awards, including the Bhatnagar Award, the topmost scientist award in India in the year 1992. And other important awards includes 1985 he got elected as member of Guha Research Conference, and even to the recent times like 2010 to 15 he got awarded or received DBT Distinguished Research Fellowship by Government of India, and in the year 2016 awarded with the Baireswar Chakrabarti Oration Award. So Professor Dharmalingam has established the Genetic Engineering Research Unit and Biotechnology in Madurai Kamaraj University, and he was a founder professor of School of Biotechnology. He has published more than 200 research papers in highly reputed peer-reviewed research journals in the area of molecular biology and biotechnology. So, and also we are all privileged that we are all his student last 30-35 years. So with this great honor and small introduction, I invite Professor for his speech on ocular diseases. Sir, please.
Hello? Yeah, thanks. Thanks for the nice introduction, Dr. Balakrishnan. And also I thank the chairs for this session. So if you give me the first slide, I'm very happy to be here as usual to participate in this excellent meeting where the Vice Chancellor and others are trying to do their best to bring MKU to real frontline research and developmental activities. I will be talking to you today about a problem which we are working for quite some time. In this, if you look at this, diabetes is perhaps one of the worst things to happen to a person. And if you look here, it is among the 10 leading causes of death. And if you look at the number of people who die of diabetes, is phenomenal. And the most important thing, particularly for India, is the so-called undiagnosed diabetes. That is almost 1 in 3 or 1 in 2 in many cases. If you look at here, there are more pre-DM, that is impaired G3, than DM in India. Because if you go and look for them in the population, you will see a large number of people with impaired glucose tolerance. And what is surprising is the conversion rate to DM is also very high. So these people are actually not aware of a time bomb which is actually going to explode sooner or later. So with this background, if you look at the actual number of people suffering. 2021 it is 537 million, and it is going to be 643 million in 2030 and 783 million in. So there is quite a lot of important things which we should remember, particularly in India. So if you look at the disease, what it does is the complications of diabetes are this important factors. And if you look, there are several things. It could be cardiovascular, it could be either macrovascular, which lead to cardiac problems and so on and so forth, or it could be microvascular. That's what we are working on in diabetic retinopathy. There are other microvascular complications like nephropathy and neuropathy, etc. But we are focusing on eye diseases because I work in an eye institution. Not only that, if you look at the prevalence of diabetic retinopathy, then you can see among the known diabetic patients, these are actually door-to-door screening of diabetic patients done in India. It is 15 to 18%. And if you look at undiagnosed diabetes, among them there are 1,783 people screened, you can see 8%. It may be higher as well. So roughly half of the undiagnosed compared to diagnosed also develop diabetic retinopathy. And if you look at vision threatening among them, that's what the critical factor. They lose eyesight. No treatment is going to help them at the later stages of the so-called proliferative diabetic retinopathy. That's about 4%. So 3 million people are at risk in what is called vision threatening diabetic retinopathy. It could be diabetic PDR or diabetic macular edema. So with this background, what are the critical factors which we should look at? All we can look at is whether it is possible to delay the onset. If you know the diabetes well in advance, because diabetic complications develop maybe after 5 years or 9 years, so on and so forth, not immediately. So if you know ahead of time or early on a diabetic condition, then you have better interventional strategies by controlling diabetes and also initiating early treatment when once you have, particularly in eye diseases, what is called non-proliferative diabetic retinopathy. I will come to it in a minute. So that means not only that, if you look at this, even if you have better glycemic control after a period of diabetes, it's not going to help at all in any way the progression to microvascular complications. It could be nephropathy or retinopathy. And also these changes are very difficult to reverse. So that's why early catching people early on in diabetes, number one, it helps. But what is more significant is this, that is, if you can predict the disease before the clinical symptom, that is diabetic retinopathy, then you have a better option to have better life. Whereas if we wait until the development of the symptoms of diabetic retinopathy, then it is very difficult to do anything about it, because it will progress from mild, moderate, severe to proliferative diabetic retinopathy. So that's what the whole objective is. Whether you can predict who will progress from diabetics to diabetic retinopathy and who will progress from mild NPDR to PDR. These are two simple questions which we asked, and how will you do this? So in the general way, biochemical markers appear way ahead of time compared to clinical disease manifestation. So that's what the whole concept. So you go through the regular phases. What is the difference in terms of biochemical markers? We are talking about two different things. One are proteins, one is microRNAs. There are two different things. And one is a discovery phase. In the case of proteins, it is purely depending on mass spectrometry and look at a limited number, maybe 10, 15 samples of various stages of diabetic retinopathy starting from DM. And then validation phase, and then comes the, I will be talking about these two which we have done already. So here briefly, diabetic retinopathy, as I said, is a progressive disorder. So from normal to PDR, where you have vascularization and it is almost become irreversible, it goes through a mild NPDR which actually shows swelling of vessels, and moderate NPDR where you see a closure of small blood vessels, and finally severe, there is a damage to blood vessels, and finally a neovascularization. So what I'm talking about is whether you can find from here to here what is the signal or what is the marker which will tell, and how is that, whether we can say anything about markers which are specific for the progression of the disease. And what we have done is in the discovery phase, looking at 15 samples of the various stages serum in mass spectrometry, we have identified a large number of proteins which are either upregulated or downregulated. So that is the first stage. Now they have significantly up or downregulated in this small sample size. Okay. Now then adding whatever is reported by other groups in other populations. I will come back to you this population specific issue at a later stage, because what is true of one population need not necessarily be true. I will tell you how we found in our own studies. And there are specific which are actually for Indian population. And totally, whatever I identified is 30 of them are found only in the PDR in our studies, 13 specific to our population, and there are 14 from other published literature. Totally, there are about 57 proteins are in the pot. So amongst the 57, we are going to be looking at a selected group to see whether they will give a clear indication of a panel which will give, okay, what is progression from DM to DR and what is from progression from NPDR to PDR. So what we have done is, when once we have in the discovery phase these 12 markers we took, and this is simply a multiplex ELISA. So we have 12 different proteins and 12 different antibodies. We looked at these in a multicentric study where what we are looking at is a huge number of samples. For example, here totally 538 samples. Actually, this 538 is after taking the ELISA data of 1,500 samples totally. That is roughly here 1,000 from India and 500 from UK. So what we got was here, we are looking at adults 40 years and older, and they are grouped into 4 groups. There is no history of diabetes. And then type 2 diabetes, at least 5-year duration and no evidence of DR. And then non-proliferative DR with diabetic macular edema, and then proliferative DR with macular edema. So these are the grouping, 6 groups. And what we have done is looked at the multiplex ELISA for all these markers. And I will tell you a couple of markers which have already been published in JAMA Ophthalmology, so you can look at it for the details. Majority of the work was done by Jaya in my lab in Arvind Medical Foundation. If you look at this marker, cystatin C, you can see from PDR, NPDR and no DR and this no diabetes. First point is this, the level of cystatin C in the serum of Indian and UK populations are very similar. I want you to note this one because there is going to be a next one coming up which will show you a different story, and this one gets upregulated. And also in the moderate to severe NPDR to PDR. Now if you look at this whole sample, this one is the next one, leucine-rich alpha-2 glycoprotein. There are 2 points to note here. Look at this is one population, that is a UK population, this is the Indian population. This is no diabetes, even in the control. The level of this marker is much higher. If you are going to compare this level from one population to another population, you are going to be in trouble. So you got to use the same population data. That's the most important point. And then it actually also follows, and we can use definitely as a marker for the progression of DM to DR. So that is what actually we have done. If you take both these markers and build an ROC car, which is important to see because you got to look at 2 different aspects in the biomarker, selectivity and sensitivity, right? So, and both are actually working out nicely here. Whether you are using, in this case, baseline is this, and if you have cystatin and cystatin plus LRG, you can have a better predictive value. Sensitivity is very high and specificity also very high. Now, if you look at the United UK population, they also follow similar pattern. So, as far as the 2 markers are concerned, cystatin C and LRG, we can use as predictors of progression from no DM to DR. Now, if you look at this, this is again very interesting. Complement factor B, this is something which we found only in Indian population to start with in our discovery phase. And if you look at here, the level of this complement factor B in no diabetes in an Indian population, this is the compared to the Indian population versus UK population, they are completely different. Just like the previous slide, you had to be careful which population you are actually comparing. The whole study is actually designed to find out whether we will get a universal setup panel to predict the 2 aspects which I was talking to you before. But then what we found is, yes, we can find, provided there are some markers which are specific only for UK populations and some for Indian populations. And next part I want to talk about is something which we have started very recently, that is looking at extracellular vesicles. Extracellular vesicles are important in the sense they are actually produced by all the cells and they represent the cell type produced or made from producing them. So in the sense, if you look, there are 2 different types of particles. There are one is large EVs and small EVs, and totally we call them as extracellular vesicles because the small, most previously they are called as exosomes because they are coming from a different pathway, or they are called as microvesicles because they are produced by another pathway. So what we are trying to look at is not exosomes and microparticles, we are looking at all of them. Everything produced by a cell and secreted out or actually sent out. And if you look at the literature, see in eye disease there are very few papers are published on the extracellular vesicles and their role. So what we decided to do is whether we can look at extracellular vesicles which are actually circulating in the blood as markers for the two different conditions, DM to DR and PDR, NPDR to PDR. So what we have done here is we took another set of markers, because 12 markers we have already checked in the population-based study, which is a cross-sectional study. And we have taken another 12 markers, because there are totally 57 we have. Another 12 markers, and then you look at the numbers. These are the numbers which are examined. To quantify, first isolate the exosomes, which is an involved step. It's all done by ultracentrifugation. And then look at those exosomes or the EVs for the various proteins here. Where is A2M or vitronectin, peroxyredoxin or CD5L or fibrinogen and TSP1. This is a thrombospondin 1. And if you look at this, for example, only in microparticles we can find these 2 plus 3, 5 proteins. We missed it completely in our mass spectrometry as well as in ELISA. We couldn't find them in the serum. See, obviously they are in the serum. Microparticles are actually isolated from the serum. But the only point here is, since they are actually sequestered in the microparticles, we were unable to detect it in the bulk serum. So when once you isolate them, you can find them, number one. Similarly here, if you look at these two proteins, WF and thrombospondin, you see them only in the serum, but for example, thrombospondin you don't see in the microparticle at all. So there is very clear separation here with respect to these 6 or 7 markers. Some are in the bulk serum, some are in the microparticles. Now the question is whether we can use them as the biomarkers for our purpose. This is just a couple of slides to show indeed what you're talking about is microparticles. These are the electron microscope images of serum microparticles. That actually you can see a heterogeneity of this. These are smaller ones and so much larger microparticles you're talking about. And also the size, etc. are given. So you're more or less in the normal size range. If you look at them, the DMK is about 124, 78 to 124. And this is normal, which is actually NPDR, this PDR you can see. There are not great difference in terms of the size of these particles. And also we have shown that indeed these are microparticles because the characteristic proteins which are present on the microparticles is CD9, and we do find them in the Western blots when you look at all these microparticles. Now, if you look at this, this is the total number of proteins identified using mass spectrometry within the microparticles. So that's very interesting. If you look at them between PDR and DM, there are only 10 which is unique to DM and 24 which are unique to PDR, and there are many which are common. But what we are looking at is not only the proteins which are found exclusively in one case or other, but also the proteins which are differentially regulated in terms of the quantity of the protein which is we're talking about. So indeed, we do have microparticles from these two conditions which are different. Actually, it is predicted it should be different. Now, if you look at the other one, there are several pathways which we can figure out from these large number of proteins which we found. On one pathway I will show you. There are other pathways I am not going to show you. This is actually a complement pathway. If you look at it, there are several proteins in the complement pathway which we measured here. C3, CFB, all those things, CFH and all those. But what is interesting is for the no DM to DM, you see their complement pathway is coming down. Whereas if you go towards this is NPDR, it goes up, and slowly as the NPDR progresses, it comes down. So this is another indication indeed between DM to NPDR and NPDR to PDR, you do have distinct metabolic profile change. So that's what actually we are looking at it. But interestingly in this case, not only the activators of complement is increasing in the NPDR, but also regulator here. CFH is a negative regulator. I'm not going to talk about this pathways and their role in the disease, but I want to tell you that you can get huge amount of information by looking at the microparticles and the actual proteins which are present, because this one, as I said, we completely missed. If you look at it in the serum, you won't be getting all the proteins of the complement pathway. So this box plot actually shows with the number of patient samples we have used. I already said it's more than 250 or 230 samples we use. But whenever I say the numbers, you have to be very careful, because if you collect about 500 samples of DM, you will know only about 250 or so will be usable, because other clinical parameters are important, other compounding factors have to be considered. So this is a major work because we have to get a huge number of samples. There are two different, I don't think we'll be able to read it, CD4161, and this is peroxyredoxin. If you look, this is a no DR to early DR and late DR, actually they're coming down. Levels are going up. This is statistically significant because we have used huge number of samples here. So that is one indication that these two proteins, CD4161 and peroxyredoxin, could possibly be one set of markers we can use to detect or actually at least predict the progression of NPDR to PDR. And there are two other markers, fibrinogen and thrombospondin. These also show very similar pattern. For example, starting from no DR, if you early DR to PDR, you can see there is a progressive decrease. Similarly here, no DR to DR, there is a considerable decrease in thrombospondin. So this is again indicates that we definitely can come up with a panel of markers for these two diseases. This is what actually is done. So far I will summarize. So we are having actually no DM, T2DM, NPDR, NPDR moderate, severe and PDR, and these are the markers which we actually looked at. And now we are going to be looking at whether we'll come up with a biomarker panel for diabetic retinopathy. Now in this slide actually shows that there are some markers which are altered significantly, but not all of them are with discriminative power. That is the problem with all these studies. You may see in your studies there are variations between one condition to other condition, but when I extrapolate to the population, they should be statistically meaningful and should have a predictive power. When you put an ROC curve, then you will know that whether you have wasted your time or actually you're lucky. So what we look at here is, in this case, for example, these are the two markers, that is CD41 and vitronectin. They have the predictive power for the DR onset. And 4161 vitronectin, fibrinogen as well as thrombospondin. All these four are capable of actually predicting the progression of the disease. So now we have definitely the leads, and what we are going to be looking at, this is the actual curve which is using the EV markers. You can show also this with this ROC curve. Definitely they have a predictive potential. Okay. Now what we are next trying to do is whether you can understand the mechanism of diabetic retinopathy, because as somebody said in the beginning, you see, you can't do any translational science if you don't do basic science. See, translation science comes after basic science, and unfortunately that is missing along the line, and people always say you can do translation, right? So what we have done is this. So this, just to summarize here, we have large EV proteins which allowed identification of 4 additional proteins which we could not find in the serum. That is 4161, peroxyredoxin, thrombospondin and fibronectin. And these two are capable of predicting DR among DM, and these are actually capable of telling you the progression of the disease. And in the meantime, we got this, which is may not be related to translation, but definitely it will give you somebody to really enjoy doing science. See, there are two things. One is enjoy doing science, and another is enjoy earning money. And if you do both, that will be nice. So now I will just switch and then go on and tell something which we found very recently. This mechanism of DM switch to DR, how is it happening? If you look at that, this very early on, it's not 71, actually 71, Judah Folkman proposed that there is definitive endogenous inhibitors of angiogenesis, and the balance between these two, pro and anti-angiogenesis factors, is responsible for keeping this angiogenesis. Now, why is it important? Because what's happening is, in diabetes, you don't have any angiogenesis of the vessels. And the complication when it starts, one way it can go to macrovascular complication, another way it goes to microvascular complications. And you look at them, it is very intriguing to look at the arterial macrovascular endothelium and the microvascular endothelium are completely different in terms of their tissue type expression of thrombospondin. That is somebody proposed very early on. So there is a tissue or a cell type specific expression pattern of thrombospondin, and thrombospondin is one of the earliest inhibitors of angiogenesis. So as an endogenous inhibitor of angiogenesis. Now thrombospondin is produced by almost all the cells, and they are produced by hypoxia as well as glucose. You just simply take the cells and add glucose, you can see the expression of it. And so what we are looking at is when you look at the previous, if you remember the previous results, when no DM you have a huge amount of thrombospondin, and DM also there is a huge amount of thrombospondin, whereas when you go to DR there is very low level of thrombospondin. Thrombospondin almost is gone. So we are looking at what's happening here. So this is what the data here I'm putting back again. This is one of the data which is generated by one of my colleagues, Dr. Ram Prasad, in his study. This is a qPCR analysis of 2 microRNAs. Now, what we thought about was there must be a regulator of these proteins, and what are they? And he did a panel of all the microRNAs actually taken from NPDR serum as well as PDR and DM serum, and he found out these two, MER27B and 320A, are actually upregulated. If you look at this, you can see DM versus PDR 8.3-fold increase, and this one DM versus NPDR is 2.3-fold increase. There is an increased amount of these 2 microRNAs. So what is the significance? What is the significance is this: if you look at the serum level, there is a opposite trend in the sense you have higher levels of DM, the thrombospondin 1 in TM, and there is a much lower level. And this has been shown subsequently by Jaya by using a large number of samples, there are about 200. So you see in no DR you have a higher level, as come to DR it is going to be in a lower level. So what is that meaning? That is thrombospondin, which is very high in diabetes, coming to DR it is going to be very low in DR. So what it means is this. So when you have thrombospondin, it is interacting with CD36, which has been shown by others, and also it interacts with VEGFR. This is a receptor for the VEGF, and when this interaction occurs, couple of things are happening. Now the TSP1 prevents angiogenesis because this particular phosphorylation actually blocking the microvascular endothelial cell mediated migration and tube formation. There is also another pathway I'm not going to be talking to you. So if you have thrombospondin in excess, that's what happened in DR, there is no blood vessel formation, there is no angiogenesis. But if you go to DR condition, what happens is there is no thrombospondin because the MIR27B is inhibiting the thrombospondin expression. But here is the contradiction. There is a report, if you take the microvascular endothelial cells, if you add glucose to them, you do form excess amount of thrombospondin messenger RNA. But subsequently when they looked at the protein, it is not forming the protein. See, that's the difference. In the macrovascular endothelial cell, there is no inhibition of the translation. Whereas in the microvascular endothelial cell, there is an inhibition of translation. So messenger RNA is there, but the protein is not there. And now it has been, I looked at it and found the messenger RNA of thrombospondin 1 has binding sites for MIR27B. So it looks like what's happening is there is an epigenetic change which has been shown by others, particularly methylation, which actually activate many genes in PDR, and also it increases level of MIR27B, which we have found in our samples as well as it has been proven in the endothelial cells, and which inhibits the thrombospondin 1 translation because transcription is not altered, and which in turn leads to the absence of thrombospondin 1. So consequently what happens if you don't have thrombospondin 1, that's what happened in PDR, so you're going to have microvascular endothelial cell migration as well as tube formation. So before concluding, am I okay with time? Okay. Now just to conclude, apart from using bulk, blood, serum or plasma, using microvesicles or extracellular vesicles is a useful thing to do, number one. Now we are in the process of taking this panel of totally now 8 markers, whether we can develop a clinical assay. As I said in the biomarker discovery process, clinical assay development is another important critical factor. It may work in the laboratory, but whether it has to be established in the clinic, because your sensitivity as well as selectivity, specificity, so on and so forth should be good, and the assay should be cheap enough to do it. That's the second part. And finally, we are going to be testing in the population to see whether this will be usable for the clinician, because who would be now predicting who will possibly be going towards this, so he will be monitoring it. Otherwise, we will be losing them completely, because people come to the clinic only when they reach the advanced stages of PDR, not early, because nothing happens during the early stages. And when once they come to the clinic early stages, laser treatments and other treatments are there to essentially cure this particular disease. And then finally, using all these things, we always look for what is the basic science which you could do, because interestingly now, thrombospondin, for example, when we realize there is a microRNA 27B is involved, then you will be able to whether target this particular interaction so that you can really find out a therapeutic intervention in a newer way, rather than looking at, one is looking at the markers to predict, another is to therapeutically intervene. That could be done. And these are the financial support, and large number of students were involved, mostly with Dr. Jaya and Ram Prasad. Thank you very much.
So thank you very much for your very interesting and innovative biomarker study, diabetic retinopathy, sir. I now invite the audience for a very quick question, couple of questions. If there is no question from audience, I just one question, sir. So you said between two populations, the complement factor is very high in UK population.
Indian population.
Indian population. Similar to the LRG. LRG is also high.
Yes.
Is there any specific reason attributed, or maybe the complement? One more point I want to ask is the complement is an indirect indication of circulation of more antibodies, so that in the complement will enhance the elimination of infection, whatever. Is there any reason or connection?
No, we really don't know, because the particular thing, the complement factors go up because of many reasons, particularly infections and inflammation. These are the two main causative factors. And as you know, we are actually in the inflammatory status of Indian population is much higher. And there is also yet another thing I said, the impaired GT, glucose tolerance of many of the people here will be a predisposing factor for increasing the complement activities.
Yes.
So what is important there is not only the complement which is going to be different, but the entire complement system is operational in a higher rate in the control populations in India. And the LRG doesn't have too much of a worrisome issue right now, where it is higher, lower in the control populations. But the critical factor is, when you look at biomarkers, whatever is done elsewhere need not necessarily reflecting the status in our populations.
Thank you, sir. Thank you. So now I invite our Honorable Vice Chancellor. So we'll be happy to hand over the memento to Professor.
I request we request everyone to go for the lunch arranged in the CIC space.
No, excuse me, wait. Yeah. There is one more lecture is an online.
Sir, no, I just want to know.
After this talk, sir. This is the last talk of this. 30 minutes. 20 minutes.
He's on
Online, sir, from Singapore.
I suggest is those who want to go for lunch, let them go. Those who want to remain, let them remain. Just don't worry about the bill.
Okay, sir.
Those who want to remain, let them remain. Don't bother for it.
Yes, sir. Okay, sir.
So it is our pleasure to invite Professor Sendil Velan, Associate Professor, Singapore Institute for Clinical Sciences. That is an A-star institute of Singapore, highly prestigious research initiative of Government of Singapore. And Professor Sendil Velan is working on metabolic imaging group with A-star. And currently he is doing a lot of imaging of lipid metabolism, MRSP preclinical imaging, clinical imaging, and tissue metabolic obesity and diabetes. So he is a member of International Society for Magnetic Resonance in Medicine, which is in Berkeley, California. And one of the very interesting fact about Sendil Velan is he is looking for collaboration and involvement of younger scientists from across the country and the continent, and very active in developing novel imaging techniques for investigating metabolic pathways and looking for industrial partners and research collaborators. So with this brief introduction, I invite Dr. Sendil Velan to deliver the lecture. Sendil Velan, sir.
Now, so is my slides are in the slideshow mode?
Yes, proceed.
Yeah, thank you very much. Yeah, good afternoon, and I would like to thank the organizers for giving this opportunity to give a talk at the symposium. And in particular, I would like to thank Nana Kumar and Dr. Ashok Kumar, who have been in communication
And for setting up this workshop. And yeah, with this, I would like to highlight some of our ongoing effort in the area of metabolic imaging in Singapore. We all know that metabolic diseases is on
the rise and is a global
issue,
resulting in huge
economic costs and also hospitalization
costs, mainly due to the rise in the pre-diabetic and diabetic cases, and in particular Asians, who are most of the are about 60% of the global population, and they have also represent about 60% of the diabetic population.
And
that's where I think it is very relevant
to address this issue of obesity and diabetes, and even in childhood obesity, and and also the heart failure hits Asians 10 years lower than and at lower
BMI compared to the Caucasian population.
So this is where
we have set up cohorts in Singapore, like a child cohort, adult cohort, and midlife cohort, and so on. Mainly because the Singapore has the ethnic population of Chinese, Malays, and Indians, who represent two-thirds of the Asia. So this is a perfect location to perform translational research in Asians, and also.
And this is where I think the government is
really focusing on key areas, including the metabolic diseases.
And traditionally in
obesity, we always characterize by BMI. However, the BMI does not give you precise information about the body composition. And this is where the image that I show here on the right side, you can see
two subjects with the same BMI, one with a very high amount of fat and one with very little fat. So this is where the
BMI will not be able to define properly on the body composition.
On the left side of the panel that you see here, it shows the insulin sensitivity index against BMI.
Somebody with a very large BMI can have a good insulin sensitivity, and somebody with a lower BMI can have a poor insulin sensitivity. And this is a distribution of the insulin sensitivity from one of our cohorts in Singapore. So this is where it is very important to go into the entire body composition imaging to understand and evolve and to understand the evolution of the metabolic disease.
So just to give a
quick intro to the fat accumulation, we all know that when there is a chronic positive energy balance,
the resting energy expenditure
or exercise energy expenditure or adaptive thermogenesis.
The first
The first
location where the fat is going to go is the subcutaneous fat, which is in the edge of the abdomen. And when there is a defect, or if the subcutaneous fat cannot expand to a certain extent, then it starts spilling the fat into the ectopic organs like liver, muscle, heart, pancreas, and also into the deeper portions of the abdomen, including the deep subcutaneous fat and the visceral fat organs, resulting in insulin resistance and type 2 diabetes and other metabolic complications.
So this is where the total body fat is not the culprit, but the distribution and the proportion of fat in different locations is really the important point here.
So the graph that I show here is from one of our study on ethnic differences. You can clearly see the green line is for the Malays, and where at low BMI, they have the highest insulin sensitivity, followed by the Chinese, who have the good insulin sensitivity at the lower BMI, but the Indians have the poorest low insulin sensitivity at the same BMI. But as they all increase in the BMI, you can clearly see they kind of converge. And so at the larger BMI, the differences are vanishing. And so it is very important to study individual organs for the various fat deposits, to understand the accumulation of the fat deposits. We also have, and also the obesity is of intergenerational. So if a mother is, mother's pre-pregnancy obesity can induce insulin resistance, and when they experience impaired glucose tolerance or gestational diabetes, they produce babies which are large for gestational age. And again, these babies can grow into wrong trajectories, resulting in childhood obesity, and they become obese adults, resulting in insulin resistance. So we now, based on the Doha concept, like the first 2 years of life pretty much programs what happens to your, the way you are going to evolve in terms of the metabolic diseases. And so this is where it is now, there are several groups which are focusing on early interventions and also deciding on when to do the intervention during pregnancy or post-pregnancy, very early in the childhood. So this is a hot topic now, which is happening in several locations. So with this intro, I would like to give a quick intro for the magnetic resonance imaging, and mainly because magnetic resonance imaging is a non-invasive way of probing the body fat distribution. And you can use that in a clinical setting with the lower field strengths, and FDA has approved. Most of the clinical studies are being performed at 3 Tesla or 1.5 Tesla, but there are sites which have 7 Tesla system which are approved for research studies. And the preclinical imaging for rodents, again, it's very important, as they also provide mechanistic information for understanding knockout models or models that can show with various interventions to test the concepts rodents are being used. And so that is also very important. And of course, with very high field strengths, you can also do the developmental studies. Like, for example, the movie that is running over here is from a Xenophus embryo, which is like 2 millimeters in diameter. What we are seeing here is the actual cell division that occurs dynamically during the imaging. And so we can, of course, the MR imaging methods or high field magnets can go up to like a couple of micron resolution, and so beyond that it will be a challenge. And so that's where you lose the cell division after certain cycles. And we can also use it for other applications like developing embryos to understand the tissue interface interactions and so on. And of course, on a clinical setting, we can use it for whole body imaging and the neuroimaging. So this is where the MR becomes a very, very important way of doing repeated imaging non-invasively, all the way from pregnant mothers to adults and the aging population. So this slide shows you the signals that can be detected by magnetic resonance, which is water and fat, because they are very abundant in our tissues. So you can see from the triglyceride signals, you can detect the water signal, and you can create a water image, and the fat signal and the fat image. And of course, if you go into the details of the fat signal, then you can see, you can detect some of the other signals like allelic, biallelic, and olefinic protons. So this can also be utilized to achieve fat composition in the imaging. In the later part of my talk, I will touch a little bit on that topic. And of course, MR cannot detect bound water, macromolecules, proteins, and phospholipids, mainly because these are very large molecules and their relaxation times are very short, and so the hardware cannot capture that information. But the imaging of water and fat is well established, and we can use it thoroughly for investigating the body composition. So this slide shows you what we can achieve with the multi-gradient echo MR techniques. And so we can image the water and fat very rapidly within a single breath hold or within few seconds. And so you can capture the entire signal from different slabs of the body. And then using advanced fat water separation algorithms, you can separate the water image and the fat image. And then you can look at the distribution of quantitative distributions of the fat volumes and the fat quantity. So in my group, we have been developing advanced methods. In the old days, like 10 years back, people were using like a single slice to represent subcutaneous fat and the visceral fat, which is not very accurate. So we wanted to develop the volumetric approach so that we can quantitatively estimate the volumes of subcutaneous fat and the visceral fat across multiple cohorts. And so this is where, and of course, for each subject, it can result in more than hundreds of slices. So it will be laborious if somebody manually computes the fat depots. So this is where our deep learning based algorithms can quantify the entire subcutaneous fat and the visceral fat. And also we have implemented algorithms to separate the subcutaneous fat into deep subcutaneous fat and superficial subcutaneous fat depots. And so this is where the large cohort studies, it's pretty easy to quantify the fat across hundreds of subjects over multiple cohorts within few hours of time. And so we have worked on several cohorts at this time. So the first cohort data, which I'm showing here, is from the adult male subjects, about 20 to 30 years old, from 3 ethnic groups. And what we saw here was like the ethnic differences is very clear in the subcutaneous fat. So the Indians seem to accumulate large amount of subcutaneous fat and deep subcutaneous fat, the total subcutaneous fat and deep subcutaneous fat and superficial subcutaneous fat is definitely higher in the Indian cohort and compared to the Malays and Chinese, but we didn't see any significant differences in the visceral adipose tissues. But when we also looked at the children cohort, like at the as early as 4.5 years, this is the cohort I mentioned about that we are following this cohort all the way from newborns. Now these kids are like about 13 years old. So still we are continuing the longitudinal imaging as well as other clinical parameters that are being measured to associate the various factors of the body composition. So the surprising factor here is you can see the ethnic differences already manifest at the age of 4.5 years over here. So again, we see the Indian kids having large amount of subcutaneous fat compared to the Malay and the Chinese kids. And also we didn't see any internal adipose tissue differences. So this is really exciting and interesting. So this is where we are, and of course there are very strong ethnic differences in terms of their metabolic properties too. Though I'm not going into the details, but we have ongoing effort in separating them. In addition to the normal cohorts, we also have something called metabolically obese normal weight subcohort. So these cohort, the way they accumulate fat is very different compared to the normal subjects. And what we see here is like you can clearly see the visceral adipose tissue is very high for the MONW cohort, and also the liver fat was very high in the MONW cohort. And we also did, and of course, we didn't see much differences in the subcutaneous fat and the skeletal muscle ectopic fat in these subjects. So when you put these subjects on the moderate weight loss to improve their body composition, what we observed was, in spite of the body composition improvement, they have high insulin secretion. So if you look at the control group and the MONW group, most of the parameters are identical, including the body fat, fat mass, HbA1c, etc. But the insulin secretion is very high in the MONW cohort, which tells us that the beta cells are overactive and eventually they may fail. So this is again a very important subcohort in the Asian population, which is present in Asia. So we also have ongoing effort in risk stratification based on our cohort studies from adult males, adult females, and the kids. We are setting up a kind of a huge deep learning based methodology where we can quantify the various fat deposits, and also then we can see the different phenotypes. So now we have identified different phenotypes and associated them with their metabolic properties from the blood and also from the lipidomics and other omics that are being performed in these cohorts. And so we have, this is a very preliminary data that we are showing here, that when there is poor subcutaneous fat, that means if the subcutaneous fat does not expand properly, and then if you accumulate more intraperitoneal fat, that seems to be a major metabolic risk factor. And so we have identified a few phenotypes, which the results are in progress, and we will be updating it very soon. And this is very, very exciting in terms of the risk stratification and understanding the body phenotypes. The way it can help us in the future is to design interventions to target specific adipose tissues by either by lifestyle or by nutraceuticals. And in terms of the ectopic fat imaging, now the MR imaging is the gold standard for looking at the liver fat. For example, if you find 5% of fat fraction in your liver, then you are hitting the fatty liver condition. And again, as the liver diseases progress, we can quantitatively and repeatedly assess the liver fat and also measure the fat images, water images, and the quantitative relaxation time images. They can also predict fibrosis. There is some evidence now that we can, using the MR imaging approach, we can longitudinally assess the liver diseases too. And so in our cohort, what we have identified is very early the accumulation of the liver fat is seen in combination with impaired glucose tolerance and GDM. It seems to be predicting those subjects very early, and also in terms of the conversion of type 2 diabetes. And so those who have the liver fat, high amount of liver fat, have a big risk factor in getting into the metabolic derailment. In terms of the other ectopic fat in the skeletal muscle, again, MR is the best approach for looking at the intramyocellular lipids and extramyocellular lipids. The important point here is, even if you do a muscle biopsy, you will not be able to quantify properly the intramyocellular lipids because of the heterogeneity and so on. Whereas the MR-based approach is non-invasive, and the lipid droplets accumulate within the muscle fiber as spherical in shape, and around the fiber as a cylinder in shape. So because of the bulk susceptibility difference between a sphere and the cylinder, you can see the two signals of the N-methylene protons coming from the intramyocellular lipids, and the extramyocellular lipids get separated, and this can be quantified. And this is now well established that this lipid, the intramyocellular lipids, is highly connected with insulin resistance and metabolic dysfunction. So we have evaluated the intramyocellular lipids too in our cohorts, and surprisingly, in terms of the ethnic differences, again, Indian kids as well as the Indian women and Indian men seem to accumulate more of these intramyocellular lipids compared to the other ethnic cohorts. We also did the GWAS analysis, and we identified a SNP associated with the sterile CoA desaturase 1 gene that was explaining a higher accumulation of IMCL in the Indian kids and adults. The other point which I would like to highlight, when we do clinical MR MRS or MR spectroscopic methods in lower field strengths like 1.5 Tesla or 3 Tesla, what happens is some signals can overlap with each other because it provides a limited spectral resolution compared to the high field MR imaging and high field MR spectroscopy. So in order to resolve these overlapping signals, we have implemented two-dimensional spectroscopic methods for the in vivo applications within a single box, like shown in the slide over here. And what we have shown is the polyunsaturated to monounsaturated ratio was changing between normal weight and overweight and obese subjects. So this was performed on the Caucasian group of subjects, which again we are trying to validate in the Asian cohorts over here. To switch the gear, we also have other fat deposits in the body, like the good fat, which we call the brown adipose tissue. And again, this has become a hot topic now during the last 10 years as early, like they identified the presence of the brown adipose tissue in adults. And so this is becoming a more interesting target for obesity and to improve the energy expenditure, mainly because the brown adipose tissue can burn fat. But fundamentally, when you look at the biophysical properties between the white adipose tissue and the brown adipose tissue, you can see the unilocular lipid droplets in the white adipose tissue, whereas multilocular lipid droplets in the brown adipose tissue with a high density of mitochondria and also with the rich in vasculature. And the intermediate fat, which is called the beige adipose tissue, can be created from white adipose tissue, where by interventions like cold exposure or by nutraceuticals. And this is a very hot topic, and we have ongoing effort in imaging or doing interventions in subjects who are metabolically derailed with interventions of nutraceuticals to convert the white into beige and to improve their fat quantity and the quality. And so the MR properties, if you see here, again, the gold standard imaging for the brown adipose tissue so far has been the PET imaging, mainly because when the brown fat is activated, the FDG glucose gets accumulated in it, and that has developed a lot of interest among the clinicians and the community, obesity community. But the challenge is, like the PET imaging involves administration of a radioactive tracer, and so so you cannot do repeated imaging, or you cannot do imaging in children or newborns with the PET technology. And so this is where we are working on MR-based technology to differentiate white and the brown adipose tissue and to quantify and understand active brown fat and non-active brown fat, and also how to convert white fat into the beige fat. And if you look at the MR properties of these two adipose tissues, you can see the water content is very high in the brown adipose tissue and less amount of the fat signal, which is complementary in the white adipose tissue. And using this idea, some of the groups have imaged and demonstrated that the fat fraction is different between the fat fraction is nothing but the fat divided by fat plus water, so which is different between the white and the brown adipose tissue, and also the relaxation properties, because there is a lot of iron content and in the brown adipose tissue, so it changes the relaxation properties of the water molecules. So you can see the difference between the white and the brown through the relaxation. And using this multi-parametric approach, we have also implemented quantitative segmentation of the brown and the white adipose tissues in rodents, as well as we are currently translating these approaches into the human studies too. But the challenge that we faced was there is a overlap of some of these MR parameters between the brown and the white adipose tissue. So there is a lot more work to be done in terms of understanding the biophysical properties of these two adipose tissues. And so what I show here is the diffusion spectroscopy. Most of the literature, if you see, the diffusion of water molecules is well studied and easy to perform using MR imaging. But the diffusion of fat molecules is a much bigger challenge because the fat molecules are large and they tumble very slowly. And so the relaxation times are very different compared to the water molecules. And so we have implemented the diffusion technology in using rodent adipose tissues. And what we demonstrate was the different properties, the diffusion properties between the water and the fat molecules within the white adipose tissue and the brown adipose tissue. So what you're seeing here is the diffusion curves as a function of the diffusion value. And so when you increase the diffusion time, that is the time for the molecule to tumble, so the brown adipose, the fat molecules within the brown adipose tissue shows restricted diffusion. What that means is the fat molecules hit a barrier and bounce back during the diffusion time. And so it shows the curves get shifted. And so if you see the diffusion values, the brown adipose tissue ADC kind of reduces as a function of the diffusion time, whereas in the white adipose tissue, you don't see much changes. So using the diffusion properties, we can also model the diffusion characteristics and estimate the droplet size or within the adipocyte size within the brown adipose tissue. In addition to the diffusion properties, we also demonstrated that we can perform perfusion imaging within the white and the brown adipose tissues. Mainly, we were interested in looking at the beige adipose tissues that can be created in rodents. And so we put them on cold exposure for a few weeks of intervention. And then when you measure the brown adipose tissue, you can clearly see it is increasing with the reduction in the fat fraction, because as it becomes brown, more brown, so the amount of water increases, and so you can clearly see that you can separate the water, the white and the brown adipose tissues. And again, in the white, this is an angular area of the abdomen. So you can see the white adipose tissues, vasculature starts forming after the intervention. And so the new vasculature can be seen very nicely with the perfusion measurements, and also the conversion of white to beige, which is shown in the sparse distribution. The sparse distribution within the white adipose tissue clearly highlights the reduction in the fat fraction. So this we demonstrated for the first time that in vivo we can perform the imaging of beige adipose tissues. So now going forward, we are translating some of these methodology into the human studies, where we are doing now multi-parametric imaging, including the including the brown adipose tissue, active brown adipose tissue, and non-active brown adipose tissue. Because most of the adults, even though they have the brown adipose tissue, some of them are non-active. So in order to understand the functional properties of these brown adipose tissue, we have ongoing effort with multi-parametric imaging in combination with interventions. With that, I would like to also highlight a spin-off, adipocyte.ai, which is a A-star spin-off, which is now focused on fatty acid composition imaging. In addition to the quantity of the fat that can be imaged, we all know that the quality of the fat is also important. And so the technology that is being developed is to quantify the polyunsaturated fat, monounsaturated fat, and total saturated fat, which can provide more detailed information on the quality of the adipose tissues in addition to the quantity of the adipose tissues. So this can become a nice markers can be non-invasively determined, for example, in the fatty acid, the fatty liver inflammation, and so on. We believe that this fat composition imaging can throw in more light in understanding in some of these metabolic conditions. Of course, in the wellness market, it is also possible to look at the total saturated fat across the entire body to understand the quality of the fat and the quantity of the fat, and also determine the risk strategies and change your lifestyle and interventions to improve the quality of the fat and improve the metabolic status. With that, I would like to acknowledge my group and also the institutes and the funding from the BMRC, NMRC, and IFPP. Thank you very much for your patience. And I'll be glad to take up some questions from you.
Thank you, Dr. Senthil Velan. It is a nice, very interesting, useful talk. Very quick question. There is
Hello?
no question. You can interact with the speaker later. So thank you very much, Senthil Velan, on behalf of the organizing committee and my own behalf. Thank you very much. So let us break for the lunch. Let us assemble after 30 minutes, the same hall. Thank you.
Thanks. And yeah, if there are any further questions, anybody would like to get in touch, I'll be glad to discuss it. Yeah.
Yes, sir. Sure.
Yeah. Thank you so much, Professor Ashok Kumar.
Thanks, Ashok. Yeah.
Yeah. Yeah,
So
thank you.
is it done, or you guys are moving for lunch? I guess you must be
Yeah.
waiting for the lunch.
Yeah, yeah. Yes,
Okay.
done.
Yeah. Thank you
Yeah.
very much. Thanks for the opportunity. Yeah.
Thanks. Thanks a lot.
Sir, is there any question? We will share your email ID to the students and they will post the questions to you, sir.
We request everyone to go for the lunch arranged in the CIC space, and we expect your presence by 2:30.
Vignesh? Projector room la yaar parka? Surya Prakash.
Good afternoon to all. This this is the first post-lunch session, and the first speaker of this session is Professor Madhavan. I welcome Professor Madhavan sir for this session too. Professor Madhavan is currently a chief scientist and senior professor, head of the Department of SN and ONGC Department of Genetics and Molecular Biology, Vision Research Foundation, Chennai. And previously he was working as a professor and head of the Department of Genetics at Madurai Kamaraj University. He was one of the stalwart at Madurai Kamaraj University. And I'm very happy to introduce about Professor Madhavan sir. And previously, sir has worked as a senior professor in genetics and genomics, specialist at LKC School of Medicine, Nanyang Technological University, Singapore. Dr. Madhavan served as a overseas visiting senior scientist at University of California, Davis, USA, University of Lyon, France, and University of Oslo, Norway, and National University of Singapore. And 25 students have been completed their PhD degree under his supervision, and he has also trained postdoctoral research fellows. And Professor Madhavan has published more than 100 research papers in international peer-reviewed journals, and recently he has been funded with a DBT project under Indo-US collaborations to study genetic architecture of diabetic retinopathy. And Professor Madhavan has received various awards and honors: UGC Research Award, University of Grants Commission, New Delhi, in the year 1998 and 99. And he received Seth Baldeodas Award, Government of India, New Delhi. And sir has been elected as a fellow of National Academy of Sciences, India. So with this brief introduction, may I request Professor Madhavan to deliver a lecture. Thank you, sir.
Hello. Yeah. It's a pleasure to be here in MKU all the time. I studied the MS in MKU for my PhD. I joined as a faculty here. I served for 22 years, I think, if I'm right. I joined as lecturer and then as a reader, professor, HOD, and so on and so forth. And I trained quite a few students from the School of Biological Sciences as a genetics department. And I'm happy that my student Dr. Kumaresan is heading the department, taking my legacy and became take over my department and also my position as professor of genetics. I'm happy that Dr. Tharmalingam is here because we were together in biology and biotechnology. Now, I thanks to him for introducing diabetic retinopathy to you because I'm going to talk about same field. He talked about protein biomarkers for diabetic retinopathy. I'm going to talk about genomics biomarkers for retinopathy using GWAS data. Can I have the first slide? So this project, maybe my talk is on genome-wide association studies identified novel genetic variants in South Indian cohorts. He was telling that each ethnic group has different specificity in biomarkers. So we focus our studies only on South Indian cohort of patients, so that we know that whole of India there are lots of variations in the cohort genomic variants. So this project is funded by collaboration by Indo-US, myself in India and Dr. Sudha Iyengar from US. funded by joint program by Indo-US collaborations. I'm working in hospital called Sankara Netralaya. It is one of the famous eye hospitals in India. We have, and Dr. Badrinath is the founder, and then we call him chief of the hospital. And we have different in places like in Chennai, in Calcutta, in Tirupati, and we have two streams of hospital. I mean, one is for free patients, where you give everything free, consultations, glasses, food, transport, everything free. It's called free patients. Other one is called paying patients who can afford to pay. We charge them, and then we subsidize those, use the money to pay for the free patients. In fact, many companies in India like Wipro, Google, all the people donate a lot of money to institutions because we take a do service for free patients. So this is my group in my lab, and so this is the first systematic study in the whole of India. This is first systematic study to look at the non-genetic parameters of diabetic patients and also diabetic retinopathy patients. Probably this is the first study for GWAS in India, though not only for South Indian cohorts, the entire Indian populations. So this was funded by DBT, and for my collaborator Sudhayangar, she got funding from NIH. She got about $1 million, and we got about 1.5 crores or so. So this is, we work on, first of all, when you look at the genomic studies, we need to recruit patients. There's a major fulcrum of the whole project is patient recruitments. So we try to, I think, offering a protocol. So the major, the fulcrum of the project is recruitment of patients. So now we have to collect samples for the patient recruitment for any of our studies. So we had recruited quite a lot of patients. It took about 3 years to recruit patients. It's a big task of going to different camps, from hospitals, running at some rural centers, follow-up cases, so on and so forth. So we recruited patients, and then we collected the image data for them, taking the fundus pictures and then OCT image, the retinal scans of the patients. We collected non-genetic data. That means non-genetic data, what I mean is how long they are diabetic, what is their blood sugar level, what is their cholesterol level, what's their hypertension level. So there are different parameters. We take all this into account, each patient. That's called clinical data. Record all the data, and then that will be correlated to the diabetic and non-diabetic DR cases. And then comes the genetic data, where we take the DNA. And then we did two major genomic technologies. One is we used genome sequencing of the patients, case control, and so on and so forth. Other one is called genome-wide associated studies using the case control samples. Genome-wide GWAS was done with my collaborators' help. The funding was from NIH. Samples are now, the data is out now. So how we recruit patients? The student in the project is called Riza Naim, and she's the one for the entire project single-handed. But of course, many technicians helped in recruiting patients. So we meet the patients, we talk to them, we take the pedigree, what the parents, family pedigree, who are diabetic in the whole family, and so on and so forth. And then they go for fundus examinations and the OCT examinations. Then they go for eye examinations by clinicians. Then we decide who is diabetic retinopathy. And a DR is also, they rate them as, we call different grades of DRs, proliferative, non-proliferative, and so on and so forth. I think Dr. Dhamalingam gave all the details. So on the whole project, we recruited totally about 3,000 patients, about 2,800 patients, of which 8,800 patients are from diabetic retinopathy cases. That means they have the problem in the retina, they're diabetic, and also additionally they have diabetic retinal issues. There are 2,000 patients who are diabetic but no retinal issues. That's very interesting. Why? Not all patients diabetic do not get retinal problems. We'll come back to that later, actually. So this took almost 2.5 years to recruit patients and then get all the entire clinical data. And it was a very successful project. And then the project was successful because we had a very coordinated meetings with the Indo-US collaborators, our collaborators, every alternate Friday. Every alternate Friday we used to meet and talk. But it goes for an hour or so, a Zoom meeting. So even now we are meeting, we were talking every alternate Friday because the data is to be published now. So we are analyzing data and then it is going to take another 6 months to 1 year to publish. So we took the images of OCT and then clinical examinations. Some images for those who are students, for those who are not familiar with the images. This is the fundus images of the eye. So this is normal. This is a moderate DR. This is the real PDR, the progressive diabetic retinopathy. That means you can see lots of holes and then destructions in the retina where you lose vision. So if we look at the retinal scan, this is a normal retina. You can see this is a beautiful retina. It's very clear in normal scan. This is the focal point. And as you get more and more diabetic, the retina get detached. Now you see a big hole here. The retina get detached, so you don't get the vision. So this is how we grade them as PDR, NPDR, and so on and so forth. So we get of course there's a parallel analysis going on, image analysis, and then correlating to the clinical data. We are trying to make an AI-based tool which you can predict. If we give you the clinical data, it can tell you what is the disease status and how fast the disease can progress, because we have about 5 lakhs of data on image and also on clinical data. Entire data is being put into the we're just massaging them to generate algorithm. This is recently funded by DBT, where we are trying to make some algorithm by which you can give the image, it can tell you the disease, or you can give the clinical data, it can tell the disease. So this is a combination of just first year of the project is going on. So few data about non-genetic factors. Non-genetic factors means clinical analysis. So we look at age of the patient, gender, how long they're diabetic for how many years diabetic, what is the HbA1c levels, cholesterol, and then albuminuria, hypertension. So you look at all these parameters for each patient. Almost 90% of the patients we get the data. Sometimes you don't get them. So this is a very interesting story I tell you. This is the range of HbA, because people say HbA1c is the measure of diabetic, and also it leads to diabetic retinopathy. Okay, if you have more diabetic HbA1c, you are going to get diabetic retinopathy. This is what the notion everybody has. Look at this case. This is a diabetic people cases, diabetic retinopathy cases. They have 6.4 normal HbA1c level, but still they are diabetic retinopathy. They are DR cases. It's not that they should have a very high HbA1c value to get DR. Even those who are normal, less than 7 is normal. There's less than 7, 6.9, still they have diabetic retinopathy, retinal issues. Look at this controls. They are the same 6.7. There's no retinal issues. Interestingly, we look at their HbA1c level. Some patients have 11 or 15. In the controls, they do not have retinal issues. They're almost like a cases. Diabetic retinopathy cases, 15 or 11 is HbA1c values. They are retinal issues. These patients do not have retinal issues. So there's not only the HbA1c is the one factor that can determine that you're going to get DR or no DR. A lot of other issues, other factors. In fact, as Dhammalingam was telling, if you are getting the HbA1c, though you control HbA1c at 6.7, you get DR. The reason being that you have been insulted with the diabetic long time ago, because you identified recently your diabetic cases, but you have been diabetic for maybe 20 years before you identify the cases. So you are insulting the early insult to the body by the diabetic conditions where you don't measure in proper time. It can also lead to the DR cases, even you have low HbA1c values. So we measured all these things. We made some calculations. Ultimately, we came to conclusion. In males, microalbuminuria is a risk factor for DR. Not worry about duration diabetic or HbA1c, but in males, major factor that can lead to DR is microalbuminuria, more risk factor. But if you look at another analysis, in male gender, again, poor glycemic condition, that's HbA1c values, and also the duration of diabetic, long-term diabetic can be the reason for, this is for DME, that's for DR. So this is published recently. So we are trying to next, in going to the genomic phase, we are trying to do some whole genome sequencing of the case and controls. We took a pedigree. A pedigree means, we try to identify father, mother, and then family 1, generation 1, 2, 3, 4, and so forth. This pedigree is a big family, a lot of people, pedigrees in the family. So we collected about 2 families which are endogenous. They're all cousin marriages. We can see this is a lot of consanguinity, quite consanguineous family. See, we have 2 lines means they married to the cousins. So complex pedigree. So we mark them as healthy controls. These healthy controls, diabetic macular edema, diabetics only, diabetic control. So we separate them as different cadres based upon their phenotype and all clinical data, and another family. So we get the entire data. We try to sequence some whole genome sequencing of the entire. We did 31 families, 31 members of the family. Some are diabetic, some are diabetic retinopathy, some are healthy controls, and try to identify whether we will be able to find any genetic variants which are associated with the diabetic retinopathy. So this is a lot of work. Of course, sequence was done by MedGenome. We didn't do it because the quality was quite good. Mapping was quite good. Genome coverage was quite nice. So then we used lots of tools to analyze the pipeline, genetic variants. So this was done by a guy called King, who is a collaborator in the project. The entire data was put into lots of tests. We used, when we filter the genetic variants from genome sequencing, we used lots of strategies. We took a CAT score 20 and above as a basic filter. Then we filtered the mutations based upon SWIFT, polyphen, mutation transducers, and non-synonymous, synonymous variants. So these are all filters by which you can say which variant is really associated with the disease. If you look at the whole genome sequencing, you will see millions of variants. Every individual is varying in the genome sequencing. So you put the entire genome sequencing, you see millions of variants, but filter them in different strategies and identify which are the variants most likely to be associated with the disease, either with the diabetic or with diabetic retinopathy. Even then, after identifying this type of 89 variants, you have to validate them. It's not that you can just accept there are real associated with disease. So we filtered about 89 variants, and then we were trying to validate a couple of them as students on the projects. We identified one particular variant in a gene called G protein coupled receptors 6A, CP6A. So this is a specific variant. Now one reference sequence, RSID. This RSID is associated with diabetic retinopathy patients and diabetic patients in the sequencing of the whole genome of the whole pedigree. So when we were trying to, we do the validations, this is quite complex. So once you have identified this is the probably associated with disease, you would validate. Look at this particular variant in about 200, 300 samples of controls, and 200, 300 samples of diabetic cases, or 200, 300 cases of healthy controls. Then check whether you find the variants only in the diabetic cases or only in the DR cases, but not in the controls. So it's you have to do by one. It's not by mass array. You have to do each individual. You have to get the DNA, make PCR of the specific locus, amplify them and sequence them and check the genotype. This genotype is either a wild type or is a mutant. So it's about 600, 800 sequencing, Sanger sequencing for identify one variant. So we did for only one variant, I mean, with 2 variants, I'm going to show only one sample here. So this particular RSID is so highly associated with the disease, and we looked at the structure of the protein. What is the protein? Where the mutation is? The mutation is in the transmembrane domain. So this has its role to play. It's a non-synonymous transmembrane domain. So it changes the protein amino acid composition. So then we looked at the functions of this particular SNP, and then how does it affect the population? The case. We looked at the structure. These are all graphical. This is all made by computers. Somewhere here, the transmembrane domain is the changes, and then we see the changes in the single base pair mutation changes the structure of the protein, which will facilitate the case. So we are trying to now. I'm trying to go to focus on the genetic variant using common and rare variants using GWAS studies, genome-wide association studies, wherein we collected entire samples collected from the whole project, about 2,000 to 800 samples of DR and 2,000 samples of non-DR diabetic patients were extracted, and then they were put into the technology called genome-wide association studies. This technology will identify the unique rare variants or unique variants which are associated with the diabetic patients or diabetic retinopathy patients. It'll lots of analysis, lots of data, lots of tools and then analysis pipelines. Ultimately, we show them in this is the composition of the samples we use for GWAS, mild NPDR, moderate NPDR, no DR, PDR, severe PDR, unspecified DR. So we categorize them on different based upon clinical analysis. We categorize them into different subgroups, and then we did the GWAS, and we used quite a few methods to 1000 genome and TopMat, and then so all these analyses were used. It's not done by me, by a guy in the US, a collaborator. So we ultimately got a Manhattan plot, which says that in chromosome like for chromosome 3, a particular loci is highly associated. In chromosome 7, you see a particular loci is highly associated. And chromosome like 12 or 13, you see 11, 12, you see a particular loci associated. So entire data generated by the GWAS is put in different pipelines, and then we should pick up and show, okay, these are all the genetic variants associated with either diabetic or diabetic retinopathy. Now, once you get the entire data, it's going to be, we look at their either they're intronic or the exonic or they're intragenic, are they what's the CAT score, what is their MAF function, what's the MAF score, and we look at all these things and then check either these variants have been reported in any other population in the world or the unique for South Indian cohorts, because we focus only on South Indian cohorts. So when you look at this type of analysis, we did extensive analysis. Now we are still doing analysis going on. So we try to identify the data for diabetic, any DR combined with the HbA1c and then diabetic duration. And also we look at any DR duration and then cholesterol levels. So this type of, we try to put the data into different combination and get the genetic variants, so that ultimately we pool all genetic variants and then try to check which one is more significantly associated, which may be the risk factor for diabetic or diabetic retinopathy. So the data analysis will go for another 3 months to 6 months before we write the manuscript. So this is a set of genetic variants identified. This is the chromosome number. This is the ID, this is locus, which is this 1000 genome mapping. What's the p-values? So stop mapping, and then you map it different databases, and then try to identify what's the p-value for this particular variant, a given method of filtrations. So we have did extensive filters, and then this is another table showing this again same table, and then but different filtering methods. Ultimately, we identified a set of variants which are highly associated with diabetic or diabetic retinopathy in South Indian cohorts. So we cannot take it granted. It's okay. It's all right. This is the cohort. This is the variant, and no risk factor for the disease. So we have to check them in a population which is not used for the GWAS. Is we call control screening. We have to take similarly a set of patients and then controls and cases from not used for the GWAS data, and then screen them in the population and check whether your finding is correct or not. If you are going to say, fine, okay, this the variant identified in the GWAS study, which is associated with the diabetic retinopathy, you find them in that DR patients in the wild collections in the new set of data, then you confirm this is associated with disease. You don't find them that it's a some mismatch or some or what you call non significant data. So we identified a couple of RSIDs from the GWAS data and looked for the publications. Is there any of these RSIDs identified in South Indian cohorts or have any shown earlier that are associated with any of the diseases, either diabetic or diabetic retinopathy? We found none of the RSIDs have been. There's no publication in about this RSID. That means these genetic variants have been identified the first time. Their association with the diabetic or diabetic retinopathy identified first time in the cohort of South Indian populations. So this is as Dr. Damani was telling, the ethnic groups have different these variants may not be associated with the European or Caucasian populations, but within Indian populations, maybe South Indians, North Indians may be different cohorts. Different variants associated with disease. So we checked them. Just show a few because we have about few hundred significantly associated variants, and also we looked at them also we checked them also in Google, directly Google and Google Scholar, no data available. So we show from this our data that from the genome sequencing data, from the whole genome sequencing data, and also genome with association studies identified few markers, few genetic variants, rare variants in fact, which are associated with diabetic or diabetic retinopathy in South Indian cohorts. This is our finding. We need to validate them, and this is funded by Government of India and National Eye Institute by the NIH. That's it. Thank you.
Thank you, sir. Now the session is open for discussion.
Very interesting talk.
Thank you.
Just to the last part, did you look for genes close to these variants that you found? So assuming that the variants might be just marker variants, that the real difference is in a gene close by.
I didn't get you properly.
So if you look for genetic variants, you might find them anywhere in the non-coding genome.
Yeah.
But the actual gene that confers the risk might be close to that variant. So if you don't find publications for the variants you have found, maybe you find some for the genes that are close by.
You're correct. Perfect. So sometimes we find them in intergenic regions, the variants and intergenic regions. Probably the flanking genes might be involved in the same disease. So in fact, I filtered out some of the data where we don't see any hit for the RSID, but for the gene we get the hits. Exactly. No, I got the point. Sometimes RSID may not be associated, but the gene is associated. But some of them, these ones are totally not associated, even gene not associated. So filtered out those variants and then I bring it here.
Sir, you mentioned that the diabetic mellitus patients, they may not have diabetic retinopathy. But sometimes the diabetic retinopathy patients, they are not DM. So how it would be originated? It could be the reason of mutation, sir?
No, I think we have done a project which I did show you here. We took patients who are 5 years diabetic, 5-year diabetic with no retinal issues, and 5-year diabetic with retinal issues. Also, patients who are 30 years or diabetic, they've been diabetic for 30 years and above, no diabetic, no retinal issues, and 30 years and above retinal issues. We took the 4 cohorts, and then we did DNA methylation studies. We found interesting data. DNA methylation can regulate or it can be a marker why the people who are 20 years diabetic, high HbA1c value still don't get retinal issues. It's a more of a combined data. It's more of a epigenetic regulator things. Dr. Dhamalingam also mentioned some people who are diabetic even very early after getting diabetic, but some people are. I know cases. We have screened cases where people are 30 years 40 diabetic. Our own patients who visited our hospital 30 years ago for diabetic, still they're coming, but no retinal issues. They come for only for diabetic treatment, not for retinal issues. But they come for screening, they're still perfectly all right. Their vision is 6/6. Then no retinal issues at all. So genetic plays a very important role in that actually, and epigenetics roles as well.
Okay, sir. Thank you, sir. So with this, thank you very much for your elaborative and very informative talk, sir. Thank you so much.
Thank you.
So now I request Professor Dharmalingam sir to give a memento to Madhavan sir. Yeah.
Just say Dharmalingam or Madhavan, don't say sir. It looks odd, you know.
We thank the organizers for having given us the chance to chair this session in the forenoon and in the afternoon. So thank you very much for your patient listening. Thank you for the speakers as well. Thank you.
So good afternoon, friends. I deem it have a great privilege to welcome Dr. Ashok, Dr. Rajendran to chair this session. Now the dais is over to the chairs.
And myself, Dr. Ashok Kumar, and he's my colleague, Dr. Rajendran. Thanks for the organizer for giving this great opportunity to serve as a chair for this wonderful session on genetics and genomics. This session consists of 4 eminent lectures from eminent professors, including Dr. M. Ganeshan from Presidency University, Calcutta, and the second one, Dr. Ravi Marudachalam from iCURE Trivandrum, and third one, well-renowned eminent scientist, Professor Kaliraj, and fourth one from Dr. Steve Reich. He is presenting his presentation through online mode. The first presentation of this session is going to be by Dr. M. Ganeshan, who is an assistant professor in the Presidency University, Calcutta. He is specialized in plant genetic engineering. He has been servicing at this institute for last 10 years. And more than 30 publications he has so far published. And he got many awards through which he visited 3 or 4 countries as a postdoctor. And many young scientist award also he has received. And with this introduction, I invite Dr. M. Ganeshan to deliver his presentation. Thank you.
Hello, can you hear me?
Yeah, thank you. So thank you very much, Professor, for the very nice introduction. Actually, I am also from Madurai Kamaraj University. I have completed my undergraduate and masters from Anja College, Sivagasi. So how to, sorry. My PowerPoint? Yeah, okay. Yes. Thank you. Actually, I am from Presidency University, Kolkata. So last week only we celebrated 208th birthday for our university. Today I selected this topic. So development of transgenic plants by overexpressing photoreceptor genes. Particularly, I selected phytochrome gene for this presentation. Yeah, before entering into the topic, I wish to give a brief introduction about photomorphogenesis. So photomorphogenesis means simply the growth of plants under light conditions. If you keep the plants under dark conditions, the plants will grow like this. You will find etiolated shoots, white, it will be white color and hook formation in the shoot tip. And if you keep the plants under light condition, particularly the shoots, it will move towards the light. We call it as phototropism. And this one we call it as photomorphogenesis. And all these parameters are regulated by different photoreceptors like phytochromes, cryptochromes, phototropins, and UVB receptors. In this presentation, I am going to focus only phytochromes. Yeah, so they developed different mutants to study the functions of different photoreceptors. Like 5 different forms of phytochromes are present, and 2 different forms of cryptochromes and phototropins are present. So phytochromes alone are responsible for seed germination, and phytochromes and cryptochromes together are regulating seedling development and de-etiolation responses. Phytochromes and cryptochromes again, they are helping for transition to flowering, and very importantly, the phytochromes are helping for neighbor direction and shade tolerance responses. And phytochromes and phototropins together, they are helping for phototropism movement. So if we check clearly, almost all the plant growth and developmental stages are regulated by phytochromes, and phytochromes are interacting with other photoreceptors. So this one recently we published it. So phytochromes, after the biosynthesis of phytochromes, it will enter into the nucleus, and you will find 2 different forms of phytochromes, that is PR form and PFR form. After receiving red light, the PR form will be converted to PFR form, which is biologically active, and then PFR form, it will go back to original PR form after sensing the far red light. And this PFR form is very sensitive. After approximately 3 hours, it will be degraded by the usual proteolysis pathway. And if we check the functions, the phytochromes are regulating different plant and growth developmental stages by regulating several genes and transcription factors, like phytochrome interacting factor, like phytochrome interacting factors, and constants mainly. These are all very important for plant growth and developmental stages. So based on these importances, several transgenic plants were developed by scientists. So I wish to focus only the rice and tomato transgenics in this study. So in this table, if we check clearly, the rice plants overexpressing phytochrome A, they showed dwarf phenotypes. Usually, dwarf phenotypes, if you produce, they will produce too much yield, and the plants were displayed increased chlorophyll content and smaller tiller numbers. And so SAR indicates the downregulated lines. Downregulation of phytochrome B displayed a reduced leaf area, which is important for drought tolerance responses. In another study, they identified chilling tolerance responses in rice plants in which the phytochrome B, it was downregulated. And in tomato plant, overexpression of phytochrome A displayed that phytochromes are very important for food development and ripening. So like that, cryptochrome 2 overexpression displayed a very important, agronomically important trait like increased flavonoids and lycopene content. So based on these reports, we plan to go with the phytochrome gene for our studies. Further, so this is simple biochemical mechanism involved in phytochrome. So this is original phytochrome PR form, PFR form. After phosphorylation, it will be easily degraded. So this S599 site is important for this autophosphorylation, and in PR form, so S8 and S18 are involved. So we plan to mutate this S59 site. So we developed CRISPR-Cas9 for base editing, as well as we developed the gene that is phosphomutant gene. So we mutated that serine to alanine, and then we overexpress this gene in different plant species. So if we do that, the possibilities are there. Higher accumulation of PFR form will be there, and it will enhance the plant growth and development. We plan like that. So for the first study, we selected Zoysia grass plant, which is warm season grass. Apart from that, the grass plants are having so many advantages, like it will control soil erosion, and it will filter the herbicides or the insecticides to escape from the agriculture field. This is a simple image of Zoysia grass. You will find tildes like this, and this is the runner and root system. It's a very common tough grass. So simply we followed callus-mediated regeneration. So these are all somatic embryos. We analyzed it by using SEM also. A simple regeneration protocol, and we got a root system in the MS basal media without any plant growth regulators. So this is our vector used for the study. So we developed 3 different type of transgenic plants. The first one with the phytochrome A wild type gene. Second category with a mutant form of phytochrome that is mutated in S59 position. And then third one without the gene of interest acid. That is control vectors. So after developing the transgenic plant, we challenged the plants with the herbicide tolerance test. Only the transgenic plants which were showing good shoot growth were considered for further analysis. So simply under in vitro conditions, we used phosphonothricin, that is herbicide, in the media. Usually phosphonothricin, it will kill the plants if it's non-transgenics. And we simply followed the fat protein assay test to screen these putative transgenic plants. Further, we did the blotting test to check the gene integration. So firstly, we did the southern blot analysis by using 2 different restriction enzymes. All the transgenic plants displayed single copy integration, and then we checked the amount of mRNAs produced and then protein produced. Based on the Western blot analysis, we selected phytochrome wild type number 1 and mutant phytochrome number 14 and 18 for the further studies. Yeah. Thank you. Besides, we also did some field experiment. In the field, we maintained our transgenic plants to check the pollen dispersal or horizontal gene transfer things. So after herbicide treatment, the weeds were killed by herbicides. Only the plants were survived. So in the first set, so the transgenic plants, as we expected, the transgenic plants were sorted when compared with the controlled transgenics. And they produced too much amount of chlorophyll when compared with the control plants. So this is simple figure. And then later we challenged our transgenic plant with the different heavy metal, that is sodium chloride salt and zinc chloride heavy metal treatments. And 214 and 18 also showed greater chlorophyll amount when compared with the control plants. And this is phytochrome A wild plant. This one also showed approximately 5% increase chlorophyll when compared with the controlled transgenic plants. So if we check this figure, we checked the number of tillers formed in the transgenic plants. This is like top view of our field. The number of tillers were significantly increased in 14 lines when compared with the phytochrome A or in control plants. And accidentally we recorded cold tolerance in our field conditions. So during September month, we photographed this image. So all the control and wild type plants were killed by the cold stress, that is natural cold stress. Only phytochrome A, 214 and 218, they survived. So based on that, we further proceeded our experiment. So we checked the photosynthetic values by using a photosynthesis analysis meter. If we check the performance index total values, It is here. There is a red color that is 18 and violet color that is 14. So maximum values under cold stress conditions means the plants were not facing the cold stress, approximately 4 degrees Celsius, around 4 degrees Celsius. So, and this is the yield parameters, and surprisingly under field conditions during the inflorescence formation, the plants they showed regular growth. If you check the control and control transgenics, the growth rate was very high when compared with these two lines. And interestingly, the inflorescence axis, that is a seed bearing organ, it was taller when compared with the control transgenic plants in phytochrome A wild type. And 214 and 18 also it was significantly taller. So we checked the yield parameters, particularly we checked the weight of 500 seeds. We collected the seeds from the field and then we checked the weight of the approximately 500 seeds weight. The phytochrome A transgenic lines showed higher seed weight, means healthy seeds, when compared with the control plants. And 214, 18 also showed significantly higher values when compared with the control plants. And we checked the total number of inflorescences per square foot. I'm sorry. And in 214, 218, the higher values were recorded. So altogether, this 214 showed higher yield when compared with the non-transformed wild plant. That is approximately 40% increase. If we check phytochrome wild type, that is approximately 20% increase in the total yield. So further, so this work was performed in South Korea in Jeju Island, and further in this island we performed, this is South Korea mainland. So further our Indian government and the South Korean government, they approved for field test. So we maintained our plants here in the open field condition, transgenic plants, and then we checked for the transient escape from the field. Approximately 5 kilometers radius, we collected plants from plants of different Joycia japonica and Joycia metallica plants, and then we checked for the presence of PAT protein. So totally 112 zones were selected from 448 sites, and from all these sites we found only no positive response. So indicated that these plants are not producing any pollen grains which are having the cross-pollination efficiency. So we were able to reproduce the same results in bentgrass plants also. This is also a type of tough grass. The transgenic plants were shorter than the wild type plants. And this is Arabidopsis, and this is sweet potato. You can see the variation, clear variation. And the leaves were broader than the wild type plants. The leaves were broader and greener. I'm sorry. And if we check the yield, approximately 8-fold increase the yield we recorded, approximately 8-fold when compared with the wild type plants. So based on these observations, we wanted to conclude that the photoreceptor transgenics are very important to develop abiotic stress tolerance plan, and it will help for crop food security issues. And due to the biomass, increased biomass production, we can use these plants for bioenergy production. And these plants are environmental friendly because we don't need to apply too much herbicides or water or fertilizers, because the plants are growing tightly. And then the fruit quality improvement was recorded in tomato plants. So high anthocyanin and lycopene content was recorded. So these are all our publications. So we started these experiments in 2012. This was our first publication. From 2008 we are working in this project, and currently I am working with sugarcane and tomato plants with this particular gene. It's not going. Yeah, I wish to thank my university for providing a FRPDF scheme, and I wish to thank NRF of South Korea for providing visiting professor fellowship. This process, I wish to thank Professor Song. He's the mastermind behind this project, and I wish to thank Professor Strasser, Professor Kim, and Lee for their excellent contribution for this project. Thank you all.
Thank you, Professor, for the wonderful lecture on Development of transgenic plants by overexpression photoreceptor genes. Any question from audience?
So you were showing some slides that you did some field trials, right?
Yes, ma'am.
So those plants were developed in India or during your visit in Korea?
We developed it in India and then we carried it to South Korea to perform these studies, ma'am.
Okay, can you just tell what modalities, like you have to get the approval and all those things when you do the transgenic?
Yes, ma'am. It's not easy in India, ma'am.
Can you
In
just
South Korea,
explain?
it's very easy, ma'am. So that project is it was supported by Korean government directly to check the pollen grain escape from the research field to nearby environment. So if you need, I will give you further contact, ma'am. No problem.
No, it will be useful for our students because especially those biosafety aspects. So that part we are requesting you to just say a few words. Like
Yes,
what
ma'am.
modalities you have to go when you want to transfer from here to Korea or something like that.
Well, the first thing, ma'am, they will not allow you to take the green tissues. You have to go with the seeds, and then we need to get government approval, particularly from the airport. I have all the details, ma'am. I'll provide it to you later.
Any other questions? Since there are no questions, I close the session. Thank you so much, sir.
Thank you, sir.
Now I invite Dr. Sridevi for sharing memento with Dr. Ganesh. Thank you so much.
Yeah, so the next speaker of this session is Dr. Ravi Marudachalam, an associate professor from iCURE Trivandrum. So, welcome, sir. So, he did his MSc in agriculture and then completed PhD in CCMB and got degree from JNU. And then he did postdoctoral research fellowship at University of California, Davis. And then he received Best Research Award from University of California, Davis, and DuPont Young Professor Award. And then he came to India back through Ramalinga Swami Re-Entry Fellowship, and then he is leading a group in plant genetics and genomics at iCURE Trivandrum. Welcome, sir.
Good afternoon to all. So at the outset, I would like to thank the organizers for giving us an opportunity to share our work. So my lab at iCURE works on understanding the molecular mechanism behind uniparental genome elimination in plants, and that's the basic aspect. And then applied aspects, we are also interested in developing tools for accelerated plant breeding and genetics. So in the first half of the talk, I will talk about the basic science, and in the second half, I will talk about how we can apply uniparental genome elimination for crop improvement. So I understand this is a mixed audience, so I would like to briefly introduce what is uniparental genome elimination, and then I walk over in detail in the forthcoming slides. So as the name itself suggests, uniparental genome elimination is a phenomenon in which after successful fertilization between when we cross two parents, one of the parents gets selectively eliminated, and depending upon which genome is eliminated, you end up in producing a uniparental progeny, and if the maternal genome gets eliminated, you end up in producing a paternal haploid, and vice versa. Whereas in the normal scenario, you end up in producing a hybrid progeny. So why we are interested? So in this uniparental genome elimination, okay, is not a desired outcome in animals or in humans, whereas in plants, it's a multi-million-dollar business. Why? Mainly for the reason that, so the haploids and when we self the haploids or double the haploids, we end up in get double haploids. So their double haploids constitute an important So the double haploids, they constitute an important plant breeding tool for the rapid development of inbred lines. So the conventional method to develop inbred lines is to cross two parents, and the F1 is 0% homozygous, and then through repeated inbreeding and cell pollination, you take that strain. So again, as you can see from here, in the F8 generation, you get only 99% homozygosity. And even if you go F50, you will never ever reach 100%. On the other hand, by producing haploids, so the haploids, since they don't have a pairing partner, so they are 100% homozygous for all the gene loci. But haploids per se is not useful for plant breeding because they are sterile. Okay, so in order to be useful for plant breeding, it should be converted to fertile plants. And how we can convert a sterile haploid? So just by doubling the chromosomes, you can make something called double haploids. So the double haploids, they are 100% homozygous, and as you can see from the picture, so the conventional method, it takes several years of inbreeding to achieve homozygosity. On the other hand, just by developing haploids, we can achieve the homozygosity within 2 generations. So this is in fact as a useful trait in plant breeding because you can fasten the variety development at least by 6 years. The conventional method, it takes around 12 years to launch a variety, whereas by double haploid breeding, you can cut short the development to 5 to 6 years. So that is a major advantage of the uniparental haploid progeny. So again, the commercial importance of haploid can be understood from this chapter in this book entitled Advances in Haploid Production in Higher Plants, where a separate book chapter has been dedicated for discussing about the patents and haploid plants. So you can imagine how much voluminous number of patents might have been filed and granted for producing haploid plants in plants. So coming to the thing, so whether this uniparental genome elimination occurs in nature, the answer is no. It is not seen in nature, but it is induced artificially by the plant breeders. So basically, so uniparental genome elimination is observed as one of the genetic consequences of distant crosses. That means you cross 2 different species or 2 different genera. So the outcome of such forced marriage between 2 different species is that you end up in producing interspecies hybrid if both the genomes exist in harmony. If not, one of the genome gets kicked out, and then you end up in producing a uniparental progeny from the winning parent. Okay, on the intermediate scenario, you also do get aneuploids, and that give rise to chromosome elimination or substitution lines. So we do have examples for all categories in plants. So this has been mostly reported in plant species belonging to Poaceae and Solanaceae, and also one example in intraspecific cross in maize. Then this has been commercially exploited for the haploid production in the maize worldwide. So, this phenomena has been observed by plant breeders as early as 19th century, but the molecular mechanism was poorly understood. So several mechanisms have been put forth to explain the mechanism of uniparental genome elimination as listed in here. And out of these several hypotheses, so the one which now we have evidence is the last one, that is parent-specific inactivation of centromeres. So now I just briefly introduce what are centromeres. As all of you might know, they are chromosomal loci that are responsible for proper segregation of chromosomes during both mitosis and meiosis. If there is a defective centromere formation, then it leads to imbalanced chromosome segregation and results in aneuploidy. And the research from several model systems have shown that centromere is in fact epigenetically determined, okay, by the simple localization of a protein called centromere specific histone H3, which we call it as CENH3. So basically what CENH3 does is that it selectively replaces the canonical histone H3 from the nucleosomes of the chromatin. Okay, so wherever this NH3 goes and localizes, that region can be specified as the centromere. So that is the reason why we say that the centromere is in fact determined epigenetically. So by, for example, if you target to the one corner, then that region can be specified as centromeres. So basically the localization of centromeres to the chromosome determines where the centromere should be specified. So this provides a platform for the assembly of a multi-protein. Okay, so if you tag the SENH3 with GFP. So this is what you see. So you can label all the centromeres. So what you see is a Arabidopsis flower labeled with GFP SENH3, and you can see a cluster of green dots, and each cluster represents a nuclei, and each dot represents the centromere. So you can clearly count 10 centromeres corresponding to the 10 chromosomes of Arabidopsis. So the loading of SENH3 is essential for the building up of this multi-protein complex called kinetochore. Without this loading of SENH3, the 100 and odd protein complex will not be assembled. Without this, there won't be proper segregation of chromosomes. So we isolated a SENH3 null mutant in Arabidopsis. Like sticking with the function, so you can see that if you mutate SENH3, it causes embryo lethality. That means as you can see from the fruit from a heterozygous silk, the white ones are the aborted seeds. Because of the absence of the SENH3 protein, they die in the early embryonic development itself. So now we wanted to address what makes SENH3 specific only to the centromeres. So because it selectively replaces histone H3 at the centromeres. So we did a protein sequence alignment, and then we figured it out that, as you can see from the alignment, so the protein can be divided into two parts: the N-terminal histone fold domain and the N-terminal tail domain and the C-terminal histone fold domain. And this is the C-terminal domain that is in contact with the DNA in forming the nucleosomes, whereas the tail one that is hanging out from the core, and this is the one that is site for all post-translational modifications of histones. So as you can see from this alignment, so there is no conservation at the N-terminal tail, whereas the C-terminal tail is highly conserved. So we thought that, okay, the centromere specific localization of SENH3 rests in the N-terminal tail. So to address this, we had a genetic null mutant. So to address this, what we did is that we made a lot of chimeric constructs. So this ones which I am going to discuss today is the tail swap construct. So what we did is that we removed the SENH3 tail, and then we replaced with the histone H3 tail, and this chimeric construct is called tail swap because we swapped the tail, and this we attach to the N-terminal GFP, and that is called GFP tail swap. So now we ask the question, if tail is important, then if we put back this chimeric construct into the null plants, then whether it rescues the embryo-lethal phenotype or not. And when we did it, we found that in fact it could rescue the embryo-lethal phenotype. And then, so this is how a normal wild-type Arabidopsis looks like. And this is the one which is the GFP tail swap hereafter refers to the plant, which is complemented by the altered SENH3 variant, that is the GFP tail swap. As you can see from here, so the plants is highly compact in nature, and the only difference between these two plants is that they're otherwise isogenic except for the centromere histone. So this wild-type plant contains the wild-type SENH3, and the GFP tail swap plants contains the GFP tail swap at the centromeres. So now, if we cross these two plants, so what happened is that okay, if we cross these two plants, we were surprised to see that instead of producing full viable seeds, we got around 20% viable seeds, and most of the embryos were undergoing abortion, which is a surprising result given the fact that the SENH3 is heterozygous in this cross. And when we germinated the viable progeny, what we found is that we could found, like as you can see from this picture here, so all the plants were completely, showing different phenotype. And then to cut the long story short, we figured out that so this F1 progeny contained flavors of ploid, containing haploids, aneuploids, and hybrid diploids. So basically, we did a genetic analysis, and here is the cytological evidence to show diploids, like you can see 5 bivalents, and in Arabidopsis haploids, you see univalents, and we did a lot of genetic analysis, and we showed that the haploids are always uniparental, originating only from the wild-type parent, not from the mutant parent. So that means upon crossing, the mutant SENH3 genome gets eliminated, and you end up in producing progeny exclusively from the wild-type parent. And then, as I mentioned, for application purposes, it should be double. The chromosome should be double, and the haploid should be made fertile. And this we did it by by treating the plants with the spindle poison colchicine. So by applying colchicine, we can spontaneously or double the sterile haploids to a fertile double haploids. And we have also shown over past 10 years or so, we have shown that not only the GFP tail swap, so several other modifications of SENH3 can also induce genome elimination. So what are those? So they are null mutations in SENH3. Even a single point mutation in SENH3 is sufficient to trigger the genome imbalance between the mating parents. Apart from that, SENH3 depletion by RNAi and also overexpression of SENH3 in maize also can trigger uniparental genome elimination. So these are simply the different alterations that you can do for SENH3 to achieve this uniparental genome elimination. And what the studies from Barley have shown that the uniparental genome elimination results from the absence of loading of SENH3 to the centromeres. So you can see the above panel is an elimination cross, and as you can see in the panel A, there are chromosomes in the middle which is lagging behind when the other chromosomes are normally segregating, and these are laggards. And in the lagging chromosome, as you can see, there is no localization of SENH3. Okay, so when SENH3 is not localized, the chromosomes cannot move and it gets gradually eliminated. Whereas in compatible cross, you can see that, the SENH3 is loaded normally and then it undergoes normal segregation. And similar is the case in the GFP tail swap elimination system also. As you can see, this is a compatible cross. As you can see, 10 GFP signals corresponding to the 10 centromeres, whereas this is an elimination cross. Instead of seeing 10, we see only 5. So that means it is not loading onto the other 5 centromeres. Okay, so now I have provided enough evidence to convince that we can mimic the consequences of distant crosses in an intraspecific cross just by modifying the SENH3, that is the centromere protein. Okay, so we do see all the combinations what we see, the genetic consequences what we have seen in the distant crosses. So with this, so the question now we ask is that we have so far demonstrated in Arabidopsis, and the question we ask is whether it is demonstrated in other crop species also. The answer is a mixed response. In certain crops, yes, and in certain crops, it's no. So these are the positive results. So SENH3 genome mediated genome elimination has known to work in wheat, maize, switchgrass, and cotton. Whereas in tomato and rice, it is working, but it is at a very low efficiency. So we have to tailor this method according to the crop species of interest. But it works at least in both dicots as well as in monocots. So again, apart from SENH3, there are after our discovery of the SENH3 mediated genome elimination, this has been reported. Several other genes as listed here are also good candidates to engineer uniparental genome elimination in different crop species. But most of them are either species specific, but we believe that SENH3, as you have shown from the previous slide, it can be applied both to dicots as well as monocots. For example, the matrilineal is known to work only in monocots, not in dicots. So again, there are issues with using these genes, but that's what I told, it should be tailored according to the crop species. So now I will switch gear and then I will give examples of how we can exploit or harness the power of haploid genetics in plants. So the first one is that to practice something called as analytical breeding. Okay, so what is analytical breeding? You know that a majority of our cultivated crops are polyploids, and doing genetics in polyploids is difficult because of the multiple alleles. Say for example, if you take a tetraploid, there will be 4 alleles segregating. So it will be difficult to generate a population and screen for the the transmission of the alleles. So what analytical breeding is aimed at is that you reduce the chromosome number of polyploids to a lower level and then breed at the lower level, and then after you select a desired genotype, then you convert back to the tetraploid, and this is called analytical breeding because we are playing with the chromosome numbers. So now we ask the question whether we can use this GFP tail swap. Hereafter I will refer to this as haploid inducer. So what we did is that we took a tetraploid Arabidopsis and we asked the question whether we can reduce the ploid to a diploid by crossing to the haploid inducer, and in fact, yes, we demonstrated that we can convert a tetraploid to a diploid. So to begin with, we had a tetraploid Arabidopsis, and then we crossed this one to haploid inducer to make a synthetic diploid, and this synthetic diploid again we crossed to haploid inducer to make a haploid, and the haploid, the diploid again can be crossed to a tetraploid to make a triploid, and in this way we made the different ploidy series from tetraploid to haploid, and this ploidy series can be used as a tool to decipher the effect of gene dosage on phenotypes. And with this, what we have demonstrated is that, so again, this tetraploid accession, what I was talking about, this is the inflorescence, and you can see the inflorescence is like the fruit of the Arabidopsis is called silic, and you can see the silics in the normal wild type, it's a bilocular silics containing 2 rows of seeds. Whereas in this tetraploid accession, we found an interesting observation that instead of bilocular silics, the fruit was showing tetralocular silics. So that means instead of 2 rows of seeds, we get 4 rows of seeds. And this is again an important agronomic trait, because you can double the seed set, okay, if you know what is the gene. But unfortunately, it is in a tetraploid and it makes the genetics difficult. So what we did is that we exploited this uniparental genome elimination, and we created the synthetic diploid, and using the synthetic diploid, we created a mapping population. And then to cut the long story short, what we have shown is that, in fact, so we narrowed down the gene, and we were surprised to see that it mapped to an already known gene called Superman. So as you can see from the name, why it's called Superman, because it's a floral boundary demarcation gene, or it's a cadastral gene, and the expression of which determines where the anthers and where the ovule should, or the gynoecium should be specified or delineated. So if you mutate Superman, so instead of female like the ovary, you will have only the anthers. So that's why it's called Superman. So basically what we have shown is that, in fact, this is an AP allele. That means the gene sequence is not changed, but the methylation status is changed. And as a result of this methylation changes, it is showing a superwoman phenotype. And basically what we identified is that it is a natural AP allele of a otherwise Superman gene. It turned out to be a superwoman. And excited from this finding, we found that we screened around thousands of genotypes of Arabidopsis, and we found 12 such accessions naturally having this phenotype. So again, this is one beauty of the haploid genetics. The second thing is that using this uniparental genome elimination, it's easy to swap the cytoplasmic genome and the nuclear genome. So basically, if you cross the haploid inducer as a female to a wild type male, so what you end up in getting a F1 paternal haploid. And if you do the reciprocal cross, you get a maternal haploid. So what is the difference between a maternal and paternal haploid? The difference is at the not at the nuclear level, but at the cytoplasmic level. So the paternal haploids, they contain the cytoplasm from the female and the nuclear genome from the male, whereas the maternal haploids contains the cytoplasm from the female and the nuclear genome from the male, both from the female. So basically, why it is where it is helpful, by just generating one paternal haploid, we can easily just swap the cytoplasm from one genotype to the other genotype. Okay, so using this method, so a Dutch group have nicely recently demonstrated that they could make a panel of hybrids. Okay, that is hybrid for cytoplasm. Okay, so they have taken 7 different accessions of Arabidopsis, and they could make a panel comprising of all possible combinations. And using these combinations, they have identified favorable cytoplasm nucleotype interactions that can be exploited for crop improvement. Okay, and apart from that, the uniparental genome elimination can also be exploited to achieve something that is impossible by diploid genetics. For example, there are class of mutation in plants called gametophyte lethal mutations. That means depending upon which gametophyte is inherited, it cannot be transmitted. If it is, for example, the one which I'm showing here is the gene called media. So media is a female gametophyte lethal mutation. So that means if the mutant allele is transmitted through female, then it will not be transmitted to the next generation progeny. Those seeds will be lethal, whereas it can undergo normal paternal transmission. So because it is maternal gametophyte lethal, it is impossible to obtain homozygous recessive sporophytes. Okay, so in that way, we cannot study the effect of the gametophyte lethal genes in the sporophyte. But taking the advantage of paternal haploids, since there is a normal paternal transmission, we could successfully generate paternal haploids, and then in that way, we can generate a paternal sporophyte. And using that, we have demonstrated that, see these media and another gene called Demeter, which is maternal gametophyte lethal mutations, they have a role in sporophyte. So I'm not going into the details for want of time. To cut the long story short, so when we generated the media Demeter double, which is otherwise not possible by diploid genetics, what we see is a twin seedling phenotype. From one seed, you end up in forming 2 plants. Okay, so what that indicates is that when we mutate media and demeter, okay, in the sporophyte, so there is altered stem cell fate such that, as you can see, several of the cell fate markers are misregulated or misexpressed, and as a result of which it ends up in producing an altered shoot apical, additional shoot apical meristem, and that give rise to the shoot apical, like twin seedling phenotype. And the thirdly, haploid inducer developed from one species can be used to generate haploids from other species. So we generated haploid inducer in Arabidopsis thaliana. So we asked the question whether by crossing the sister genus Arabidopsis suecica, which is having different chromosome number, we can generate haploids, and the answer is yes, as shown in here. And sorry. Yeah, so this is the haploid suecica, and this is the FACS data to show that it's in fact haploid. And then, so this is one thing. And another important thing to how we can exploit uniparental genome elimination is to combine the haploid induction with genome editing. So now we know that the genome editing, the CRISPR-Cas genome editing has won Nobel Prize a couple of years ago, and this is known to work in all species, including plants. So how we can combine haploid induction and genome editing? And this technology is called as HI-edit, which stands for haploid induction edit genome editing. Okay, so the major drawback in genome editing technology in plants is the transformation of the gene editing construct into the desired crop species of interest. And you know that majority of the crop species are recalcitrant for transformation. Okay, so here I'm taking the example of a maize into wheat hybridization, which is a genome elimination cross. So when we cross maize and wheat, what happens? Maize chromosome gets selectively eliminated, and you end up in producing a wheat haploid. So compared to maize and wheat, maize is easily transformed rather than wheat. So if you want to do gene editing in wheat, what you can do is that you can transform the wheat genome editing construct in the maize, and then you do the cross, that is maize into wheat hybridization, and then what happens is this. So you have a maize with wheat-specific CRISPR cascades integrated in the maize genome. You cross it to wheat. So what happened in the F1, the wheat gene gets edited. And then because of the genome elimination, what happens? The maize chromosome, wherein we have integrated the CRISPR-Cas construct, gets eliminated. And then within one generation, you end up in getting a haploid with edited gene and free of the CRISPR-Cas edited transgene. So this is a beautiful method that has been demonstrated to work in maize, into wheat, as well as in the rice as well. Okay, so in this way, with one step genome editing, you can produce like a mutation in your gene of interest as per your will and wish. And again, by using haploid induction, you can easily generate multiple mutant combinations. For example, so if you have a gene mutation at 5 different loci, and if you want to get a quintuple mutant, that means homozygous for all the 5 mutations. So by conventional diploid genetics, you have to screen around 1024 progeny, okay, to get one quintuple mutant. Whereas by screening the haploids, you need to screen only 32 progeny minimum to get one quintuple mutant. And imagine if it is 9. Okay, if it is for 9 different loci, you have to screen around 2 lakhs or 2.5 lakhs of progeny, whereas by screening 512 progeny, you can achieve the desired mutant combination in quickly. So this is another advantage, and this Karel Rihag group from Austria have demonstrated it for telomere binding proteins in Arabidopsis. And again, screening, for example, all recessive mutants can be screened only in the M2 generation. Just by using haploid induction, we can screen in the M1 generation itself, and this we have successfully demonstrated in Arabidopsis for phytoene desaturase gene as the proof of principle. And in addition, we combined the haploid induction with a diploid asexual gamete producing mutant. Okay, so similar to HI-edit. So what we did is that we combined haploid induction with asexual gamete forming mutant, and we generated the progeny exclusively from the diploid gamete producing mutant. So this mimics the apomixis. So basically apomixis is nothing but clonal reproduction to seeds. Okay, so this we have demonstrated in collaboration with Imran's lab from CCMB and Rafael's Mercier group, and it was published. So for more details, you can have a look at the publications. In addition, using the uniparental genome elimination, we can also practice some new breeding technique called reverse breeding. So what is reverse breeding? So in conventional forward breeding, you cross two parents, you generate F1 progeny. So what is reverse breeding? From the F1 progeny, you can recreate the parents. So that is reverse breeding. So how? For that purpose, we have to suppress recombination. And if you suppress recombination and you produce haploids, you end up in producing chromosome substitution lines. So by crossing two desired chromosome substitution lines, what you can do is that you can regenerate the parental combination by using the F1 progeny, and this is called reverse breeding. Okay, so again, this is feasible only because of the uniparental genome elimination, and this we have demonstrated again with a Dutch group, which was published in Nature Genetics. And so thus far, like I've shown that, okay, so this is in summary how we can exploit uniparental genome elimination for various plant breeding and genetic techniques, some of which I have gone through, and for want of time, I'm not going through the remaining thing. So with this, I will stop the presentation, and I would like to thank my PhD students and also all the collaborators from different parts of India as well as abroad, and also the funding sources, especially from DBT, Ministry of Education, DuPont, and iCURE NASF for grants, for supporting the grants. And with this, I'm happy to take any questions. Thank you.
So, thank you, Dr. Ravi, for your excellent summary of your work done during almost a decade. So now the session is open for discussion.
So I understood the SEN-H3 part because it's associated with segregation. Now, but you also said that there were other genes that were involved in it, right? Including a phospholipase and so on. So how do these play a role in doing pretty much the same thing?
Yeah, that's a good question. So actually, the phospholipase you are talking about, it is the main cause behind the STOC6 mediated haploid induction in maize. So I think if you are a maize breeder, you know that this has been discovered in 1940s. And people are trying to understand what is the gene that triggered this haploid induction in maize. But for 60 years or so, they were not able to identify. So when we discovered this in 10 years ago, we thought that this might be the culprit in maize as well. But it turned out that SEN-H3 is not the one. But it turned then later on, so just 3-4 years ago, 5 groups who have worked for several long years have narrowed down to this sperm-specific phospholipase. And here the mechanism is completely not understood, but it is completely a different gene. So otherwise, similarly, the other genes also have a different mechanism. So so the first thing, the classic textbook example of this uniparental genome elimination is the barley bulbosum method, which is an important, well-studied one in the intra-specific or distant hybridization technique. So most of the interspecies hybridization follows this SEN-H3 system, and we believe that SEN-H3 acts as an ID card for a species, because even a point mutation in SEN-H3 is sufficient to trigger this imbalance. So basically, this is one of the emerging hypothesis is that this enforces the post-fertilization speciation barrier. So if the chromosomes, they detect there is a mismatch at the SEN-H3, then that triggers genome elimination and it prevents the formation of the hybrid. So whatever escapes, we get as a haploids. So of course, each gene has a different mechanism, and not much is known, and now only for us at least we know that the epigenetic mismatch between the SEN-H3 triggers uniparental genome elimination that we recently published last year. But still, a lot has to be understood to completely understand the mechanism for which our lab is working on.
Very nice presentation, Ravi.
Thank you, sir.
You also mentioned that high HA edit has been achieved in rice.
Yes.
Can you elaborate on that?
Yes, sir. So rice actually, it's a Nature Biotech or Nature Genetics publication. So what they have done is that, for example, like if you want, if you generate a CRISPR-Cas editing line in one genotype, then if you want to edit, bring the mutation in another genotype. So simply what they have done is that, so they have brought this HA edit in the matrilineal background, which I was showing, he was mentioning about phospholipase. So in rice, matrilineal system works well for inducing haploids. So what they have done is that they have used the matrilineal mutant line along with the CRISPR-Cas edit, and then cross to a wild type genotype, and then they could generate a haploid without the CRISPR-Cas edited construct from the that HA edited genome, and then within one generation you can get the edited line free of CRISPR-Cas construct. So this is again, it's it has been successfully implemented and is practiced by Syngenta, but I'm not sure whether it is available for commercial use. And what I heard from recent presentation is that they are trying to bring it to the academic purpose or for regular breeding purpose for free. But that I heard for wheat, but I'm not sure about rice because it's developed by the private company, Syngenta.
Yeah. So thank you, Dr. Ravi. So now I request Professor Veluthumbi to hand over a memento to Dr. Ravi.
Yeah, thank you.
Thank you, sir. So now we'll have a break for tea for 10 minutes. I'll be back. We'll be back by 4:10.
Yeah, it's a short break. We have two more lectures, one from professor from Harvard University and another one from our senior professor, Professor Kaliraj. Please assemble back in next 10 minutes.
யார் வரணும்? சரி, ஓகே.
Sir, okay. We are experiencing some difficulties loading the session, then we will be there.
Very much. You can proceed, Professor, sir.
Do you see me? Do you see my screen?
No, sir. Yeah, your screen is visible, not visible in your presentation PPT, sir.
Yeah. No, I don't know. I'm not able to use the. I don't know how this is working.
You just click on the below on the 3-dot button, sir.
Three dots. Okay. Full screen. Is that working?
Yeah. Proceed.
Do you want me to begin?
Yeah. So there will be a formal. Proceed.
Yeah.
Sure,
So welcome, delegates. So the next speaker is David Reich from Department of Human Evolutionary Biology, Harvard University, USA. So he did a PhD from University of Oxford, and then had postdoctoral training at Harvard Medical School and Whitehead Institute at MIT. And then he's a recipient of National Academy of Science Award in Molecular Biology, Darwin Wallace Award for Major Advances in Evolutionary Biology, Herman Muller Award from Indiana University for contributions to our understanding of genes and society, Mastery Prize in Physiology and Medicine, shared with Vanto Pobo, and then Antiquity Prize for outstanding work in the field of archaeology. It's a smaller introduction only. So he has made a significant contribution in the area of human evolution and ancient DNA research and everything. So welcome, sir.
Hello.
Yes, Professor, we can.
It's a pleasure to speak with you. I'm sorry I'm not in person. I genuinely am. I would prefer to be there. It's also very early in the morning for me, so I'm sorry if I'm a little bit slow right now. I think that I've been asked to speak for a little bit less than a half hour, leaving some time for questions, about the work that my group and my colleagues and I have been privileged to do studying South Asian population history with genetic data. And so the way I'm going to frame this talk really is as a parallel story where the two stories I'm going to tell in this talk are those of the two large subcontinents of Eurasia, one of them being India and the other, or South Asia, the other being Europe, and they're about the same size. They've historically had approximately similar populations. And in fact, there's some striking parallels about their history in the last 6,000, 5,000 years. So the take-home message that if you only leave with one message from this talk is that Europe and South Asia have a remarkably parallel history. They're a parallel history of being mixtures of three major ancestral populations, some of which are non-indigenous and intrusive into the region within that last period of time. So I'm going to begin talking right now about the new technology of ancient DNA, which really became possible to use in a genome-wide scale over in a really proper scale only within the last 12 years, beginning in 2010. It starts with remain from ancient humans, or we could also study plants and animals, but this is a picture of a piece of the skull of a human, and in particular the inner ear part of the skull. And in a clean room, such as we have in our institute, and there's a number in India, we get beneath the surface of this remain, and we prepare the piece of DNA, a piece of bone that we think is going to be particularly rich in DNA. This is the cochlear structure of the petrous bone in the skull. We then remove some powder, so it's typically a tiny amount of powder, maybe only 40 milligrams, and then we release the DNA that's there in a solution that's designed to remove the protein and mineral content and the inhibiting substances that prevent the DNA sequencing reactions from going forward. And finally, we sequence it in some kind of next generation sequencer. This one that's pictured here is not the one we currently use. We use a more efficient sequencer than this sequencer here. And as a result of a series of technical laboratory innovations, as well as tricks, the amount of DNA that's been produced in this field has increased from the first data in 2010 by more than 1,000-fold since 2013 or 2012. And it's made it possible to ask and answer questions that weren't possible to answer before, because we can now peer back in time and get DNA from ancient people who lived at certain times in association with archaeological cultures and ask how they relate to each other and to people who live today. And the results are consistently surprising and not what are expected from most people's guesses about the past or reconstructions based on archaeology and history. Of course, there are many things that are right about those reconstructions, but they're often quite wrong about how people are related to each other. So it's clearly an interesting type of information. In South Asia in particular, there's very limited data, and that remains so even today, although there's a few small successes, some of which I'll tell you about. So I'm going to first tell you not about South Asian population history, but rather about European population history. This other subcontinent of Eurasia that is of about a similar geographic size and has a parallel history, and historically over the last thousands of years has had approximately a similar population within a factor of 2 or 3. So in Europe, we know a lot about the history of this subcontinent through multiple lines of scholarly study. One of them is, of course, archaeology, which looks at material remains from the past, like stone tools or other types of remains, pots, and tries to use it to try to understand things about how people lived even before the time of writing. And one of the things that archaeology has made clear, along with the technology of radiocarbon dating, which was really an explosive revolution in archaeology beginning in 1950, was the timing and path and impact of the spread of farming on this region of the world. Farming didn't develop for the first time in Europe. It developed for the first time anywhere in the world in the Middle East, in the eastern part of what's present-day Turkey, and in the northern part of Iraq and the Levant. And that happened 12 to 11,000 years ago. And then after 8,500 years ago, farming exploded into Europe. It exploded from Turkey first into the Balkans, places like Greece, and then it moved along the routes shown in these arrows to different parts of Europe. It took a fair amount of time and eventually reached the far reaches of Europe. So a question that's always been sort of present in archaeology is, to what extent did this movement of farming move through spread of people from the Middle East and Turkey, and to what extent did it spread through copying of these technical innovations that allowed people to extract more energy, calories, resources from the landscape in which they lived and live at higher densities? How much did that was that copying, similar to the way that people today copy use of other innovations like cell phones, for example, even though it's not spread through movements of people? Another important line of evidence is related to the spread of language. So almost everybody in Europe today speaks a set of closely related languages called Indo-European that go back to a common ancestor, maybe 6 to 7 to 8 to 9 to 10,000 years ago. And these languages also include languages like Persian in Iran and Armenian in the Caucasus, and also the languages of most of the people in India today, especially in northern India. So it's a mystery, and it's been a mystery for more than 200 years, how these languages came to be so closely related. We know something approximately about how fast languages change, and and from that we can know it's not too far in the past that they've shared a common ancestor. So a question has always been where these languages were spread from. So here's what ancient DNA has shown. So in 2014, we had data from very, very few individuals, and for historical reasons, the community focused first on the early European farmers and hunter-gatherers. They studied Europe first because this technology was invented in Europe, and that's the samples that people had available, and it's now spread more broadly. And 5,000 years ago or more, if you look at the vast majority of people in Europe, these bar plots show data from real people, and they show the proportions of DNA ancestors that in each of those people that come from different populations. And you see only two components in these bar plots here. There is the dark blue component, which is related and almost certainly derived from Anatolian farmers from Turkey who spread into Europe after 8,500 years ago. And then you see this bright green component, and this is the indigenous population of Europe, which varied in its contribution to these different people, farmers in different parts of Europe. So in the far west, furthest from the Middle East, typically these proportions were largest, in this case 30%, and also in the southeast, like in Greece and Bulgaria, it was very small, the mixture with the local group. So it was a mixture of these two groups, and importantly, most of the DNA was coming from these incoming farmers. So it seems that farming spread largely through movement of people, not through copying. Although some people did copy, like the ones in Spain, there was a lot of local retention. So what's really amazing, and what we already knew in 2014, is that that situation 5,000 years ago is profoundly different from what we see today in Europe. And in Europe today, if you take modern people from different parts of Europe, the bottom ones, the outliers, are from Sardinia, this isolated island in the Mediterranean Sea. But the others are different populations from all different parts of Europe. And the biggest component of contribution to many of them is this red component that simply did not exist in most of Europe more than 5,000 years ago. So something really big happened, introducing a new source of ancestry that wasn't there before. So I'll show you what happened. This is the result of a whole series of studies that have piled onto each other and have provided really an exquisite picture of this profound transformation that happened in this region of Eurasia. So what I'm showing here is data from about 1,000 individuals from the dots shown on the map. So this is a map of Europe, just for orientation. That's Arabia, Iberia, Spain, Great Britain, Scandinavia, Iceland. And the dots up here are Northern Europe in red, and blue in Southern Europe. And the squares over here are from the Middle East. And what you're seeing here is the analysis where we actually don't use the locations or the labels of what populations people are from at all in the analysis. All we use is the genetic data. And so the way the genetic data looks is we're looking at about 600,000 positions of our DNA that where people vary. They're called, as you probably know, single nucleotide polymorphisms. And some people might have 2 adenines, some people might have an adenine and a cytosine, some people might have 2 cytosines. So it's 0, 1, or 2, what you have. So you should think of the data as 600,000 rows corresponding to each of these polymorphic positions, and about 1,000 columns corresponding to each of these individuals. And the cells of the table are 0, 1, or 2. And you multiply this matrix or table by itself, and you get 1,000 by 1,000 table measuring how closely related individuals are to each other. And then you run a simple mathematical technique called principal component analysis that most efficiently separates these 1,000 individuals from each other based on how much variation is explained. And what you see here is a plot of an x-axis, which is the combination of those positions in the genome that explains the most variation, and a y-axis that the positions that explain the second most variation. So the x-axis might be 0.1 times your value at position 1, 0, 1, or 2, plus 0.3 times your value at position 2, minus 0.4 times your position at 3, etc. And so when you do this, you see this amazing pattern. And the amazing pattern is 2 parallel gradients. One is the Europe and the other is the Near East. And there's a gap in between with very few populations. And the populations that are in between are ones of plausible, more recent contact between Europe and the Near East. And in the top is the non-Mediterranean northern populations. The bottom are the Mediterranean populations. And what you can do is gray out those spots and see how, same spots, but they're now gray, and see how the ancient individuals fall using the variation learned from modern individuals. And what you see is a really amazing pattern. So if you look at the hunter-gatherers of Europe before 8,500 years ago, they fall here. So this is kind of an amazing place for them to fall. They fall beyond European variation in the direction of European differentiation from the Near East, as if Europeans are a mixture of a group related to like this and a group like this. And that's actually what is the case. Next come the European farmers after 8,500 years ago, and they fall in an extremely different place. And that's because they're migrants from a part of Turkey, part of the Near East, and they pile up at the bottom of the European variation you see today. And that's reflecting a large scale migration. The first ones are to the right. The later ones that mix with hunter-gatherers shift this way toward the hunter-gatherers, and they shift onto this gradient. Next, 5,000 years ago in the steppe north of the Black and Caspian Sea, present-day Eastern Ukraine, you see groups like this, and they are far at the top of this present-day European gradient, but you still don't see people like Europeans today. So this happens 5,000 to 6,000 years ago, but in Europe itself, you still don't see anybody like that. You see almost only people like that. And then after 5,000 years ago, bang, you get European variation today forming. So something really big happens after 5,000 years ago. So to summarize what happens is that Europe is massively transformed in this period by two migrations, large-scale migrations. The first is bringing farmers from the Near East with an average of about 80% arrival of new DNA. So the resulting people are 80% from these Turkish Anatolian farmers and 20% from the local population, varying from one place to the other. And the second big finding is the arrival of a new group of people, this red component that is so important today, and that happens after 5,000 years ago, and it results in up to 70 or 80% arrival of new people in some parts of this subcontinent. These people, here's a skeleton of one of these people, who are called the Yamnaya, and they're quite important archaeologically. They're the first people in the steppe to use the wheel and the horse, at least this part of the steppe, and they probably used the newly domesticated horse, which they may even have domesticated themselves, and they hitched them to wagons to bring supplies out into the open steppe lands and to exploit the vast grassy landscapes. And to use horses and wheels to do that more efficiently than people had done so before, and they expanded extremely dramatically. So whereas previously the steppe was a patchwork of many cultures that differed from each other very dramatically in terms of the types of pots they made, after these people spread all the way from Mongolia to Hungary and Europe, they almost ran over all these previous groups, and you see a large region of homogeneity where people with similar behaviors, and as reflected in the types of pots and artifacts they made, spread over this vast region. They were economically very successful. So how does Yamnaya ancestry spread? Well, it spreads west. And here is a picture of what happens in Britain. So this is the period from 6,000 years ago to 3,000 years ago. And if you look at the proportion of DNA from people like with ancestry from these Yamnaya people, there's none of it from when the farmers first get to Europe, Britain about 6,000 years ago to 4,500 years ago. So the last big stones at this famous archaeological monument of Stonehenge that you may have visited if you visited Great Britain in the south, go up at the end of this period, built by people without any ancestry from the Yamnaya and from Eastern Europe. And then after 4,500 years ago, bang, everything changes. And there's a minimum 90% arrival of new people. And what I'm doing here is I'm coloring each of these dots by the Y chromosome, which people inherit from their father if they're a male, and the empty dots are female individuals. And what you see is there is also a very radical change in the type of Y chromosome people carry to a red type that's typical of the East and wasn't present in the region before. So here's what happens in Spain and Portugal. So same period of time when there's no steppe ancestry, and same period of time when steppe ancestry arrives. But here there's only a 40% population replacement. And however, there's a complete replacement of the Y chromosomes. The dots all turn red. And what's actually happening is the incoming people from the East, males mix with the local female population, and most of the resulting population derives its ancestry from the local population. However, the incoming males essentially almost completely outcompete or displace the resident males. They almost completely fail to reproduce. And so this suggests maybe something about the nature of the interaction between the incoming people and the local people. So now I'm going to switch to the other subcontinent. That's the focus of my talk, South Asia. And I'm going to begin by talking about what we collectively have learned about South Asian population history before ancient DNA data. And I'm going to focus on work I've been involved in. There's been lots of really important work, much of which I haven't been involved in, but I've had the privilege to work with several groups in India, and especially with Kumarasamy Thangaraj and Lalji Singh at the Center for Cellular and Molecular Biology in Hyderabad, which we first began publishing together in 2009, and in this paper. And in this paper, which was an early paper using genetic data to study human variation history, even before the ancient DNA revolution, we started by studying 25 diverse populations around the world, around India. We Thangaraj picked them to be as diverse as possible, and we applied the same methodology I mentioned to you before to carry out a principal component analysis of their variation. And what you see here are the 25 Indian populations all up here, or nearly all up here, 23 of them. And then you see 2 West Eurasian populations, a European origin population, 2 European origin populations, and here's some East Asian populations. There's Chinese here, as well as some groups in Northeast India, 2 groups in Northeast India, which are genetically similar to the Chinese, or distantly similar to the Chinese. But the really amazing pattern that we perceived was what we called an Indian cline, a gradient of different degrees of proximity of groups in India to West Eurasians, Central Asians, Near Easterners, and Europeans. And it's very tempting to think that this might reflect some kind of mixture event where these groups have different proportions of ancestry from groups related to these. There's also really interesting history going on with these groups that are off-cline, like these are often, but not always, groups that speak Austro-Asiatic languages, like Munda languages and tribal groups. They're super interesting. We and others have written papers about them, but I'm not going to talk about that today. So the model for this mixture that we proved in 2009 was that a massive mixture indeed had happened, and that Indian populations today are mixed. Almost all populations in India today are mixed between 2 highly divergent groups. One we call the ancestral South Indian, the other we call the ancestral North Indian, and the proportions of mixture differ in different parts of India, and that they have not completely homogenized. So just as in Europe, people have different proportions of these bars, here people have different proportions of these ancestries from these proposed 2 ancestral groups. So we and others have estimated the proportions of ancestry from these 2 ancestral groups, and this was our estimate in a paper from a graduate student in my group, Priya Murjani, from 2013, from a series of groups. And one interesting feature of the proportions of Western Eurasian related ancestry, which range in this estimation from 70% to 30%, is that if you look at the people with relatively more Western Eurasian relatedness, they tend to be speakers of Indo-European languages like Hindi, and people in the speak Dravidian languages tend to have relatively less. And even within a region, even within a state of India where people all speak one language or not, people who have relatively more West Eurasian related ancestry tend to be of traditionally higher social status in the traditional caste system, and people who have relatively less. So both caste and language, and these are statistically independent signals, and it's both significant. So caste and language both seem to be independently associated with the mixing process. So that tells you that the mixing process happened in some way in the those cultural phenomena happen in some ways in the context of this mixing. So what's the history behind this mixture? So we entertained 3 very rough hypotheses that of course the whole world was transformed by the last ice age, which ended after 14, maybe somewhere between 14 and 19,000 years ago, when you start thinking about when the warming happened. And landscapes changed everywhere, even in places that were not glaciated, like South Asia, but the landscapes changed in terms of their forest cover and so on, and ecology and the types of life they supported. So maybe there were post-ice age migrations that brought these groups together. Well, maybe it's the arrival of agriculture in this region after 9,000 years ago. The earliest agriculture in South Asia is around the Indus Valley, coming from the west, from the Near East, same time approximately as it arrives into Europe, from that same region. And then the peninsular part of India, agriculture spreads about 5,000 years ago. And actually the impact is both from the west and from the east, as well as from local domestications. So East Asian agriculture, which is also quite early, has a major impact, but also there's local domestication. Or maybe it's related to cultural phenomena such as the archaeological changes associated with the Indus Valley civilization and the rise of cultures in the Gangetic Plain, after 4,000 years ago. So what do we do? What we would try to do is estimate the date when this mixture occurred. And we did this by the following methodology. So this bars are supposed to represent 2 chromosomes. So, you'll have every person has 23 pairs of chromosomes. So this might represent 2 copies of a person's chromosome 3. And here's a person who has 2 ancestral North Indian, more West Eurasian related chromosomes. Here's someone who has 2 ancestral South Indian. And if they have a kid, they'll have 1 entirely ancestral North Indian, say from their mom, and 1 entirely ancestral South Indian, say from their dad. So if you look at someone who has 8 great-great-grandparents 4 generations ago, maybe they'll be mostly ancestral North Indian, and some of them will be ancestral South Indian, and some of them will be a first-generation mix. In every generation, you make 1 or 2 recombinations and send that DNA down to your offspring. And you send mixed chromosomes to your kid. So this breaks and produces a spliced first 2/3 red and next third blue. And then do that again, splice, 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 until you have today this mosaic genome, mosaic chromosome 3, that has these chunks of red and yellow ancestry. And so what you do is you just measure the average chunk size. And it's kind of like the recombination process after mixture. It's kind of like a knife chopping vegetables, and the vegetables get smaller and smaller at a regular rate over time. And you just look at the size of the little chunks. And if they're very, very small, the mixture started a long time ago, and you know exactly how long. And if they're quite big, you know it was quite recent, and you can estimate how long ago. So what we found is that the mixture occurred about 4,000 to 2,000 years ago. And so the summary of this is that before ancient DNA is that South Asians we were able to show were a mixture of at least 2 populations, ancestral North Indians and ancestral South Indians. And they mixed very dramatically between 4,000 and 2,000 years ago in a way that's related to caste and the spread of Indo-European languages. So what happened next? So now I'm going to talk about how Yamnaya ancestry spreads east. So this is a paper we published in 2019 along with a twin paper based on a single ancient genome from India. One of the papers had about 530 genomes, ancient genomes from the red dots from Central Asia and also from northern Pakistan. And the other one was a single genome from a site called Rakhigarhi, which was a Harappan site in South Asia. And we studied more than 260 modern Indian populations for comparison. So a very complicated paper, and I'm just going to focus on one aspect of the results, which is the data from these sites over here in Uzbekistan and Turkmenistan, which are from a civilization called the Bactria-Margiana Archaeological Complex, which built these immense immense walled, often walled towns, very large, in Uzbekistan and Turkmenistan about 4,000 years ago. And here's one at the site of Gonor. And this whole civilization was maybe the last great civilization of the ancient world to be discovered by archaeology. It was discovered by Soviet archaeologists in the 1970s. And so what we see is that most of the people from the graveyards around sites like Gonor are in a big cluster in a principal component analysis. But there's all these outliers, people with different ancestry. And what you're seeing is cosmopolitanism. You're seeing that it's a little bit like you sequence DNA from a city like Tokyo, and the great majority of people are all going to be in one cluster of ancestry, genetically similar. But if you have enough people, like we do now with ancient DNA with big sample sizes, you also see outliers, people who are different from the main group. And so that tells you who the people in Tokyo are communicating with. You'll see people with Chinese ancestry that might reflect economic relationships, and Korean ancestry and South Asian ancestry and European ancestry, and that tells you where the connections are. So in the same way, that's what we did here. So most of these people buried in these graveyards in this blue cluster, genetically similar to ancient Iranians. But this ancestry related to Siberian foragers arrives before 4,000 years ago. It's the only type of outlier ancestry you see. And then after 4,000 years ago, you start seeing ancestry from Yamnaya buried in these outlying cemeteries in mixed form, often with these Iranians, showing that these people had started arriving after 4,000 years ago. And we also see it in our ancient DNA from more than 100 people from the Swat Valley in present-day Pakistan, more than 30, shown by 3,000 years ago. And with the mixture chunks, we can see it arrived more than 3,500 years ago. And we also detect over here these outliers with South Asian ancestry. So there's also migration from the south impacting Gonor and similar sites like this. So what we were able to reconstruct are 3 layers of mixture with the ancient DNA. We only could perceive 2 layers before with the modern data, but now we see 3 layers. And if you look at the data we have from this time, we have data from these outliers with South Asian ancestry that buried at Gonur, and we think of them as probable immigrants to sites like this, Gonur, or another site called Shari Shokta in eastern Iran. And we think of them as probable immigrants from South Asia based on their South Asian ancestry and other aspects of their DNA profile, which is very distinctive. And we confirm that also by seeing that the Harappan individual from India is quite consistent with them. So 4,000 years ago, there's a gradient of ancestry in this triangle ancestry between these three sources, Iran and peninsular India, with people with relatively more Iranian ancestry and people with relatively more Southeast Asian tribal Indian related ancestry. And you see this gradient over here. So then after 4,000 years ago, that gradient becomes completely different. And you see these green types of ancestries, and that's when these individuals from Pakistan mostly, as if a point at this gradient over there, before 4,000 years ago, started mixing with a point over here, steppe Iran mixture. And then mixtures of these mixed ancestries. So the ANI, the ancestral North Indian, forms as a mixture of these Indus Valley type individuals and steppe pastoralists after the decline of the Indus Valley civilization. And then another group forms we call the ancestral South Indians, and they form as a mixture of these and presumably unsampled groups, which could be studied by analyzing ancient DNA from the right period, presumably from southern peninsular India over here. And then what happens after that is mixture of these mixed populations forms the modern gradient in South Asia. So you have this parallel history where you have this hunter-gatherer local population, maybe it's here. You have this farmer population, maybe, or maybe not, which is Iranian. I actually don't think it's likely to be a farmer population in some ways. And then you have this steppe ancestry coming in. So the modern Indian gradient is sex biased, relatively more Y chromosomes from this new population than mitochondrial sequences, suggesting that this was primarily coming in through males. Although today it's only a small proportion of ancestry, at most 20%, in some groups close to 0%, and it's not as an extreme a sex-biased event as you see in Spain. Also, you see that people who are Brahmins and Bhumihars today, especially in some parts of India, have significantly more Yamnaya ancestry than other groups in India. So these groups over here, the ones that are off the gradient, have too much steppe ancestry compared to what the main gradient is. And if you rank people by how well they fit this line and color them by traditional priestly status, the ones that are most misfitting are people who are Brahmins and Bhumihars, with some exceptions, especially in parts of southern India. So what we see is two parallel subcontinents in Eurasia, Europe, and India. This gradient is showing the spread of farming. Farming spreads out of the Near East after 11,000 years ago, arrives in Europe and South Asia after 8,500 years ago, and then and it spreads into the peripheries of these places somewhere between 7,500 and 5,000 years ago. And then meanwhile, steppe pastoralists begin spreading after 5,000 years ago and impact Europe after 4,500 years ago or 4,700 years ago, and impact South Asia after 4,000 years ago. I think there's an aspect to this model which is wrong, in the sense that we now no longer think that this is actually Iranian farmer ancestry coming into the region. We think that this is probably an indigenous Iranian-related Indus Valley hunter-gatherer population that's contributing this second layer of ancestry. And then after 4,500 years ago in Europe and after 3,500 years ago in South Asia, you get this mixing of populations. So there's extraordinarily high population substructure in India today. So if you look at pairs of groups, in India, the differentiation is measured by a classic measure of differentiation, is about 3 times higher, and it's not related to if you control by state or by language group or traditional social status, it's still much higher than in Europe. And so what's going on? So the right way to think about this, I and many people think, is that India is not really well thought of as a large population. I know there's 1.4 billion people, and I'm not saying that's a small number. I know it's a very large number, but instead there's thousands and thousands of relatively small and sometimes medium-sized populations. And that these groups are to a large extent, or at least have historically been separated from each other by traditions, which mean that there's relatively little mixing with each other. So we know that the founder events are old. So for example, Vaisya from Andhra Pradesh, we think from the genetic pattern are 2,000 to 3,000 years old. And it's been very clear from looking at this population structure, which persists from thousands of years ago, that this endogamy that you see in many groups today is quite old in some groups. So some people, for example, Nicholas Dirks, as art anthropologists, have argued that caste in modern India is invented by colonialism in the sense that it became more rigid under colonial rule. But genetics shows that many current distinctions among castes are ancient, and strong endogamy must have shaped marriage patterns for thousands of years. So in summary, what genetics has done is it's documented a cultural change in India. After 4,000 years ago, there's this convulsive mixture to form the ancestral North Indians and ancestral South Indians, who are in turn mixed from three ancestral populations: the Iranian-related group, probably from the Indus Valley region, the Southeast Asian-related indigenous group, probably from the peninsular part of India, and the steppe group that comes through after 4,000 years ago. And then there's a profound switch to endogamy that's partly intact today. So this is also a huge medical opportunity in India. An example is that a third of Indians are from groups with clinically significant founder events. There's a paper we were involved in 2017 that showed this. So we looked at almost 300 groups, and their ranked order from the strength of their founder event, from least to strongest. This blue bar is the strength of their founder event. And in red, I show the Ashkenazi Jewish groups and Finns, which are two groups in Europe that have very strong founder events and result in a high rate of diseases, of rare diseases in the population. I'm from one of these groups, Ashkenazi Jews, and I've had a cousin who died of one of these diseases, and my mother had a cousin who died of one of these diseases. So these are really common and serious issues. And the rate of these diseases is related to the size of your bar. So a very large number of groups in India have much even stronger than Ashkenazi Jews, who are very well studied in the West and seen as a paradigm case of this group very severely affected by these diseases. And in couples who are both from this group, such as my wife and me, we got tested for these rare diseases that are not so rare in our community. And these are not just small isolated populations. So there's ones with millions of people, Reddys and Kumars, Baniyas, and so on, Vaisyas. And there's anecdotally known recessive diseases like butyrylcholinesterase deficiency, bad reaction to certain kinds of anesthetic, in, for example, the Vaisya, that people anecdotally know about. But someone should really systematically study this in each of these groups that are known to have strong founder events and come up with the types of interventions that are so effective in my community. So in my community, we basically know all the rare recessive diseases. Couples in thinking of having a baby can get tested for them, or if they have already conceived, can test the baby. And if you're in a group where there's arranged marriage, such as is the case in religious members of my group, people may not even be introduced to each other if they're genetically incompatible from the perspective of one of these diseases. In India, there's predicted to be 100,000 cases a year of these deadly recessive diseases, and they could be essentially completely avoided through awareness of these issues. So it's a real medical opportunity. And in my community, again, in the religious community, where there's prenatal testing, where there's matchmaking and arranged marriages, in those communities, these diseases are almost completely gone. So this is a real opportunity. Thank you. I don't know how we do it online. Maybe I can answer questions, or you can write to me or something like that. Thank you.
So the session is open for questions.
Dr. David, am I audible to you? Hello? I think the laptop you need. Okay, yeah. Right. Yeah. Thanks, Dr. David. So I have a question. Am I audible to you?
Okay.
Okay. Okay. Right. I have a question. Yeah. Right. Yeah. So, right. Right? Yeah. What is your suggestion to reduce this prevalence in future?
So if I was, I think this is the biggest medical opportunity I'm aware of in genetics, and anywhere in the world, actually. And my although some people are working on it, I think many people more should work on it. So what I think should be done is people should study Indian groups that are susceptible to this the same way that groups like my group have been studied, like Ashkenazi Jews or people in Finland. And it's actually not so expensive. So we know that, for example, if you wanted to find all the rare diseases in my group, for example, what you would do is you would sequence the exomes of 2,000 people, 200 people, and you would find all the rare mutations and you would see them because they would be predicted to be pathogenic. So then you would have them all, or you'd have almost all of them. Or you could sequence the whole genomes. So what you should do is take every group in India, 4,000 or 5,000 or 6,000 of these endogamous groups, or whatever the number is. We actually know a lot of this, what the groups are. Identify 200 individuals and whole genome sequence them, or exome sequence them, but probably whole genome sequence them these days. It's not so expensive. And run the data through a disease predictor, find variants that are very much more common in these populations than in other populations that are predicted to be pathogenic by interpreters of the genome, and you'll have a list. That's one way to do it. Another way to do it is to do community engagement in these communities and figure out what diseases are common in these communities and collect cases. That's the opposite sort of way of doing it, and it's been very effective. And that too could be a very effective approach. Probably both approaches are valuable.
Sure. Thank you. Exome sequencing for 40 cases from South Indian population. As you said, I just move on. I will just try to do more whole exome sequencing. Thank you.
Yeah, and I think it would be good to focus on, I think there's value to doing sufficient data from specific groups. So for example, in our 2017 paper, Nakatsutka et al. With Thangaraj, and there's about 70 or 80 groups with very strong founder events that must have many rare recessive diseases. So I would pick a group that had a significant groups that were demographically large and had predicted very strong founder events. And I would focus on exome sequencing or whole genome sequencing in one of these groups, or several of these groups. And there's a very, very strong prediction that you will find many, many significant variations that are both interesting biologically, but also very significant and important to these communities. It's also like, as a funding issue, I think very, very obviously a good idea. So, these communities, I mean, when we published this work in 2017, even me, who's not well connected in India, people, say, oh, I'm a Bania, Mark, we are a kind of merchant community, we're well off, and, we have a founder event, 10 or 20 times stronger than Ashkenazi Jews, according to your paper. Is there a way we could get involved in supporting this type of research? I didn't pursue that, but there's so much opportunity of that nature.
Sure. Thank you. Thank you so much for your nice presentation.
Thank you.
Yeah, thanks a lot. So
Goodbye,
that
everybody. Bye.
Yeah. Yeah, bye.
So on behalf of the organizing committee, we convey our sincere gratitude to the chairs, including Professor Ashok and Professor Rajendran. Thank you so much, sir. And now may I call upon Dr. Geeta from Sociology to chair this session.
Good evening all. I am happy to introduce the speaker of this particular session, Professor Kaliraj Perimal, ICMR Emeritus Medical Scientist and former Vice Chancellor of Anna University, Vice Chancellor of Bharatiyar University. He is right now Professor and Director, Center for Biotechnology, Anna University. And it is so very interesting to know from his biodata that he is a alumni of Madurai Kamaraj University, where he did his UG program. To his credit, he is having citation of 1937, H index of 21, I-10 index of 45, and his research area is biotechnology, molecular immunotechnology, recombinant technology, parasite vaccine technology, and he had received awards like, he had received so many awards like BRSI Industrial Medal Award of Biotechnology, Research Society of India in 2003, and Professor B.K. Ikat Oration Award of ICMR in 2002.
Next is
he is also a fellow of American Society for Tropical Medicine and Hygiene, USA Royal Society of Tropical Medicine and Hygiene of UK. I welcome you, sir, to enlighten us with your view on disruptive reforms in research in era of Industry 4.0 and 5.0. Over to you, sir. Thank you.
Thank you. I sincerely thank the organizer to invite me to talk about the disruptive reformation. I listened to many of the lectures. It has been done with excellent research work, with a lot of data. So we would like to go for the translational research also, which is an important aspect. Now, India consolidates its position as one of the global innovation powerhouses. It is a responsibility of the universities and academic institutions to strengthen the research and also the research to reach the society. And if you just look into the innovation, it might be a simple innovation like our Aadhaar card. Covaxin is another important example of innovation in a short period. Then innovation in space technology, healthcare, smart cities, and telecommunication. Now, what is the scenario of Indian universities in technology transfer? This evening I was talking to one of the faculty members. He's an excellent worker, but he's finding it very difficult to reach the industry and translating his research into a tangible product, which is an important aspect. We have a lot of money is being invested on basic research, which is must for innovation. But ultimately, if you don't translate your innovation, it is not going to be useful. Now, what has to be done? What good practice you have to do to inculcate the innovation? The intellectual property awareness has to be taught in school level. How many of your parents are asking what you have done today, what kind of research you have done today? But if you go to your school-going children, every day you'll ask whether you have completed this or that or something, what is your marks? And if you are teaching, many of our parents, they are teaching their children about various subjects. So the scientists who are teaching, if there is an IPR awareness, for the school children, even most of our faculty members also will be reading. APJ Abdul Kalam, he was in Anna University for a couple of years. His target is a student population. He claims, unless I make their mind, it is not possible to make them best when they are growing. Even he was promoting the translation research to the school children. Now, what is the situation of patent? Now, from 2009 to 19, a large number of applications have been filed. For a long time, people used to ask, how many patents you are having in the biodata? They will give some number. It is a filing number, just like a dispatch number. and only very few have been awarded. Out of that, how many have come to the market? What is the use of patent? It's a paper. You can't need the paper. Very few has come to in the form of technology transfer. So a large number of steps have to be taken, starting from acceleration of translational research up to the adding to socio-economic value. Now, what is the future of science, technology, and innovation? We have to go for self-sustained resources like world's energy, water, food, and sanitation needs. Automation has become an important. தம்பி கொஞ்சம் பேசி முடிக்கிறியா? I will continue after your. முடிச்சாச்சா? பேசி முடிச்சாச்சா? Thank you. So, the automation has become an important aspect, starting from self-driving cars up to the, things like machines that read the X-rays, then experiential learning using augmented reality, and good health or improvisation. Now, how our education system has to be organized. The education is for society, because we all earn from the public money, whatever the project we are getting. So you have to give back to the common man. And from time to time, the education has been mapped and prepared, curriculum and syllabus has been prepared based on the industrial need, industrial revolution. Now we are in the fourth industrial revolution, that is a digital transformation. Now what you have to do for making our studies and education matching with the fourth industrial revolution. And we have several industry 4.0 components like Internet of Things, big data, augmented reality and all. All these components have to be incorporated into the subject. So we have written 5 textbooks on the research on Industries 4.0 and the use of Industries 4.0. And 4 books have been already released by the governor. The 5th one has already published, and it will be released shortly, and it has been prepared by Taylor and Francis. This is a basic textbook now, it is available. How you are going to use various technology in various disciplines. Now in Bharatiya University, in a short period when I was a Vice Chancellor, we revised the syllabus even when the NEP was in the draft stage. How you are going to introduce all these data? We introduced the introduction of Industry 4.0, artificial intelligence, robotic process in all departments and all the courses, including Tamil linguistics. And big data and data analytics have been introduced in many of the subjects. IoT has been introduced in many subjects, and augmented reality has been introduced. And there is a need for adopting the technology for the improvement. When the vaccine was developed over a period of 15 years, now the technology is available to prepare in 6 months. Covaxin is the best example. Now, each university, they should have the international collaboration, and they should have some of the courses not only for our students, present students, even for the alumni. When they complete the course, when they are going to the industry, he has to update the knowledge from time to time. So we have the collaboration with Harvard Business School in Bharatiya University, where many of the skill courses as well as the knowledge generating courses are being taught. And another important thing is the skill courses for enhancing the employability of the students. The basic subject is a foundation. Then you need a good interior. And if you are a good interior designer, you'll be able to make any one of the industries or any one of the institute into the best possible manner. While doing your MSc course or MA course, you should have at least 2 to 3 job-oriented certificate courses. Again, in Bharatiya University, we have started a large number of skill courses in collaboration with the industry. Both the industry people as well as the institute, we take care of that. Take for I'll give you, in Arbito Asia, we are teaching the molecular techniques. A MSc student doing biotechnology or zoology or something, he might not be having the opportunity to go for molecular techniques. We teach over and above the MSc. Similarly, so many other subjects with industries. And even we have the UG program with industries. One of the UG programs that we are having which is very popular is BSc Manufacturing Science for the employees of Tata Electronics in Hosur. Government of Tamil Nadu has given about 600 acres land in subsidized price. With the understanding is they should recruit especially the girls from the underprivileged area and offer them the job, some 10,000 rupees to 15,000 rupees. And it is just like the strategy of China to have the cheap labor. And in Hosur, they started assembling the many components, and shortly they will be going for manufacturing the semiconductor. In Coimbatore, they are going to have 2 or 3 semiconductor manufacturing industries. Now, in all these branches, the technology is getting changed and the expertise is getting changed from time to time. You take a student from the school or college, within 2 or 3 years, they become obsolete. So what they are doing is, along with Bharatiya University, we started BSc Manufacturing Science to the people who have come with the 12th standard background. We enrolled about 1,000 students who have gone for job in this one, Tata Electronics, with the government of Tamil Nadu support. And the company pays for that from the corporate social responsibility. And this particular course, by the time the girl is completing in 3 years' time in the particular industry, she will be having BSc Manufacturing Science. In another 3 years' time, the electronics industries are going to play a very important role. Wherever she goes after marriage, she will be able to get some job. Similarly, we have so many other courses. We do it with industry, and the industry knows what they want for their company, and it is very popular. Now, looking about Industry 5.0, now we have to talk about Industry 5.0. You have the physical world and virtual world. There is a lot of problem in handling the automated equipments, understanding the equipments. So Industries 5.0, they talk about the interaction between the human and machines, how to handle it. You might be having the technology, you might be having the software, you might be having good equipment. How you are going to effectively use it to produce the goods in reduced cost, because you have to compete with the people. Then empowered workers and attracting the best talent, skill development training, competitive edge, safety and well-being. These are the components of Industries 5.0. When you are adopting Industries 4.0, you have to go for Industries 5.0 for effective use of that. So here there are a lot of advances in the Industries 5.0, like individualized human-machine interactions, bio-inspired technologies, digital twins, and so on. Now, the recent advances is linking the physical world like bio-inspired technologies and smart materials and energy efficiency, autonomy and all. That is in the physical world. And you are having the virtual world where you are having the data transmission and all. So the Industries 5.0, they intertwine both virtual world and physical world so that the ecology and society, they will be able to come for the better economy. Now there are challenges also. Industries 5.0, especially the productivity is required while large investments are needed. That is an important aspect, but there is a need for the better translation. What is the impact on work? How the jobs are going to be in 2030? We have to prepare our students who will be suitable for the jobs in 2030. We are going to have leadership or the leading